Hello, everyone. We'll be starting with the research symposium in 12 more minutes. Thank you for joining. I would like to welcome those who have just joined the session. We'll be starting in more 10 minutes. And in the meantime, you can introduce yourself in the chat box and let us know about your educational or research background. Thank you. A very warm good morning, good afternoon, or good evening in whichever part of the world you are. I would like to welcome you all to the much awaited event of the year, Omics Research Symposium. Before we get started today, I would like to introduce Omicslogic. To begin, we must ask, what is bioinformatics? It is the development, implementation, and usage of methods and software tools for understanding biological data. Bioinformatics has practical applications in healthcare, agriculture, and mitigating climate change. As this field progresses, bioinformatics poses several challenges for both students trying to learn it and faculty trying to teach it. In this presentation, I will explain the current challenges and limitations in bioinformatics, describe how we aim to address these challenges with the Omicslogic portal, describe Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework, explore the Omicslogic portal and how it utilizes Bloom's taxonomy in structuring its course, pipelines, and projects, and finally describe several outcomes of the Omicslogic portal. I'm Sonalika Ray, and I'm the Omicslogic Community Manager. I'm responsible for daily interaction with thousands of users that are a part of our bioinformatics community, which is managed by Pmi Biotech. Over the several years, we have had the privilege of working with a number of organizations developing a training program for research-oriented students, faculty, and democratizing bioinformatics, making this discipline accessible to thousands of students and faculty around the world. Omicslogic research-based training programs are a result of close collaboration between Pmi Biotech and numerous academic institutions that have participated in content evaluation, curriculum review, and project design. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are usually very large, and they take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. In a university setting, students have the opportunity to learn the bioinformatics project of designing and implementing a project, but do not receive practical or hands-on experience with the full workflow due to the limitations that I just mentioned. Similarly, faculty faces challenges with being able to cover a diverse range of topics if they are specialized in a certain area of bioinformatics or if they are unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know much about the metagenomics of seawater. To overcome these challenges, the Omicslogic portal was developed. This program aims to standardize, organize, and systemize bioinformatics training in order to make these analyses usable for any biologist or clinician. There are some traditional problems with the standardization of bioinformatics coursework, which includes materials for courses that are usually developed in-house, taught sporadically, and are rarely updated. The Omicslogic portal introduces new material that keeps up with new technology. These courses need to be organized because the challenge is to combine several different topics together. The flow and transitions between subjects needs to be developed. This is addressed at Omicslogic through introductory material, which is being woven throughout the courses. The last challenge we face is systematization. How can you know that someone is stuck and needs help or allow someone to run forward? To address this, Omicslogic uses standardized modules and interactive sessions, beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. Here is an overview of the learning process. In order to create a path of skill development, Omicslogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics, where students recall facts and terminologies and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The students will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do Omicslogic resources help advance bioinformatics teaching? Omicslogic is a portal for practical and theoretical learning of bioinformatics, composed of courses designed to take you through the theoretical knowledge. Omicslogic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give a student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, Omicslogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or beginning a data analysis project. These 6 courses were taken from the core Omicslogic curriculum, which is available online, beginning with an introduction to application of bioinformatics and an overview of data types. The student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, and data science are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each one. This allows the students to gain the conceptual knowledge in order to begin their own projects. Omicslogic expands upon its basic curriculum with specialization track projects. Bioinformatics methods are best learned in the context of real case studies prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available datasets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. In this symposium, we will hear from speakers from different domains of life sciences, talking about relevance of omics, integration of omics technologies, advancements in biomedical research, and also we'll hear about machine learning view of multi-omics integration. What we will discuss today and throughout is a result of hard work by an amazing team led by company co-founders, Dr. Alfred Tauber, who comes from a medical background, has an extensive expertise in oncology, immunology, and biochemistry of information, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center at the University of Haifa and is an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over his course of his career, he has developed multiple novel algorithmic approaches to biomedical data analysis. Under his leadership, the Tauber Research Center started collaborations with research teams at such universities as the University of California, San Francisco, Institute Pasteur, Tulane University, and many others, where research labs are working on cutting-edge research projects studying viral and bacterial infections and designing novel drugs and vaccines to treat and prevent infectious diseases. The rest of the team includes experts in bioinformatics, computer science, and training. Some of our team members will be presenting today as well. The module gives students the options for analyzing a given dataset: code-dependent paths or code-independent paths. These datasets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly T-Bioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs, where experts share their experience and support research projects developed by you. Here is an example of the code-independent pipeline. The pipeline builder interface on the T-Bioinfo platform is a color-coded pipeline that helps establish a logical connection between the technical names of data analysis methods and the logical steps needed to build the full pipeline. Data is put in as an input file, and then analysis can be chosen based on the type of research. Each analysis is separated into 3 colors: blue for pre-processing, red for mapping, and green for quantification. Each time you click a button, a pop-up will contain additional information useful for building the pipeline. The ease of the color-coded buttons allow programs to be run without any coding knowledge. Interactive outputs allow easy visualization of the data. Several demo pipelines with real data sets are available for students to practice the workflow, including demos on genomic variant calling of cancer cell lines, 16S amplicon analysis on diet, and its analysis on the microbiome, and a pipeline for examining a phylogenetic analysis of Ebola virus. To answer your curiosity about the T-Bioinfo platform, we have among us researchers for Tauber Bioinformatics Research Center who will highlight few areas of analysis available on the server. Dr. Julia Pano, who will talk about analysis of bulk and single-cell transcriptomics data on the T-Bioinfo platform, and Dr. Elizaveta Taranenko will talk about metagenomics data on the T-Bioinfo platform. After using the T-Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, the remember and understand levels of blooms, and apply that in a project. They apply, analyze, and evaluate levels of blooms. Then check their comprehension with levels and batches, certificates of completion, and test. Similarly, faculty should be able to assign the Omicslogic portal to their students with an understanding that it will give them holistic understanding and comprehensive knowledge of bioinformatics, beginning with bytes and molecules and onto designing their own question-based project. Omicslogic employs a hybrid approach, combining learning and research to ensure that students are building off of the first level of Bloom's taxonomy. Omicslogic tracks progress with activities like pipelines, coding, and literature reviews, quizzes on topics, key terms, and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then students are able to continue their knowledge base with a research assessment, making sure they are able to understand methods, challenges, and results, as well as being able to comprehend primary research literature and recreate the methodology. They should also be able to ask questions based on data exploration. This leads to the ability to encounter errors in their analysis, apply theoretical knowledge of why those errors are occurring, and find a solution. Bioinformatics combines life science terms with data science skills. which allows researchers to answer life science questions with reproducible data analysis methods. Oncology is proud to collaborate with many diverse programs across the globe. We also work with over 15,000 students at community colleges like Glendale Community College, top-tier universities like Georgetown University, as well as 20 universities around the world. We are pleased to have among us researchers from our partner universities and institutions who will enlighten us with their talks on different topics relevant to any student or researcher, from knowing about technology and soft skills for industry, skill sets, the missing link between academia and industry, to understanding genotypic data, personalized therapies for cancer patients, and hierarchical patient models. In this presentation, I explained the current challenges and limitations in bioinformatics. I described how we aim to address these challenges with the Omicslogic platform. We discussed Bloom's taxonomy for learning progression and how it can be used in bioinformatics coursework. We also explored the Omicslogic program and how it utilizes Bloom's taxonomy in structuring its course, pipelines, and projects, and finally described several outcomes of the Omicslogic program. Want to know how to get started? The Omicslogic portal is easy and free for anyone to register with many courses and online mentor-guided training programs in the field of omics that includes genomics, transcriptomics, single-cell transcriptomics, metagenomics, and epigenomics. Also, you will find programs in the field of bioinformatics, structural biology, infectious diseases, and precision medicine. These programs are available all around the year, and you can get started with your specialized training anytime at your own convenience. Once you have a specialized training, then you can work on an independent research project, again under mentor guidance on different specialization tracks, which includes the analysis of data types you learned in the training program. These specialization tracks include oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data analysis, metabolic disorders, space omics, agriculture, and comprehensive training on omics data analysis. Throughout the year, various students and researchers join Omics Research Fellowship Program and work on their research projects to either gain new skill sets or to complete their college dissertation or for a publication to add to their achievements. Omics Research Symposium will provide a stage to such research fellows to present their innovative research projects. Also, all the research projects will get a feedback from our panel of judges. The Omicslogic training has been completed by over 24,000 participants from 187 countries in over 500 workshops. And due to this fast growth, our team is now working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. As a result, we have acquired professional certification to offer our curriculum and support research projects using our Omicslogic project-based curriculum. And here are some amazing stats from an year-long training programs that were conducted by us. The symposium is divided in 3 parts, where we listen to speakers from Asia first, then Africa, and finally the Americas. So to begin with an introduction to Omicslogic Asia, I would like to call upon stage Dr. Mohit Mazumdar. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for Omicslogic Training and Research Fellowship. Over to you, Dr. Mohit.
Thank you, Sonalika. I think we are running a little ahead of the time, but let me start by thanking you all. And it's great to see you all here today with us. So this symposium is extremely important for us, and I hope you all are as excited as we are to hear about these cutting edge approaches in life sciences and also omics space. We will learn about these challenges not just from our team, but from experts who have spent their lives struggling with these challenges and, more importantly, coming up with these great solutions. Also, it is great to see a much broader appreciation of data in life science domain, as well as a great excitement about this potential impact of bioinformatics on areas touching the lives of everyone. For example, medicine, vaccine design, genomic surveillance, and even space travel. So in this presentation, I will explain our initiatives and highlight the partnerships and collaborations for Omicslogic bioinformatics and data science training research in Asia and upcoming expert sessions and discussions on day one of this Omicslogic Fall Research Symposium. So let me start by taking this opportunity to briefly introduce myself and our team. Since 2019, we have launched Omicslogic in India with initial collaborations at several universities where we piloted our training and worked with faculty to develop programs that addresses the major gaps in bioinformatics education. So since then, we have been working with thousands of students from our partner universities who are going through this training and working on projects. So we will be discussing that. So working in project work and faculty who are interested to utilize this collaborative research platform to incorporate bioinformatics or project-based bioinformatics and data science curriculums. So to tell you a little bit more about this growth and about our collaborations. So we have been able to work with a lot of students, and that's why it has been a thrilling journey. And this experience has enabled us to work on variety of challenges that comes with the educational training, as well as the translational aspect and the translational research. So with the agenda to bring awareness about data-driven healthcare research, our team is reaching out to reaching out to universities to make bioinformatics and high quality data-driven research accessible to everyone. So this engagement has helped us to collect a lot of this data and improve on our learning models. So after many programs coordinated with universities, thousands of students across India, we saw that there is a gap. There is a gap in skills. There is a gap that students help effectively could integrate into their research project. Thinking critically about the data, applying appropriate methods to the right data. These are some of the gaps which are quite evident, which we were able to find out and propose solutions to. So in this presentation, I'll be taking you through those case studies and also the solutions that we have got and the innovation that we were able to bring into this. So every student, we started with this realization and with this goal that every student needs to be enabled with data skills and analysis logic to succeed in their research, to succeed in their academia, excel in their career, and to be ready for the world of data we now live in. So schools, universities, and colleges, they needed to be equipped to help faculty and students learn and use data. Now, this is a big task, and hence it is a big task on our hand that can only be accomplished or achieved by a community, not by only us, by a group of people, group of like-minded researchers, group of like-minded institutions and universities. And our appreciation really to all of our academic partners who have come forward and joined hands to bring this awareness, to bring these new tools, to bring this new technology and an international standard to the training and research that we are providing in collaboration. So not only academia, but also industry, and that is why we are here today to discuss what can our educational system do about this gap. How do schools, colleges, and universities prepare students for the future of biotechnology and life science in general? So where biology, we can see that like this is quite evident because we see that every day that biology is acquiring the characteristics of data science. We are generating so much data. How can we merge data science and life science together in a practical way that is effective as well as inclusive? How to prepare students for successful research and industry career, give them skills and experiences that help them navigate their future. So this is something that we have learned and that we have experienced through our own journey, as well as through the entire experience of working with these thousands of students. And we are delighted and have welcomed all our industry collaborators. Those have really contributed in this, in mentorship, in industry transition and soft skill development, in translational projects for expert Omicslogic fellows, internships and job opportunities, career guidance and placements, research opportunities, and also startups and hackathons. So there is a lot of contribution about through industry, through academia, and we are seeing these different components here and there. So during this entire symposium, we will be able to do one thing. We will be able to connect these components together and see that how this could be a part of, it could be an ecosystem in itself. So to be able to do that, that brings me to this slide where we would want to make a major announcement for our 2020 fall semester. And that announcement is the launch of Omicslogic Partnership University Program. And this unique or this program was developed by contributions not only by the team, but subject matter experts from our partner universities and research institutions that you just saw on the previous slides. Not only them, but also thousands of students who have completed the Omicslogic training and have provided this valuable feedback. So this Omicslogic partnership is a unique opportunity for universities and is a comprehensive and a flexible program designed to help students, faculty, and industry to succeed in this age of data. So our Omicslogic training is designed to make complex topics like bioinformatics, machine learning, and system medicine easy to incorporate into existing curricula and help students and faculty transition to the data-driven research projects where bioinformatics curriculums are designed based on the need and the level of participants, focusing on the personalized level of training, personalized level of help that many of the students require or many of us require to get the best outcome to work on and to be able to apply bioinformatics, data science, biomedical data science, and some of those topics. We prepared this very unique curriculum along with our partners, which is now available for anyone, any university to opt for free, is to use these topics which are not usually are the part of their curriculum. So the Omicslogic training, again, is a personalized training and research experience that enables participants to develop critical scientific thinking, utilizing modern bioinformatics and data science practices on industry relevant problems. So the sessions that we have designed, they introduce statistical methods, visualization, introduction to popular machine learning techniques for omics data analysis. Participants do learn about public data repositories and various file formats that are used in bioinformatics for storing and efficiently processing omics data. So as I was discussing all of this to you, I think the best way to do this is through a practical experience. And that practical experience is the case study that I wanted to discuss with all of you today. So a memorandum of understanding, an MOU, was signed between MIT University, Uttar Pradesh, and Pine Biotech US for joint research and training in the areas of bioinformatics and data science. All right. So this collaboration helped us pilot the Omicslogic research-based training on a diverse student cohorts coming from a non-bioinformatics background, applying data science for biological problems. Now, this could only again be possible with the help of a team. So the collaborative partnership program, or this partnership that we have just launched, enabled all our partners to work together while sharing ideas, while sharing research proposals, while sharing curriculums or curriculas that are protected by the agreement between these three organizations. So the first organization, MIT University Uttar Pradesh, Pine Biotech, and our major academic partner, Tauber Bioinformatics Research Center at the University of Haifa in Israel. So scientists from this center are providing academic oversights and guidance in curriculum preparation, development of current bioinformatics analysis on the T-Bioinfo platform. As you have seen, we just did that, saw that Sonalika discussing about that allows non-bioinformaticians to run complex algorithms on large-scale databases, and the support team who have been working relentless to help students go through these different stages of training through online and virtual, as well as on-site support. Now, to be able to do this, we would be needing a way to evaluate students, evaluate their journey. So to learn about the progress of student during the training, and to learn about the challenges, as well as to encourage students to move through these different stages of training, we developed and utilized a point-based system that integrates Bloom's level, as Sonalika was telling you, and helped us create a dynamic and a unique interface ever in bioinformatics, and a great experience for the users. Now we have the platform that could enable and encourage students. And now what we would want is a pipeline to be able to help students go through these different stages that I've been talking about. So to do this, one of the things and one of the challenges, or I could say the next challenge for us, was to develop the right components to get a meaningful outcome from this training experience. So the reason is that many students, and it's an experience working with students from all around the globe, they need a soft landing into this field of bioinformatics, where they can get introduced to the application of bioinformatics through projects and gain access to the community with mentorship, guidance, and structure. So these are some of the components that are required at a basic beginner level. So we developed these different stages and resources: basic training, example projects, and coding. So these are different stages. You start with a basic training, you move to example projects, and you learn coding, and then you practice to work on projects by practicing on various data sets and algorithms. Finally, you are ready to work on your own research projects or a faculty-driven research project. So that is the pipeline that we prepared and created to be able to help students who are coming from different backgrounds, different knowledge, and different interest. Now, in the next few slides, I'll discuss about the outcomes of the partnership training program that enables our team to meet face-to-face and address gaps, and work together and find solutions to the challenges, and translate ideas into actionable tasks and outcomes. So this is again an experience, and this is one of the pilot that shows, it's a case study that shows that how this route has helped us develop these outcomes that I'm going to show you in the next few slides. So as I was telling you about some of these points, integration of new ideas, that is very important because of this opportunities applicable of this open source data. And then application of new technology in life science. And then, kind of becoming an expert to be able to contribute to this ongoing scientific advancements. These are really exciting opportunities for students, for faculty who opted this field to contribute actually for the human well-being, right? So the Omicslogic training was able to provide these opportunities not only to the few, but the entire cohort of students, and the simplification and the integration of NGS and omics technologies and algorithms through the training. Now, as I'm talking about integration of next generation sequencing, omics technology, data science algorithms, and that is one of the focus of our company. And then to be able to do this, we were able to work together with the faculty, with the administration, and develop curriculums that has industry insights. So we are looking at job opportunities. We are looking at that industry is looking into these candidates or these students, and then we are trying to highlight that through the curriculum, and that is the industry insight that we are bringing to the curriculum of this universities. Now the last component of all of this, is to be like, first you learn, and then you apply, and then you do a research project, then you gain experience from a project, and to be able to work in a group, learn those soft skills, communicate, how to communicate in a group, and to how to make a meaningful way to showcase your research, do a presentation, and do a publication. But after all of this, what is that is needed is the job opportunity, or the next opportunity that you are looking to. That could be a program, that could be a master's program, that could be a PhD program, or that could be a job opportunity. So finally, to get job and internship opportunity, to build excellent profiles on Omicslogic portal for recruiters and startups, we integrated this the Omicslogic job opportunity port into the portal, and that helped us to guide students directly to the related opportunity with the skills that they are working on and gaining. So the partnership university program, as discussed, is to simplify the training and make it accessible to students from all background, and not only from all background, but from all demographies, from low income countries, and from places where it is really difficult to access technology, access infrastructure. This is entirely online, and we are offering the basic training, which is the stage one of this pipeline, freely to the interested universities who wants to pilot the training for their students. And to tell you about the research aspect of all of this, and one of the major outcome is to be able to work on the aspect of your doing your own research and working on your own research idea, or how a faculty can propose their research ideas, and the students can help with their skill sets to overcome some of the challenges in their own research. So one of the important aspect of all of this is to be able to help students gain those skill sets, and to be able to come up with complete a project. Because many students, and it's also us, that we tend to start many things, and then to complete that and to finish that and to publish that, it takes a lot of time. There are different stages here. It gets complicated, and there's a lot of research, and there is a lot of literature review, and there are different aspects of research. So to overcome these challenges, we developed this research fellowship training, where we were able to help students work from any remote area at their convenient time, working on industry designed example projects that we curated, and then mentor guidance with group projects that are from PhD and postdoctoral level solution, providing PhD and postdoctoral level solution. So we have Dr. Raghavendran, and we have a huge community here who are helping us guide these students with their independent research proposals and integrating faculty ideas into a research project. Now, the students who were able to do that were the ones who benefited from this, and also the faculty who were able to work with us collaboratively are also some of those who have benefited from this research work. And now to tell you about the way this has moved from one year to another year, and what are the differences that we saw in this by students who have completed these different topics, are from coming from one background in 2021, we see that there was a lot of students from BTech Biotechnology who joined the program and completed the program. They were interested in oncology and application of precision medicine. They applied a lot of genomics and transcriptomics data analysis and worked on different of research topics. But by 2022, we saw that there has been more applications, there has been more research areas, there has been diverse community of students who are joining and working on their research projects, and these are all from non-bioinformatics background. These are the students who have done who came without any knowledge of coding, without any knowledge of bioinformatics, and they started from the basic training and they went through the pipeline where they after working from learning about these different technologies, learning about this different applications where this technology is applied. For example, how genomics is applied in precision medicine, how a transcriptomics data analysis is utilized in oncology, what are the different ways to look at the biomarkers in neurodegenerative diseases, where machine learning is mostly applied, and what kind of data it applies to. So there was a lot of specialization training which was involved in this internship period of the students, which was also from 45 days to 90 days to 160 to 120 days to 6 months. So we had this challenge where students have different timelines, but we kind of created these different modules through which we can check their progress and see how they are moving from one stage to another based on the timeline they have registered to. Now the students were able to complete some of these projects that I am showing you in this slide. So to highlight what the students were able to achieve was one of the major outcomes of this research initiative or this collaborative initiative, where we have been able to record a lot of this data to be able to showcase, or also the students who were able to see this and showcase to their recruiters or to their faculty, and faculty can see their progress. So there is a progress link associated with this training with every student, where where we've been able to understand that what is the level where the student is working on, what project he is doing, or what kind of expertise the student is developing, which is also open to the community, and look for specific expertise in these students. Now, another benefit that the students have been able to achieve through this is to be able to work on a real project. Now, this is a new project, a noble project, in the sense that the data which we have looked into, and also the research problems that we have looked into, are specifically designed with the help of the mentor. And there were a lot of discussion, a lot of research, literature review before opting for a specific pathway of to go for the use of the tools and the technologies, and also the different visualizations and other reporting which is required to complete a project. So, I mean, for the students going through this entire process of completing a project, going through these different steps of research, and then to be able to do a presentation and do a presentation and get that acknowledgement, that and also which actually helps them to build a better CGPA, an entire growth that could be seen through this project, and also achieving the brilliant results that they have been able to do in a very short period of time, was very rewarding. And in this entire symposium, we will have those opportunities to hear from some of these students in our upcoming session. Now, these projects and other focuses on different specializations. As we are discussing about precision medicine, we are discussing about oncology. So some students with their PIs have very specific ideas to do maybe landscaping of cancer immunotherapy, which is a very broad topic. Now, when we are looking at it and when we start working on it, and if it's a collaborative project, then the chances of completing that project is very high, because not only one person but different students working together with the help of a PI and the help of the mentors at Pine Biotech. Now, all of these projects are published on Omicslogic Learn, and you are free to check out some of these projects if you feel interested and learn about these projects. But this Omics Symposium is also an opportunity to learn from some of these experts who have experience in this field, and also from these student examples that students have done. So we will have experts next in this Omics Symposium day one, where we will be learning about the relevance of omics. So you saw that how students have utilized different omics technology and have completed a project, and we will be hearing from Dr. Suchi Nagar soon from about the relevance of bioinformatics in omics, and you will have those questions ready, and you can have those questions ready that you have to understand that how this is relevant in studies or in understanding, some of these diseases that we see, such as the neurodegenerative diseases. And also at the same time, here are some of the links that students have been able to work. For example, transcriptomics analysis that they did on finding out the gene signatures regulating Alzheimer's diseases. So looking at and working on multidimensional data and applying those algorithms such as principal component analysis, looking at the gene expression and finding which are the relevant and significant genes when we are comparing two different states, and then looking at further doing the downstream analysis, leading to the analysis of Alzheimer's variant in embryonic stem cell derived microglia. So this is a project that it's also an example that you can take a look at. So again, these are some of the outcomes of the student driven and or the faculty driven fellowships, where students have been able to work on important topics such as precision oncology. We saw that as an example. We also saw the use of in neuroscience. Now, this is an example of infectious diseases, and this is an example of surveillance of SARS-CoV-2 COVID-19 variant of concern from wastewater samples based on the NGS data. And there's a recent article that actually highlighted that how important it is to do a genomic surveillance and how important it is to use the wastewater samples. Now, this is a practical experience, what is happening, just happening in the industry, just happening in that you see around you, and you have an opportunity to work exactly on a similar research, use scientific tools, and come up with those, maybe find new insights, find new mutations. Those are some of the opportunities that the faculty student driven research fellowship was able to provide to the students who worked with us during this with this partnership program with MIT University, which is the case study that I'm showing you all to highlight what we have achieved in the last 6 months working with this different students. And as an opportunity, as you all are interested here, and I see this great enthusiasm and a lot of new participants joining, and really happy who are joining for the first time, that we will start our presentations from the experts, and we will be learning from experts from IIT Delhi, where we will discuss about Dr. Ishan Gupta, where we will discuss about the COVID-19 pandemic and his research on this. Similarly, we are discussing the research that students have been able to do while they are using this different types of data types. For example, genetic and epigenetic basis. So genetic information, epigenetic information, a multi-omics approach. What are these different approaches? How do you learn about these approaches, and how do you apply this into your own research or your CV, that you have learned this and applied this? So we have those opportunities in today's session, where we will be learning about the application of disease types, complex diseases, use of multi-omics data from experts who are going to be joining us in and letting us know about their research, and what are the different aspects that they have gone through completing that research. At the same time, yeah, the students who have gone through this entire experience themselves will also get an opportunity during the poster presentation poster presentation session, which is coming up, and really excited to look for. We are looking forward to that session where the students will get opportunity, and also for sessions such as these, where we are actually going to learn from experts like Dr. Anshu Bharadwaj, who have expertise and have been worked across different institutions, and a rich experience in bioinformatics, and understanding the genotypic phenotypic correlations in complex diseases. And we will be getting those opportunities to learn from experts, and I hope all of you are now excited to be a part of these talks, upcoming talks, and have questions and insights ready so that we can discuss this further. Now, after we learn about this, first we started by learning about COVID-19, and then we will be learning about the application of omics technology in different type of in Alzheimer's, and then we will learn about the complex diseases. So what we are learning here from one session to another is also how the use of bioinformatics, which is such an interdisciplinary subject and with substantial and growing influence. So bioinformatics, as you see, that it has a huge application. It has huge applications in several of these interdisciplinary subjects. So what are these subjects? So health is one of such important topic. Another very important topic is environmental sciences and society, and which is utilized by scientists from many diverse academic backgrounds. So education in bioinformatics therefore necessitates effective development of skills in interdisciplinary collaboration. It involves not only skills, but also communication, ethics, and critical analysis of research. So in addition to practical and technical skills, insights from bioinformatics training can additionally, inform, and that is what one of the agendas for the today's presentation, that how this training and collaboration and integration of academia and industry who are working on data science and artificial intelligence and cutting edge technologies can be accessed, and everyone is now has that opportunity to be able to achieve that. And what are these different opportunities? What are the different tricks and ways to be a part of this? This is something that we will be learning in the academic panel discussion, which is going to be happening at after the three keynote speakers discussion, that how we can transition from academia and how we can learn and get opportunities in academia. And after that, we have the poster presentation session, which is planned at after the academic session, where the students will get opportunity and will be reviewed by these experts for best technical and research project, for promising research direction and most innovative approach. And I would request also maybe Ojasvi to share the final list of posters who got to the next step and are now ready to be evaluated by the experts and judge panel for best technical and research project, where after getting an opportunity and winning, there will be cash prizes available for the research group or the research student who will be presenting their poster in the session, which is after the academic panel. Now, that's how it looks for the Asia panel. And I am hoping that all of you who have joined us today are really excited to be a part of this and to learn from the experts, and also to connect with all of us and get some of these opportunities that we are discussing, and be a part of this Omicslogic community by logging into Learn Omicslogic, and also by reaching out to us and be a part of our social network. Now, after the session that we have planned for Asia, we will move to our next chapter, which is Omicslogic Africa, where we will be discussing, where it will be led by Mr. Adikunle Adilio, who is overseeing Omicslogic Nigeria, and is also a faculty at Leeds City University, Nigeria, who will be discussing about the programs that we did in Africa and the outcomes of the student training, and what are the different projects and ideas that students are working on, which universities are applying and implementing this, and what are the different directions that the Omicslogic community is looking at. Now, we will also have speakers who are very experienced and experts in their multidisciplinary research, uh, including biomedical research, use of omics data, and computational biology, where we will be looking at specific problems that are discussed in the aspect of in Africa. And then we have sessions where we are discussing about opportunities of scientific research in Africa, and also opportunities what are there in some of these universities, and where we will be discussing and knowing about these opportunities by Dr. Marion. And then we have an important group discussion with a panel, exciting panelists from Cameroon, Zimbabwe, Nigeria, and our team members from Omicslogic Africa, who will be discussing and summarizing some of these important aspects. And it will be great if the participants from Africa, or those who are from African origin, can also be a part of this panel discussion and ask their questions that have been bothering themselves, or participate by giving us insights that would help the entire community. Now, after the Omics Logic Africa conclusion, we will move to the next chapter, which is the Omics Logic US. And in the next chapter, led by our CEO, Ilya Brodsky, we will be learning from experts from different areas of research, and coming from different areas of research, as well as coming from applying this for very innovative research, which involves machine learning view of how to do multi-omics data integration. So some of these advanced topics that we will be learning from the Bioinfo team, also the Tauber team, Dr. Julia Panov, Associate Director at the Tauber Bioinformatics Research Center. She will be talking about analysis of bulk and single cell transcriptomics data. So these are some of the technologies that are really now being used by the pharma, by the top research labs. And it would be a great opportunity for all of you to learn from the Bioinfo or the Tauber group, uh, from uh, from single cell to metagenomics data, and different use of technologies of omics data. Then we have the keynote speakers, as Dr. Sara C's Pico will be talking about interdisciplinary forensic science integration and how omics technology is used in forensic sciences. And then we have an expert on precision medicine from pediatric oncology on the state of art and perspective, Dr. Luis Sokamoto, who is an MD physician in Brazilian pediatric and as an oncologist. Now, then we have another session, next keynote speaker, talking about bioinformatics and cancer system biology. So there are exciting sessions which are coming up, and I'm hoping that many of you will be staying tuned to these sessions. And as you get time, keep on joining and learning from these experts, and asking the questions that you think are relevant, and we will be able to record those questions that you put in the chat and that you put in the YouTube chat as well, and we will be able to reach out to you with a relevant answer, if not by now, but we will be able to give that solution or answer by email. So I hope that you will make this a lot more interactive. You'll get this is a great opportunity for students. So those students who wants to learn from these experts and also wants to get support on some of these resources could reach out to us. That brings us to our first session, and really excited to have Dr. Ishan Gupta here with us, assistant professor from IIT Delhi, to talk about too many ways to die from COVID-19, results from the world largest cohort of lungs. So Dr. Ishan Gupta works with the functional genomic group at IIT Delhi and develops and deploy bench to code novel omics technology to measure biological phenomena with high resolution single cell, high precision isoforms, and high throughput technologies. So using this technology, he wants to understand the nature of biologically complex diseases. So with that, I would like to invite Dr. Professor Ishan Gupta. So over to you, sir.
Yeah, hi. Can everybody hear me? Hello.
Yes, yes, we can hear you fine.
Hi. Is my screen visible?
Yes,
You can see my slides.
yes.
Okay. All right. Thanks to the organizers for hosting this talk. I think there is some issue with my computer, so I'm gonna basically be changing like this. I hope this is fine with everybody. Yeah. Does
Yes,
this work?
yeah,
Because
it
I at
works.
this point of time I'm on
Yeah,
Linux
yeah.
right now, and yeah.
So can you do a full screen, or this will be the screen size?
Yeah, this is the screen size, unfortunately.
Yeah, yeah, that's fine. It's visible. I think it's kind of visible for the audience, and they can also, like, please let us know in the chat if you guys can see the presentation. Please let us know if there is an issue. It's visible. So yeah, it's visible. So so
All right.
you can go ahead, Professor
Yeah.
Ishan.
So thank you for having me. So today we are going to talk about I'm going to talk about some of the results in my lab where we have looked at COVID-19 patients from India, and we have looked at their like lung biopsies and autopsies, actually autopsies after the patients are passing away from the disease. And typically there are three messages that we have that I can give immediately. Is that it's very important to actually do such studies on Indian patients, on Indian cohorts, and not only rely on the results that have been published from cohorts from outside India, because the genetics and how the patients, the environment that is the patients are exposed to is very different, which leads to very different kinds of results. So that's the first thing, first take-home message. The second take-home message that I can give, which is from the title itself, we find that there are two very distinct ways in which patients could be responding to the disease and therefore are succumbing to the disease. And the third thing is there is a big underappreciated role of the resident lung microbiome, or the microbes that are residing in your lung, that could also play an important role in COVID, as I will try to motivate in this talk today. So most of the work that I'm going to be presenting here was done in collaboration with Dr. Dipali Jain at AIMS, and was led by three students in my lab: Anshul on the left, uh, Anubhav in the middle, and Abhilash on the right. So this is a paper that was recently published in the journal DMM, Disease Models and Mechanism, about 2 months back. All right. So COVID-19 in India, there have been about 5 lakh people dead. So 5 lakhs would approximately be 500,000 dead, but there is no molecular data from India. So although we are a country with one of the highest disease burdens, of the top 3 disease burdens, and a lot of, like, 500,000 people have died because of this disease. There is, I mean, very few data is there for exactly looking at the patient types or patient sub isotypes, looking at the molecular biology of the patient. There's a lot of clinical data, epidemiological data, but very little data from exactly how the patients look like under the microscope or at the molecular level. So this is what we are setting to do. This is what we had essentially doing. So we are essentially took 10 patients as controls, and 32 patients who had COVID-19, so patients who had died of the disease, and we took core samples from these autopsies very fast. So this is with a technique called MITS, is massively invasive tissue sample, massively invasive. I believe I don't think it's invasive. Just that you have an injection which gets like you put an injection onto the lung samples or the tissue that you want to sample, and you can quickly it's minimally invasive tissue sampling. You can quickly get a tissue sample. So the idea was that we sequence these samples and did the RNA sequencing. We did the quality control. We aligned these samples to the human genome. We found differential transcriptome analysis for these samples. So basically figuring out differences between the COVID samples and the normal samples. And besides the data from the human genome, we also had data from the microbiome that was resident in the lung, and that's what I'm going to talk about. So one major result from our study was that when we take a look at the gene expression level of the patients, we find two kinds of patients. So just to show you the figure, in this figure, you see that each dot is a patient, and the normal patients can be seen in the color red, and COVID patients can be seen in the color steel blue, right? And what you see is that when we are plotting the gene expression of these patients in higher dimensions, specifically capturing the first two principal components, basically characterizing how these patients could be different at the level of gene expression, we find that the patients are segregated into two parts, right? So on the left side and the right side of the graph, on the left you have sets of patients, COVID patients and normal patients clustering together, and on the right side you have only the COVID patients clustering together. So this suggests that there is a subgroup of COVID patients. So this is approximately 8 COVID patients who are actually very similar to normal patients at the level of gene expression. And on the other hand, there's the majority of the COVID patients, about 3/4 of these COVID patients look very, very distinct from normal patients. So what could be the reason for this is something that we try to figure out in this paper. So we had some clinical annotation for these patients, like whether they had some cardiovascular complications, whether they had diabetes, if they had liver complications or other comorbidities, and we did not find any signature. So basically these comorbidities could not be the reason why the patients were separated. So here we have colored, a no is colored in red and a yes is colored in this steel blue color, right? So what we find is that you cannot clearly see that, okay, one of the comorbidities like diabetes is more enriched in the patients which are similar to the normal patients versus patients which are extremely distinct. Although we do know that patients with cardiovascular complications and diabetes, from our own work, we have shown in Indian patients that patients who have diabetes are more susceptible to COVID-19. But that's at the clinical outcome level, but at least at the molecular level of how their lungs are looking, the gene expression of their lungs are looking, that's not the case. So then what do? We essentially go ahead and assume that these two groups of patients are distinct, and then we compare each of these groups with the normal sets of patients. So normal sets of patients here would be these patients in red. So what we do is we do a comparison of differential gene expression between this group, which is similar to normal, which we call G1, and this other group, which is very distinct from normal, that we call G2. When we do this, we find, as expected, lots of genes that are different between these two groups of patients. Specifically, there are about 1,000 genes in total that are different between these two groups. Some of them are going up, some of them are going down. And if you can see on this plot on the right, this is a typical heat map. On the first row, each column is a patient. The first row gives you the color of the type of the patient. So a light pink color means this belongs to the G2 group, or the group that was most distinct. And on the right side top, you have the dark maroon color, which would be patients which are in the G1 group, and you have this blue color, which patients who are normal, right? So if you look at the first, first rows, each row here is a gene. You see that there are certain genes which are only expressed highly in your normal patients, and they are not expressed in either G1 group of patients or G2 group of patients. Similarly, if you go down, you have certain genes which are only expressed in the G2 group of patients, mildly expressed in the G1 patients, and not expressed at all in the normal patients. And then you have another sets of genes which are expressed highly in both G1 and G2 patients, but are practically absent in the normal patients. So in order to understand what's going on, what separates these groups of patients and what unifies these groups of patients, we can actually take these lists of genes that we obtain after such a differential expression analysis and try to understand what are the common pathways that these genes belong to. All right, so when we do this, when we are basically separating these genes, differential gene expression, treating it separately, as expected for G1 patients, we have a much less number of genes as opposed to G2 patients. And what we find when we do pathway analysis is you have pathways such as fibronectin beta chain, serum amyloid, apolipoproteins are upregulated, these pathways, and genes which are relating to, say, scRNA or FABP4, they're downregulated here. And similarly, in the case of G2, some interleukins, some angiotensin enzyme by vitronectin, these enzymes are sort of upregulated, and a lot of SNO RNAs or specific genes are downregulated, right? There's a very little overlap between these two patients, just to confirm what we saw in the PCA plot. But in order to go deeper, we now try to look at which pathways are enriched for these genes. So specifically, I'm showing you here the pathway enrichment analysis for genes that are upregulated in G1 patients versus N patients. And what you see is that lipid metabolism is upregulated. Coagulation could be slightly downregulated, and there is upregulation of, coagulation could be slightly downregulated as negative coagulation, and neutrophil-mediated immunity is slightly downregulated. And this is something that was confirmed in other papers, by other papers in smaller studies, by targeted analysis, and the papers are mentioned here. When we look for G2 patients, specifically one pathway which stands out is complement activation, pathways in xenobiotic metabolism, and peroxisomal protein transport. And this was also supported by some of the publications that came out early last year. Now, this essentially means that the disease that is being experienced by G1 patients and the disease that is being experienced by G2 patients may look very differently molecularly. So what could be the reason? Everybody would have heard of ACE2 receptor and TMPRSS2 protease playing a role in SARS-CoV-2. So we try to figure out if the expression of these two genes could be sort of indicative of what's going on. But for ACE2, definitely we did not see any major changes in gene expression. And for TMPRSS2 as well, there were certain changes, but when we correct for multiple testing, these changes do not hold that significant. There are other proteins which can substitute for TMPRSS2, which is a protease which helps the processing of the spike protein. When we tested these were also not really convincing and cannot distinguish between why these two patients are experiencing the disease differently. So the next thing that we did was we tried to enrich for specific gene sets that were known to be implicated in COVID-19, basically some pathways that are known to be sort of playing a role in the disease. Some of the pathways, you can see here, this was done by a tool called GSVA, which is basically gene set variation analysis. So as you can see, again, we have similar heat map plot, but instead of plotting genes as in rows, we are now plotting a score for a pathway in each rows. So if the pathway is upregulated, you would see a more green color. If the pathway is downregulated, you'll see a more purple color. And you can see there are pathways like unfolded protein response, which is known to play a role in viral infections. You see that this pathway is highly upregulated in the G2 sets of patients. So the G2 patients are all these patients in green. Your normal patients are in blue here, and the G1 patients are in red. So you see in G2 patients, typically you have high unfolded protein response. There is high oxidative phosphorylation, fatty acid beta oxidation is also high, and all kinds of complement systems, classical, alternative, lectin-induced are all high. while in the case of both normal patients and G1 patients, these pathways are not high, right? Similarly, there are certain pathways which are high in the normal patients and in the G1 patients, such as these IL-1 cytokines, which seem to be low in the case of our G2 patients, the other patient subtype. So what this kind of analysis essentially tells us that there are certain mechanisms or certain pathways that are upregulated in both kinds of patients. So interleukin receptor, interferon response, JAK-STAT signaling is upregulated in both these patient types. But if you specifically look at G1, chemokine ligand signaling is specifically, such as the cytokines, is specifically upregulated in G1 patients. And here, if we go down to what kind of chemokine ligands, specific genes that could be found upregulated, we find that CCL19, which was shown to be a potential biomarker for patient stratification in the blood of COVID-19 patients, that was only upregulated in G1. While in the case of G2, you had very high presence of all the complement system, some chemokine receptors, TNF factors, TGF beta signaling, so on and so forth. So with this analysis, we can suggest that why is it that the patients look so different? Because the molecular biology of the lungs looks very different, specifically in the context of how chemokine signaling is working and in the context of the complement system in the G2 patients. And with CCL19, we actually have one way, one biomarker which should be and which has been used for stratification of patients to tell which patient could be a G1 type or which patient could be a G2 type. Beyond this, we also can look at which specific cell types in the lung could be affected. So their lung is composed of many different kinds of cell types. And what we find is we can have gene sets with respect to each cell type. And what we find is that in the case of your COVID-19 patients, specific cell types like proliferating basal cells or alveolar epithelial type 2 cells, they seem to be disrupted. And if we make a sort of a plot for what kind of cells are changing their proportions between G2 and G1 patients, we specifically see that on this x equal to y line, your classical monocytes, lipofibroblasts, dendritic cells, they're all changing similarly. So either they're going down in both G1 and G2 patients, or they're going up in G1 and G2 patients. But two cell types, specifically proliferating basal cells and TREM2 dendritic cells, are positively changing in G2 patients, but they are negatively changing in G1 patients, which essentially means that suggests that these cells could potentially allow you to understand why these patients are behaving differently, and maybe even design distinct strategies to or drugs to treat these two patients differently. One thing that I started with in my talk was to sort of motivate that such studies have not been done in the Indian context and why they're important. So in this slide, I would like to motivate that. So there was a study in 2020 by Ninehold et al., where they took 22 patients, the biopsies from 22 patients, fixed samples. We have fresh frozen samples. And what they found was, they found that there are 68 highly upregulated genes and 30 significantly downregulated genes, because they were using actually a gene panel instead of doing RNA sequencing. And they found that specifically interferon genes were changing in this process. And what they did was they were able to subclassify their patients into these interferon stimulated gene high category and low category. And they said that there are two kinds of patients, the Caucasian patients that they were looking at. There are two kinds of patients in which have different kinds of COVID-19 manifesting. Now, when we do look at the same gene sets in our patients using GSVS scoring, we don't find the results to be replicating. We don't find our patient two subtypes, the G1 and G2 subtypes, to represent the same subtypes that was found by Ninehold et al. So this essentially means that the way that the disease is manifesting in the Indian patients is potentially different from the way it is manifesting in the Caucasian patients, or at least the we are using RNA sequencing the difference in technology could actually be giving you different results. So therefore, this needs to be looked at. At least we are not able to reproduce the results that they have shown. So the take-home message for the talk so far is that clinical covariates have little impact on how the gene expression of COVID-19 is segregating between the two groups of patients. There are two potential kinds of these patients. The patients recapitulate the molecular aberrations or the changes in molecular biology which has been reported. So there is unfolded protein response, there is IL-1 inflammation, there is change in the complement pathway, there is changes in drug metabolism because most of these patients were under treatment. And there is downregulation of pathways like olfaction, which was one of the first signs of COVID-19 where smell was lost, and downregulation of dendritic cells and T cells, which are basically going to respond to the disease. And we found certain specific things in our COVID samples. There was enhanced lipofibroblast and basal cells and ciliary cells, and there were diminished dendritic cells, specifically IGF-21 positive alveolar type 2 cells and basophil mast cells. And there were certain cell types, as I said, which are distinguishing between our two patient types, and we can actually distinguish these patient types based on certain chemokine markers as well. The second thing that we try to actually look at is the remaining 20% of the reads which did not align to the human genome when we were doing our analysis. And these remaining 20% of the genes we found by aligning to custom databases of bacteria, viruses, and fungi, they're actually coming from the resident microbiome of the lungs. So what we essentially found was, if we compare COVID samples with normal samples, the microbiome diversity was massively downregulated. So not only did these patients have infection from the COVID virus, but as a result of infection from the COVID virus, there was also loss in the diversity of the bacteria that could be normally residing in the lungs. And you can also look at this loss of diversity in the context of a PCA plot, where the abundance of each genome that was detected, or each microorganism that was detected is plotted. And you see that most of the COVID patients are on the right side, separating from all the normal patients, unlike at the level of gene expression. So there is definite dysbiosis in these patients, and that is something that one has to look at. So one of the things that we looked at in the case of dysbiosis was that we were able to actually detect complete genomes of the SARS-CoV-2 virus itself from the sample. So we were able to actually get 4 samples had high confidence detection of SARS-CoV-2 virus, and out of these 4 samples, in 3 samples, we were able to actually reconstruct the entire genome sequence of the SARS-CoV-2 virus. And when we looked at which lineage these viruses corresponded to, so they were into the Nextclade lineage 20A or Pango lineage B1.36. So this was the beta variant. So this was corresponding to the time of sampling of the variant. So this sampling happened in late 2020, early 2021. And it corresponded to the time where the samples were taken. So these samples were deposited. I'm sorry for the image quality. If I could have make it bigger, you would have seen it. So these samples were submitted on a GSAI-8, and we could see that all of them were actually sort of clustering with other samples that was submitted from India and from AIIMS, the hospital. And there were specific mutations that we could see, which would be interesting for people who are actually looking at how the virus is evolving. So as I said, the variant that we found were from this late 2020, early 2021 variant, which is during the time of sampling. So that sort of is a sanity check for the fact that we were actually capturing COVID samples of that time that we wanted to look at. Now, if we specifically look at how the dysbiosis is happening in these samples, instead of plotting all the COVID samples together, now I'm plotting the COVID samples separately, the G1 and G2 samples separately from the normal samples, and you see that both the G1 and G2 samples have has dysbiosis with a reduced Shannon diversity index. And although there is no differences at the level of the G1, G2 groups themselves, that the way that the dysbiosis has happened in the 2 patients is quite similar. So in both the patients, the bacterial composition or the microbial composition, actually we are also looking at viruses, is similarly diminished. And now if we plot the at the phylum level, how these in each individual samples, how these are looking at. So for example, each column here is normal samples. So the first 10 columns here are normal samples. The next 8 columns are G1 samples, and the next about 24 samples are the G2 samples, G2 patients. And what we see is that there is a similar kind of trend. There's nothing which is like outstandingly different between the G1 and G2 patients at the level of phylum. But now if we specifically go and take a look at the level of specific species that we were detected, there are some trends. So if you look at this heat map, again, each column is a patient. The patients who are corresponding to the blue column are normal patients. Patients who are corresponding to the red column are G1 patients. Patients that are corresponding to the green column are G2 patients. So now if we specifically look at these rows, which are marked as G1 versus GN, so these rows represent strains which are specifically found to be present in a high number in G1 patients and much lower number in G2 patients. So one of these strains is Staphylococcus epidermidis. So this basically is an opportunistic pathogen which is on our skin, and this pathogen was found to be more enriched in G1 patients than G2 patients. Similarly, there is Staphylococcus conii that was found to be more enriched in G2 patients. They were present in a slightly higher number, right? And the G2 patients also had pathogens like Aspergillus oryzae and some viruses. Similarly, there was other sort of resident microbiome which could possibly be protective, such as shown here. So all these bacteria, Mycococcus luteus, Ralstonia, all of these are the bacteria which are potentially depleted in the COVID patients. So what this sort of suggests is that there could be specific species of bacteria which can colonize the immunocompromised niche of the lungs of the patients who have been suffering from COVID-19. And interestingly, what you'll find is that the COVID-19 was only detected in like 6 patients. So the lungs had cleared out the infection. So basically, the take-home message for this part of the talk is that dysbiosis, you see dysbiosis in almost all the patients of COVID-19. And there is definitely enhanced presence of Staphylococcus infections in both the kinds of patients. And we were able to recover COVID-19 even in these patients during close to their, just before they gave up in 20% of the samples. And we were able to actually recover with high confidence in 4 samples. We were actually able to recover the entire virus itself. So the outlook that one could have from such an analysis is that working on a country-specific cohort like we have done revealed new subtypes. So it's important to actually go around, and whenever you are actually looking at some molecular phenotyping of a disease, it should be done across the world, across different populations, and definitely large population groups of the world should be represented, like India, which is typically not. What we find is that there are specific pathways which are changing between the patients, and there are specific suggestions that we have for stratification of the patients based on these pathways or based on the molecules that could be found in the blood that could act as a proxy for these pathways, such as CCL19. The fact that we have gene expression changes can be used to also do some in silico drug screening, something that I've not shown, but if you're interested, you can go and take a look at the paper. And also, we could find that these changes are specifically happening in certain cell types in the 2 different patient types, which can be used for designing targeted therapies. And then there is definitely a role of the microbiome in the lungs of the COVID-19 patients that needs to be further investigated. And we cannot rule out the possibility that the reason why the patients are not, the patients had a tough time could be because of these infections rather than the COVID-19, because these patients mostly had cleared away the infection. We were not detecting COVID-19 in actually majority, 80% of the patients. So I'll end my talk here and I'll be happy to take questions. Thank you.
Thank you so much, Dr. Ishan, for such an interesting talk. And I have some questions for you. We want to understand, is it the normal patients which were taken or healthy volunteers? Like normal patients suffering from disease, if yes, then what? Or normal patients who survive?
Yeah, so this is a good question. So we took basically autopsies of patients who were suffering from lung cancer. So typically when you were taking autopsies for, or not autopsies, or biopsies, so you would take some tissue which would be normal tissue and some tissue which would be cancer tissue. So we were using the tissue that was normal tissue from lung cancer patients. So these lungs were already inflamed. So they should have had some inflammation signatures. So those were the available samples that we had for ethical clearance.
All right, sir. And even within the G1, the first group, you seem to have two sets of patients based on heat map. Do you want to comment on that?
So, yeah, some set of patients, yeah, they are not really. Yeah, I'm just showing here the top 20, top 50 genes. But when we take a look at like all the genes, which is what these kinds of PCA is taking into account, there seems to be all the G1 patients are much more different than the G2 patients. So there could be some patient-specific variability, which is typically taken care of when we are doing these differential expressions. So yeah, this could be attributed to patient-specific variability. But if we would look at the heat map for all the genes which are involved here to be found differentially expressed, maybe this difference between sub-patients might not be so stark.
Type 12, would you say the cytokine storm signature is different between G1 versus G2?
So actually what we think is that the G1 patients uh, may represent the cytokine storm signature. The challenge is that in the time in which the patients were sampled, it was very hard experiment. I mean, this is not an experiment. This is a very hard, challenging work to do, because to basically take blood from the patients and measure the cytokine levels all the time, typically in the setting that we have in India, is very hard. But yeah, so we suspect that the way that the patients look like, what we could be seeing is actually a cytokine storm G1 patients versus a more classical long-term COVID-19 G2 patients. But this did not correlate very well with the data for how long the patients spent inside the hospital, so we cannot be sure of this. It's a good question. I don't have an answer to that.
So we have a few more questions, but as we are running out of time, would it be all right if I mail you out the questions and then we can answer the questions for our participants?
Yeah, I'll be happy to answer any question.
All right.
And I'll suggest
Thanks.
if people are interested, please go and read the paper. Yeah, it's available. It's freely accessible from PubMed. Because what I said was a very short description of what is there in the paper. It's quite more comprehensively explained in the paper.
So can you share the link of the paper in the chat with us?
Yeah, I'll do that.
Okay. Thank you, sir. So now it's time that we move to our next speaker. I'll just share my screen. All right. So we learned how omics data can be used to reveal the biological complexity of COVID-19 disease. But how does bioinformatics come into play during this process? And what are some other areas where bioinformatics analysis is used? So now we will hear from Dr. Suchi to understand the role of bioinformatics in omics study. Dr. Shuchi Nagar is working as an associate professor at D.Y. Patil Biotechnology Bioinformatics Institute, Pune. She did her PhD from Department of Chemical Technology, University of Calcutta in the field of cheminformatics and drug design. She has 17 publications to her credit. She has guided around 20 project students. Presently, she has 2 funded projects. One is related to designing anti-cancer drugs, and the other is designing quorum sensing inhibitors. Further, her expertise is also being applied for studying Alzheimer's diseases and for generating Ayurvedic formulation. We are pleased to have you with us, Dr. Shuchi. Over to you. You may share your screen now.
Yeah, thank you, Sonalika. I hope I'm audible.
Yes, ma'am, you are audible to us.
Okay. Yeah, so good evening to all. So today, as said by Ms. Sonalika, I'll be actually going into a very brief understanding of what is omics and how bioinformatics is going to be helpful in studying the data that we get through omics studies. And we've just had one talk by Dr. Ishan. So he has used quite a few places. He's used these techniques that we will be just discussing. So it's going to be an overview of the omics technology with bioinformatics rather than being some kind of a specific work that we have done. So I'll just share the screen. Yeah. So I hope the screen is visible.
Yes, ma'am, we can see your screen.
Okay. Okay. So before I start off with this topic of relevance of bioinformatics in omics, I would like to just introduce my institute where I'm working. And just to give a brief, the courses that are offered in our institute is we have BTech with biotechnology, BTech with medical biotechnology. We've just started with BTech with artificial intelligence and data science. There's MTech biotechnology and MSc biotechnology. So these are the few courses that are being offered at our institute. And we have different research areas that are there, which are very functional. And there's a lot of research done on these areas. It's microbial diversity. Then you have plant and environmental biotechnology. You have genetics and molecular biology, cancer and translational research, protein biochemistry, and bioinformatics. So these are the 6 broader areas on which all the faculties of our institute are working. And it is we really have a good strength of faculties who are working and doing really well in all these fields. Now, to start with the topic of relevance of bioinformatics in omics, I just want to say that when we talk of omics or when we are talking about bioinformatics, it's like saying that what has come first? Has the hen come first or the egg has come first? That's because it is a part of omics and part of bioinformatics that is common. So you have certain part of omics wherein that part of the omics is completely done on wet lab. Then you have the data that is generated is stored in certain databases. So you collect those data from there, and then you use the different bioinformatics tools to analyze them. So that is why we can, it's very difficult to say whether bioinformatics came first or omics came first. So it's the same story like hen came first or your egg came first. So we can also say it in this way that so it's like, the omics and the bioinformatics are coming together, joining hands to give you a good information that will further help us in systems biology. So systems biology is basically when we are trying to study different pathways that are there in very details. And followed with system biology, we can go to studying or doing chemoinformatics studies, where we can go ahead with designing of new molecules, that could be lead molecules for a particular disease of interest or a particular target of interest that we have. So to just understand the flow chart that is there with it, what we actually do is, this is your omics part that is there, wherein we are doing certain kind of your, you can say experimental works. You have n number of different experimental works through which you can get data. So all these could be gene data, genome data, it can be RNA data, it can be protein data, it can be any kind of data depending on what kind of your experiment are you doing. That data, and then we take it and we are storing it in different databases, and then using various algorithms and various tools that are available, we are actually doing all these studies, which is actually the omics studies. Okay, so this is your omics studies, which is your genomics, transcriptomics, proteomics, and metabolomics. So we can say that it's the follow-up that is there is, it's like the central dogma. That's what is there. You have genomics, that is the gene data. So that then you come to transcriptomics, which is your mRNA and other data. From there you have your proteomics, so that's your protein data that you have. Then comes your metabolomics. So ultimately the product that is being formed is what is your metabolomics. So I will lead it. I will discuss on each of these omics in details, as in where can these be applied, and what are the different tools that are available for this. So what are the different kind of studies that you can do with different types of omics data that is available. So definitely you have to have some kind of an experimental work. Only after doing this experimental work can you go ahead and do this study, which is going to be using the data that is generated from here, and then the computer and the various computer algorithms and programmings are used to study the data to get some kind of an output. So omics, as I said, is kind of different techniques. So that is why it is a very high throughput technique. That's because whatever data is generated when you are doing any kind of omics work is very huge. And it's not possible for any human to study it in that details. So that is why you do require some kind of an tools. So that is why we use computational tools. Now the data that is generally generated through this high throughput techniques would be, you can have n number of genes that are being generated. You can find out the expressions that you have with regards to genes, various proteins that are being expressed, and also it could be a combination of any two or three, all the three of them. So depending on what kind of study you are doing, that will tell you what kind of data you are getting in. And that data can be then analyzed using computational methods, which is the bioinformatics part, because the bioinformatics is taking into consideration the biology as well as the informatics. It is combining it to give you or helping you to solve certain problems that otherwise would have been difficult, or you can say it could be time consuming at the same end. Say for instance, you have generated a lot of genes, around 1,000 genes for instance. So analyzing that genes and seeing which gene is the most apt is very difficult if I say it from human point of view. Using computers, that is what becomes more simplified. So so omics is basically signifying the measurement of the entire complement of a given level of biological molecules. The information that is obtained is basically encompasses a variety of new technologies, and that helps us to explain how there is a differentiation that might exist between, say for instance, a normal and an abnormal cell. And when I'm talking about the differences that is there between normal and abnormal cell, I'm also talking in terms of whether there is some kind of a pathway that is affected, whether there is some kind of a gene expression that is getting affected, is there any kind of a mutation that you have come across, or whether everything goes good, but at the end point when there is the final product that has to be obtained, there is a problem with that. So whatever the place, which we say the 4 omics, all these is what we can study using the tools and the databases that are available for analyzing things. So why do we use bioinformatics? Is obviously because computers is very much helpful and giving us faster results. Along with computers, we also use certain statistical approaches, and that's the best way of studying the variation. And it helps us to perform extensive omics related research that which is present in the biological database, and that helps us to do some kind of comparative studies. Thus, at the end, it helps us to identify sequences of proteins that differ between diseased and healthy tissues. So the branches of omics are, you have genomics, which is nothing but your quantitatively studying of protein coding genes, whether you have regulatory elements and non-coding genes. So these are few things that we study with regards to genomics. Then we have transcriptomics, that is your RNA and gene expression. Then is proteomics, which is focusing more on proteins, and metabolomics, after proteins, whatever we get as a byproduct or as an end product of any kind of a pathway is metabolomics. And that also indirectly helps us to study the various pathways that are involved in that. So it is a clear, you can say very much clear follow up from your central dogma, from genes to mRNA, to DNA, to mRNA, to proteins, and then to the product. So what can we achieve at the end, or what are the things that we can get after you do any one omics study or a combination of omics studies? You can do data processing. You can identify certain proteins that are being expressed. You can go away with statistical analysis. You do pathway analysis, and at the end, you can also do various type of modeling studies, wherein when I get to know, so target identification is also one thing that can be there when I'm talking about your pathway analysis. So now focusing on individual omics that we have. So when I'm talking about genomics, we always obviously get sequences from various experimental sources, and then they are stored in the databases. Now you have two kinds of databases that are available. You have primary database and you have secondary database. Now how they are different is, when I'm talking about primary database, I simply have done some experiment and that data is stored in the primary database. If I further analyze that data and study as to say what is its functionality, where is it expressed, what is the situation under which it is expressed, all these things, then the information that can be stored would be in the secondary database. From here, then we can retrieve the information. So since it is genomics, we retrieve the sequences that are available over here. And after getting the sequence, we can do various studies. The first thing that you can see over here is we can do sequence similarity. And this sequence similarity, you have it, you can do various types of sequence similarities over here. Okay. So sequence similarity would be, say you are doing pairwise alignment. So so you have sequence similarity, which can be, say, pairwise alignment, wherein I am trying to see whether the same gene from two different species, how far they are different from each other or how close they are to. So what is the one thing that is varying? Say, for instance, if I'm talking about hexokinase, that is present in bacterial genome as well as in, say, a plant genome for that matter. So how are they different? Because the length of the genome is different. Sorry, the gene is different. So what is it that is varying? That is going to help me to understand how there are things that are changing down the line from by evolution. And even though, there's a change in the length of the genes that you are getting, though both of them are for the same enzyme, but still the length is different. So what has changed down the evolutionary tree, because of which, even though it is coding for the same particular enzyme, still my sequence length is different? Or what kind of a variation is actually existing? The second type that we can do is we can do multiple sequence alignment, wherein we can actually again compare and find out similar genes or similar enzymes from the same same species. So I have, say, just E. coli. Under E. coli, I have different subgroups that are there. So how these are different from each other? A particular say a particular protein is present in all the sequences, but still what is that is different at the genetic level is what we can do with regards to genomics. And a very frequently used tool that you have is BLAST, that is used for doing that. Or you also have MBOS, that is used for your multiple sequence alignment. The next is gene prediction. We get to find out because we already have databases that are stored. So in case when you get some kind of a sequence, say after performing any kind of an experimental work, now by doing a searching for those genes that are present in the database, whichever gene we see that there is a similarity that is there, that is one way in which we can find out the genes. On the other hand, we also have many tools that are available, for example, gene parser or UGenes, which help us to find out which gene it is, what is the functionality of the gene, what is this gene coding for, or is there any kind of a difference that is there in this gene. Say I have got this gene from some kind of a diseased person. So what is it that is different from this person and the normal person for that matter? Any kind of a mutation can be studied using the tool mutation tester. We can study the variation that is there within the species. So n number of different species that are there, we collect the genes of the similar kind, and that's how we can study the variations that are existing. And obviously, we can study the evolutionary relation that is existing among various organisms that are present. Okay, so these are your different types of tools that are available for doing their individual tasks. So we can, at the end, what all things that we can do? We can predict or go ahead and find out biomarkers. We can obtain evolutionary history. We can assess risk management. We can also know about the bioarchaeological, anthropology, evolution, and human migrations. So it's not just restricted to any kind of disease related stuff. You can also go about doing a lot of things over here, like your archaeological survey, anthropological survey, or even for that matter, human migrations. So these are the applications. So you can use the genomic study to find out molecular medicines, and it's very much going to be effective with regards to doing personalized medicine. Then you can go ahead and study microbial genomics. So as in the present scenario, you can see that the gut microbes are said to be of very much playing a very high role in any kind of a disease state. So you can obviously go with the microbial genomics, where you can study the gut microbes in details. Risk management would be with regards to whether you are capable, or whether there are chances of you having a particular disease in the future. So there are certain diseases that are genetic in nature, certain diseases that you have it from due to the environmental stress that is there. So you can definitely go ahead and do genomic studies to find out if there is any kind of risk related to these things. Bioarchaeology, anthropology, evolution, these are again various ways in which we can use the genomic studies. Then you have DNA identification. You can also use it for agriculture, livestock breeding, and bioprocessing. So in short, what I can say is with regards to genomics, it's not just restricted to your disease and non-disease states or things like that, but you can also have its application in various categories. Next, coming to transcriptomics. So that's transcriptomics means now we have the mRNA that is available. So you do have, again, databases that are there which can be used, where you have the mRNAs that are stored. Now, what you can do first is you can find out the exons that are available, or you can directly use the database that is stored. From there, you can obtain the RNA sequences and convert them to your amino acid sequences. After doing that part, you can do again various kind of studies that are there. You can study the gene regulation studies. So after you get these sequences, you have a lot of genes. So how well are the genes regulated? Say again here, what you're going to actually do is you're going to take it from a set of people who are normal and say a set of people who have some kind of an abnormality or something which is different from others. And accordingly, we can see. Say, for instance, if I just talk about a situation in COVID where we had seen that the Indian population were not that much affected as against the US population or the European population. So we could always do this kind of a study to see what is one thing or two things that is present in the Indian population or absent in the Indian population, and vice versa with, say, US population or the European population, wherein this kind of a discrepancy is seen. You can, so this is just one instance. You can have plants that you can see that they are growing, having the same environmental conditions. They are growing in a particular area, but they're not growing in some other, say, country. So what is it exactly that is affecting? How are the genes getting regulated? Which gene is the one that is being regulated at a higher level or regulated at a lower level? These are the regulatory gene studies that we can do, which can give us some kind of a solution to solve the problem that we are facing. At the same time, you have gene expression studies also. So again, gene regulation, when we are talking, what are the genes that are expressed? So many a times it might be possible that it is expressed, but it doesn't go further. So those kind of studies are also there. We can get to know what are the genes that are upregulated, what are the genes that are downregulated, and that helps us to understand the biochemical reactions that are taking place in the system of one group and the system of the other group. You can observe mutation at the mRNA level, and obviously you can go about doing the evolutionary variation studies that are there. So that will help us in prediction of biomarkers and also help us in searching for new targets that would be helpful for drug development. So various approaches or applications of your transcriptomics approaches are, you can do transcriptome assembly and profiling. You can do small RNA characterization. You can do mapping of the metabolites, protein and transcript profile. You can do population breeding studies, and you can do regulation of gene expressions. So these are the various tools that are available for studying the transcriptomics data that is available, or that we can get it from the various experimental techniques that we have. Next, coming to your proteomics data, again, we get the RNA sequence. From there, we can go about doing the predicting the uniqueness within a family of proteins. We can predict the structure of the protein. We can study evolutionary relationship. We can observe the sequence changes that occurs between normal and disease states. So most of the applications you can see is quite common to all the omics studies that is going to be there. It's just that depending on what data is available to you, you are going to do accordingly. One more thing that we need to keep in mind over here is, as we go down the ladder from, say, genomics to transcriptomics to proteomics to metabolomics, we are narrowing down so it becomes much more reliable data. The observation that we see with genomics data compared to the observation that we see with proteomics data would be very different. Proteomics you can rely more on as compared to genomics data. Okay. So all these things are there. You can always, when you have the proteins, you can always obtain the RNA sequence. And the second is where we always get the 3D structure available in the databases. From there, we can do any kind of an interaction studies. So if we get to know, say from transcriptomics, if we get to know certain genes, that are playing a very important role, or say there are certain genes which are getting upregulated in a particular disease. So those genes become our target. And from there, we can, if the structure is available, well and good, we go through this protein. If the structure is not available, we can do the prediction of structures using the information from the evolutionary relationships or some kind of a uniqueness that is present in that protein family. So this information as well as the domain information help us to predict the structure of the protein. And after getting the structure, we can again go about doing all these studies. Okay, so when we have the sequence itself, we have the structure itself, by doing functional similarity, we can go ahead and do functional predictions. We can predict the domains within a family with regards to the known structure as well as just through the sequence. So at the end, we can find structural changes for the same gene expressed and different environment and tissues. So you have one gene that is coding for, say, more than one proteins. So we can also see that whether the same gene is expressed at the same level into different environments or not, or what is one environmental conditions, or what is one condition in which it is expressed in tissue X, and in the change in the condition is helping it to be getting expressed in, say, the tissue Y. So those kind of information again can be gathered through the studies of proteomics. So again, the various applications that we have is, you can go for proteome mining, you can go for protein expression profiling, post-translational modification, protein-protein interactional, structural proteomics, protein quantification or differential analysis, and functional proteomics.
Thank you, Dr. Suchi. Actually, I think we are running out of time. So if you can conclude the session.
Yeah, so these are the various tools that are available. Then you have metabolomics that is helping us at the end. And this is also a very important place where you can get good prediction of markers, because the initial, you can say the samples that are there is not something which is going to be invasive. So that's the importance of metabolomics. You can use just say blood sample, urine sample. These samples are enough, so we do not have to go to too much of ethical issues when we are doing metabolomics as compared to genomics, proteomics, and transcriptomics. So these are the various applications of your metabolomics. So very quickly, I'll just discuss a small work that we had done that would probably help you to understand the omics that is there. So what we had done was, we had taken few data sets from your GeoNCPI data set, and we had applied Geo to R to get the information that is there with regards to test and control sample. So our actual work was to sort out the differentially expressed genes which are upregulated and downregulated for Parkinson's disease, and those who are tobacco smokers and those who are not tobacco smokers. Because it was seen through literature that tobacco is helping to delay Parkinson's disease. So we were just trying to see how this is affecting the effect of tobacco is there on the expressed genes. So which are the genes that are being expressed, upregulated, and which are being downregulated. Then those genes we had taken in David, and we had seen what are the different pathways that they are getting affected. So we had obtained around 17 genes that were there. And now we are in the process of finding out new molecules, that are interacting with these, so that we could suggest some new molecules that could be helpful for delaying of the Parkinson's disease. So that's how we can actually go about using the whole omics study, using different bioinformatics applications to get down to the conclusions. Okay. So take home message would be, you understand what is omics, you understand you are able to do personalized medicine, which is very much coming in the recent future. You get to know about the different pathways involved and the genes that are expressed. And the whole study is actually very much helpful in prognosis, diagnosis, and treatment of any kind of a disease or conditions. And I would like to acknowledge our director, my colleagues, my students, and my family members for their constant support. And last but not least, I thank the organizers for helping me, for allowing me to giving me this platform actually to share the information that I have. Thank you. And any questions?
Thank you, Dr. Suchi, for this in-depth
Thank you. And I'm
presentation.
sorry to take a little bit of time extra.
No problem. Thank you for this in-depth presentation and highlighting genomics and its application, transcriptomics and its application, and the entire flow chart or one of the common pathways to do various bioinformatics analysis, showing how data and bioinformatics could be applied not only to diseases or healthcare, but other areas like evolutionary studies and other biotechnology areas. I think the audience and our participants have been benefited a lot from this talk and knowing some of these resources that you have discussed, and also the case study. Thank you so much for your time and for this presentation.
Thank you.
All right, so now it's time for our next speaker, and we are really excited to have Dr. Anshu Bharadwaj with us, who would be talking about understanding and addressing genotype-phenotype correlation in complex diseases. As we have been discussing, Dr. Anshu Bharadwaj is a biologist and a data scientist. She has obtained her PhD in 2008 in understanding of genotypic-phenotypic correlation in human mitochondrial diseases from CSIR CCMB Hyderabad. She has over 40 publications to her credit and have trained over 500 young students, and also has served as an associate scientific advisor for science translational medicine, and is on the editorial board of Journal of Genetics and Frontiers of System Biology, Protein Bioinformatics, Computational Genomics, and F1000 Research Galaxy Gateway, where many of our students have already published some of these articles that they have worked on. So we are really delighted to have you, Dr. Anshu Bharadwaj, and we are looking forward to your presentation and learn from your talk. Thank you. Over to you, ma'am.
Thank you so much, Mohit, for that nice introduction. So can you hear me clearly?
Yes.
All right. Are my slides visible?
Yeah, they're coming up. Yeah, they're visible now.
All right. So first of all, thanks for giving me the opportunity to actually talk to students. That's my, that's my lifeline. So what I decided today, since the entire symposium is organized around bioinformatics and data science training and research, I have kept the talk very, very simple, but we have included one component of our work so that you can also see the application of how it can be done, again, in the most simplest possible fashion. So my lab primarily works in two domains. One is basically genotype-phenotype correlations when it comes to infectious diseases as well as mitochondrial diseases. And the second vertical, which is a smaller component, is computational drug discovery. So just a second. So as you can see here, there are several projects that we have done over years. But today I have selected projects which are on the bottom panel of this timeline, which is mostly to do with mitochondrial diseases, because I thought genomics is something which is basically picking up the pace. So it's important that we understand the genomics basics in terms of where are we going, where do we stand today. So as we all know, DNA is the blueprint of life. And one of the first objectives is to actually learn to read this DNA, right? That was the first thing we started doing, because till the time we read what is in the blueprint, we cannot make sense out of it, right? So in early sequencing days in like 1960s, the first RNA sequence, which is of alanine tRNA, was sequenced. It took 5 people for 3 years working to determine just 76 nucleotides. So this is how we started sequencing. We were not sequencing genomes in the beginning. We were trying to standardize the technology to understand how you read this. And subsequently, over years, we started taking baby steps, where we went ahead from that to 1 base per month, then to 1,000 bases per day. And by the time we sequenced the human genome, we were already doing 10 million bases per day, right? Until that time is when, we actually, this is a quiz for kids who would like to enter their answers to say what this event is. So this is basically a landmark event in the history of current biotechnology and data sciences, is when we launched the draft of the Human Genome Project, right? And this is the big machine that at that point was used to sequence it. it took effort from several countries and a very large number of sequencing centers to give us the first draft of the human genome sequence, which was 23 human chromosomes with 3 billion bases, right? Why were we interested in sequencing? Simple reason: to understand human disease and evolution. If you can compare the genome sequence of somebody who is diseased versus somebody who is normal, the anticipation was that we will be able to understand diseases better at a molecular level. And if you can understand something better, it is easier to treat it. So as the previous speaker was referring to a large number of methods for this kind of data analysis, the objective is very clear that you need to understand human disease and evolution, and that is why there was an intense amount of effort put into place. But this was only a project where you were trying to understand and standardize the technology. And then we went ahead from this basic sequencing methods to something we understand as next generation sequencing methods. And with the advent of NGS, now there are a very large number of sequencing projects going on across the world, where a few countries are actually sequencing their entire population. So these sequencing efforts actually have gone, the scalability is unimaginable. We are generating data at a pace and we don't really know how to analyze it. So that is where the power of data sciences lies in. It is very critical right now for students who are basically deciding on a research objective. Try to learn data analysis, because you actually have a lot of data available that needs to be analyzed in context of what is now disease associated among all this plethora of sequence that you see. And the starting resource that I would recommend to all of you is GenomeAD. So Genome Aggregation Database is trying to aggregate all the genome sequencing projects at one place. So you don't have to wander around several places for this kind of data. So you can go to GenomeAD for that reference. Now, when you say you are trying to compare individuals with and without disease, what are we trying to compare? Or what are we trying to look at? We are trying to come back at a point where we can look at genomic variations, right? Now, genomic variations can be of several types. The most common ones are single nucleotide polymorphism. These are called, in general language, substitutions. We'll read more about them in the next slide. Then there are indels, which is insertion deletions, and they can actually change the protein function. Then there are structural variations. Then there are tandem repeat variations, segmental duplications, translocation. And most of us in our undergrad or in masters must have come across these terminologies, where these actually are impacting the way the chromosome is, or the sequence is arranged in an order. And then when that arrangement goes haywire, you expect to see that there can be a disease phenotype. Now, when you talk of single nucleotide polymorphism, it means that it's a variation at a single nucleotide. Say, for example, let's say there is a gene A, and that gene A has an exon, right? We all know what exons are. In exon 1 at some position, let's say 500, in a population, you either see A or C. Now, in technical terms, we call alleles at the nucleotide level. So there is an A allele and there is a C allele. Now, let's say A is the most common allele, like 90% of the times, A is observed. So we call it as a major allele, and C is observed the rest 10% of the time. So it's a minor allele. So since humans are diploid, we get one pair of chromosomes from our mother, the other from the father. Then, so basically what you actually will see is that either the individuals will have AA, AC, or CC, right, at this particular position. Now, how do you decide that it is an SNP? Is when the minor allele, and in this case that is C, is present at a frequency of more than 1% in the population. So let's take two examples. In the first case, if 94% people have C, which is the major allele, and 6% have T, which is a minor allele, you say this as an SNP. In the other case, if you see a similar situation, but the percentage of the minor allele is not even 1%, that you start calling it a mutation. Now, this you need to understand is heavily context dependent. The reason being that if you are looking at rare variation, then this definition will change. And that is one has to now look at single nucleotide polymorphism definition or genomic variation in context of what you are trying to look for. And obviously, when you are looking for rare variants, you need to sequence a very, very large number of individuals. And then of course, when both the maternal and the paternal allele are same, that's an homozygous SNP. When they are different, it's an heterozygous SNP. And they are present everywhere. Like they can be in UTRs, introns, splice site. They can be in coding regions where they may or may not change the amino acid. In almost all these cases, they might have an impact on the phenotype. Now, the question that you might ask is that you sequence an individual, sequence another individual. So one person has disease X, the other person does not have disease X. Just compare the sequence and you are good to go, right? You can now actually identify the differences and say this is what is causing the disease. It is not that simple, and that is where what we are discussing today. Any complex disease is not happening because of a single genomic variant. There are many genomic variants which we call as polygenic. In interaction with the environmental factors are actually causing the phenotype. In this case, I have shown the example of coronary artery disease. So complex disorders, I must repeat this because this is a very, very important factor, results from contributions of multiple genomic variants and genes, and that is why they are called as polygenic, and they work in conjunction with the physical and the social environment to bring out the phenotype. And this is the challenge we are trying to now resolve. When you compare two individuals, you will get anywhere between 3 to 5 million SNPs which are different. Which of those SNPs are associated with the disease? That's the first question. Second, which of them might cause the disease? There can be several reasons why the disease is manifesting. And that is where the challenge lies. In order to address this challenge, there are two basic approaches that are applied. One, we call it as a candidate gene approach. The other one is a genome-wide association studies. And we'll very quickly look at both of them. What do you mean by candidate gene approach? So in a candidate gene approach, what you actually are doing, you are basically making an intelligent guess. Like say, for example, you are interested to identify a genetic risk factor for a particular disease condition, which is a complex disease. And you now need to know what are the genes contributing to this phenotype so that you can now look at the variation in those genes. Now, this mostly is done within family members, or it can also be unrelated cases. But the point is you need to have some estimate of what kind of biological components might be involved. Say, for example, I've taken an example here. If you are studying alcoholism, why some people are getting addicted to alcohol, and you want to know the genetic components of that, the most logical choices would be to look at their alcohol metabolic pathways. And particular genes like alcohol dehydrogenase or aldehyde dehydrogenase, these are logical options for you to start. And if you do see that there are several alleles of these genes in a population, making it a very strong case that you now know that they are actually vary among individuals, now you can actually apply a candidate gene approach, start looking for variation only in these set of genes, and then identify polymorphism which might be associated to the disease, and subsequently establishing why this might also cause the disease. So here you are actually also digging deep into the mechanism of what that variation might be doing. The problem is, in most cases, this kind of educated guess either has failed or it is extremely difficult to make. And that is where genome-wide association studies are done, where you are actually following a very data-driven approach. You take two cohorts, two groups of individuals, one normal for the phenotype, the other one actually has the phenotype. And when I say phenotype, I'm saying the disease. And then you sequence them, compare, identify SNPs, and then start looking for association of these SNPs with the disease phenotype. Now, the word associated is kept very, very categorically here. Reason is association is not equal to causation. You may find SNPs which are associated with the disease. They might not be causing the disease as such. So GWAS actually will help you identify, at least shortlist those candidates and then explore them deeper with respect to what is their role in disease manifestation. Several studies have, actually there are many GWAS that have been done till date, and we do have some success stories where if you look at this publication, they actually have listed several traits against which genes were identified using GWAS, and they ultimately actually allowed discovery of drug candidates to treat these diseases. So we do have success stories, means these methods work. Obviously, the number of GWAS that has been performed versus the outcome is not comparable. So we need more intelligent methods of doing that and maybe a better readout. So what actually is going on right now is there is a very big effort going on to catalog genomic variation of different populations all across the world, and then also trying to assess which of these genomic variation might have a clinical implication. So there is, if you look at, I have listed a few papers from last 2 to 3 years. Some of them are old like ClinVar, but ClinVar keeps updating very regularly. Then there is the Genomacia 100K project, but some of them are very new as this month. So I would recommend that you actually read these publications. It will give you in context of what is really going on, how are these genomic variations documented and analyzed, and why do you need these kind of resources when you actually have a reference genome. So you actually, reference genome is just, it's like 70% one individual's readout, despite the fact that when the genome sequence was done, they were like the pooled sample of 20 individuals, but it is mostly read out from one individual. So it's not really representative of how diverse we are. And that is why you need to sequence from various populations in different contexts. Now for today, I have kept a very simple example of what we did in our lab, and I'll be very quickly describing that. So you need to focus because I'll very, very quickly finish this. So our lab has been interested to understand what is the role of mitochondrial DNA in human diseases. And if you look at I've just shown a representation of mitochondria. If you look at the electron transport chain, it has 5 complexes. The ones which are shown in green are encoded by nuclear DNA. The ones which are shown by red are encoded by mitochondrial DNA. It is not very difficult to make a judgment that there has to be a very intense crosstalk between the 2 genomes for mitochondria to function properly. And it is not just the energy metabolism. Mitochondria is involved in practically all central metabolic pathways in the cell, making it an extremely critical organelle. And given that this organelle has its own DNA, makes it a very strong case to look at what mutations in mitochondrial DNA might lead to disease phenotypes. Now, one of the things, not everybody understands the structure of mitochondrial DNA, so I have kept a very small introduction. It's a circular molecule. It is 16 kb in size. It is mostly maternally inherited. There are 2 rRNAs, 22 tRNAs, and 13 protein coding genes in mitochondrial DNA, a single non-coding region, which is D loop, and over 3,000 other proteins are required for mitochondria to function properly, which are encoded by nuclear DNA, right? And there is a very, very big repertoire of genome sequence data available across different populations that one can use to now look at disease association context of mitochondrial diseases. The problem with genotype-phenotype correlation in mitochondria is that when you actually see these point mutations, the symptoms manifest in many, many different ways, making it very difficult to understand or establish pathogenicity of a change in mitochondria. And that is where we developed an algorithm, which we called as BIT-SNP score. It is published long time back. And the objective is, how do you score variation for their disease association? Again, I'm not talking of causation here. So it's very important that I keep using the word association again and again. And comparison is performed against normal data sets. So I'll very quickly show you what we did. So first things first was, I needed to understand which kind of mutations in mitochondria have an experimental evidence that this actually might cause the disease. So that was my first objective, is to first list out reported biochemical defects, which are like low complex activities, or functional studies, which are done using trans mitochondrial hybrids, to establish that some of these mutations are actually causing the disease and not just associated with them. Use them as a basis to select which tools are good to now select these kind of variations from the plethora of other variation that I see. And then subsequently, I designed a scoring scheme, which I will not have time to explain today. But the whole point is, if a variant is of very little importance, it was given a score of 1. If it is of very, if it is really causing a problem with mitochondrial replication, say for example, it is given a score of 10. So between this 1 and 10, the other scores that you have seen here are assigned based on the anticipated impact that these mutations might have on the mitochondrial function. And then when you actually have several such mutations in an individual, we multiply all the scores and not add them. So this is the AND logic. I would request you to go and read the paper on why we applied this logic. It's very interesting. And once we did that and the pipeline was ready, we actually applied it on patient data sets, which are ataxia and MetSNP, and we applied it on almost 600 individuals, which are both having metabolic as well as neurodegenerative cases. Okay. For normal sequences, we use the context-dependent normal. For Indian ataxia patients, we used the Indian normal data. For Japanese disease individuals, we used Japanese centenarians so that the background variation effect is not there. And centenarians were an extremely good control set because these are people who were 100 years old and they were absolutely healthy, which means any mutation in these individuals might not be causing the disease by themselves. So when we applied this whole algorithm on the 600 plus individuals, whole genome mitochondrial DNA data sets, we could actually prioritize only 3 to 4% of total variations. So it's not 5 or 600 between any two individuals. We actually could prioritize very, very small set. And obviously, we also were able to identify novel variations in some genes which were not reported before. So it was very interesting to see that the method was working and we were able to identify some novel variants which were not even reported by that time. So basically, this is a new, this actually still is something which most math methods cannot do. One observation we made back then was that rare variants are high scoring variants as opposed to the general understanding that common variants are disease associated. We didn't observe that even at that point. And now there are more and more papers who are actually trying to scout for rare variations for disease association. So this is one of the first studies where we could actually observe this and reported it. And we could find novel disease associated variants with this application. Moving forward, again, I'll not be able to describe much of this work, but since you know that in each cell, there are several mitochondria, and within each mitochondria, there are several mitochondrial DNA, and we could actually assess this mitochondrial DNA for disease association. The second logical thing was to now look at more than 3,000 proteins which were coded by nuclear DNA, but have a mitochondrial function to see the holistic impact of variation in an individual, both for nuclear and mitochondrial DNA for disease association studies. Now here we used several resources to identify which nuclear encoded proteins have mitochondrial function. And just bear with me on this slide for a minute. So if you look at the left-hand panel, we utilized several resources to identify nuclear encoded proteins with mitochondrial function. Identified which of the mutations in these proteins actually have been reported to be pathogenic in ClinVar, which means they are clinically actionable, and mapped these variations onto the Genome Asia 100K project, which actually also has data on almost 600 individuals from India. We then took all these mapping where you actually found this and map this to DisGenNet data, which is genes and variations associated to human disease. So we could generate a mitochondrial gene disease network. And in this network, we could also identify loss of function variations from genome data sets. If some of you actually find it difficult to follow in this presentation, you can always write back to me, because there is a lot of background information one needs to know to understand this. The logic that you need to understand is, what we are trying to do, we were trying to see whether pathogenic variations reported for the rest of the world, are they also pathogenic, or are they also loss of function in the in the Asian subcontinent or particularly for Indian population or not. What we actually observe is that one needs to perform population specific mapping of clinically relevant variation. We cannot rely on loss of function variation data coming from the rest of the world to our population, because we don't see that happening. So this is very important now, that one has to generate population specific references and population specific variation data, which might not be reported actually for the rest of the world because they're not LOF there, but they can be here. So this is a very important message. And this recently was also endorsed by Genome AD in the recent paper, where they actually say the same thing. Now, one of the things that one can do with this kind of platform is you can study comorbidities, because now it's a network. We are saying maybe one disease and several genes, and one gene and several diseases. So this is something that we are looking at. And particularly from Indian population context, when we did enrichment analysis, we could see that certain pathways like defects in vitamin and cofactor metabolism, tRNA amino acylation, DNA repair pathways, these are very, very important to study in Asian population context. So this is the first platform that we have identified to look at comorbidities. I didn't actually focus it from a comorbidity perspective because it requires a little bit more background to understand it at a more detailed level. But if any one of you are already working on this kind of strategy, we have actually provided all of this resource as a workflow system in Galaxy. So no programming experience is required. If you have a question, you can come on this platform and generate all your own custom workflows on the fly. You really don't have to learn anything to make this, to basically ask questions. And a lot of information is already provided as part of this platform, and we have already analyzed more than 150,000 individuals with almost 600,000 variations. So it's a very large data that we analyzed for this project. Now, some of the things that I'm not discussing today, but I would really recommend you to go and read them, is that many times you will actually hear that synonymous variations, the variation that do not change the amino acid, will not have an impact on the disease phenotype. That is not true. We actually have shown in this work that it actually can have disease association impact. And second is, since the data size that we are now looking at is going to, by 2025, in another 3 years, the genomic data will bypass the data that we generate on social media. So now you can imagine we are talking of exabytes of data. We have not even talking of petabytes now. Now that kind of data when you not only have the challenge of genotype-phenotype correlation, you also have the challenge of analyzing and handling this data, and that is where we developed Frog. Again, it's not for discussion today. So basically what we are now doing with all this computational platform, you might say, what is the application of this? So the application is pretty evident. In 2016, the Mitochondria Medicine Society actually recommended next generation sequencing as the first line of diagnosis for mitochondrial diseases. So it is not something that you might do. This is something which is a highly recommended method to diagnose mitochondrial diseases. And I'm part of the clinical domain working groups. We are actually trying to map every variation as per the approved guidelines by ACMG and AMP standards that we recommended 2 years ago and were approved by ClinGen. And now we are working as a clinical domains working group with a very large team of clinicians to see how do we actually translate this knowledge into a clinical context. Now, my lab has been developing these resources over a very long period of time, and we continue to do that from different contexts now. We are now trying to see in one of our new projects is how pathogenic variations in mitochondria make individuals more predisposed to infections. Because the other vertical of my lab work on infectious diseases, so we are now merging the two domains in a very logical context. Now, I would like you to tell that why is this important? Because these are basically the 4 tenets of personalized medicine. Personalized, as in you need to know how genetic variation drives individual treatment. It has to be predictive. One should be able to identify what kind of disease condition one is predisposed to. it should be preventive. If I know that somebody is predisposed to diabetes at the age of 30, there can be lifestyle changes introduced and the person may not manifest the disease at all. And it should be participatory, like the patient has to have informed choices and take responsibility of their own health. Now, one thing that I would recommend all of you to go back and check is the new developments happening in precision medicine. So we have evolved from the reference human genome as a reference to population-specific genomes to human pangenomes. Because now we know, as and when we are sequencing more and more human genomes, there is additional 4 to 5 million sites that are getting added to the data sets, which means that we need to now capture the complexity of human pangenome with respect to precision medicine if we have to go anywhere forward from this point onwards. So have a look at the human pangenome's progress. That's something that is very, very recent and is still being discussed on how do we really do it. There are recommendations, but they are still not implemented. So this is something which is an ongoing activity. So I would recommend any one of you who is interested in genomics should be reading about this context. Now, I would like to basically take last 5 minutes to before I close is to tell you that genomics is not only about personalized medicine. And I'll give you an example here. Now, Kathleen Folbeck actually was imprisoned for killing all her 4 babies, right? It is actually said to be one of the most horrific crimes that somebody can perform. Now, the problem was that scientific evidence, so Mayo Clinic pediatric cardiologist, actually Michael Ackerman and a team of scientists, linked the death of these babies to a specific mutation which was leading to sudden infant death syndrome. So this lady actually has not killed at least 2 of her babies, is that what they could establish. For the other 2, they required data from the father, which was not available. But the whole point is that imagine genetics actually freed this woman who was convicted of killing her 4 babies. So genetics and precision medicine, this precision medicine is for health, but genetics and genomics in general have a wide array of applications. Like even within our lab, which I have not told you today, we actually use it to understand AMR. So there are a wide array of applications that you can think of. And these are all places where you can probe as future prospects for your career. Now, as I told you that my lab works on 2 domains. One is AMR and the antimicrobial resistant infections, and the other is mitochondrial diseases. I would like to show you something very different in last 2 minutes that we have done. So for some of you who does not understand what antimicrobial resistance is, so I'm actually shifting gears here. So there's no more genomics from now onwards. So I'm pretty sure all of you know about the discovery of Sir Alexander Fleming. He discovered the first natural antibiotic, penicillin. And in his Nobel Prize speech, he actually said that if we don't take penicillin or other antibiotics carefully, the bacteria will develop resistance against these antibiotics and they may no longer work. Now, this was something that he said in 1945, and this actually is coming out to be true because now we are dealing with the problem of antimicrobial resistance. It is a major issue. I wanted all of you to know about it. It is called a silent pandemic because we don't discuss it the way we discuss COVID. But WHO has declared AMR as one of the top 10 global public health threats facing humanity today. So it's a very major problem. And that is why we thought it is important to sensitize younger generation about the issue of AMR because it cannot be solved without participation from all of us. And we also wanted that this participation need not start when we are already adults. What if we can explain this concept to young children, as young as 11, 12 years old, 13 years old? Can we explain a very hard scientific concept to them in a manner which allows them to change behavior? It is not just understanding something, it should influence individuals in a manner where they are able to change their action against something which is not happening right. So I'll, with the permission of the organizers, I would like to play a 1-minute video and I hope you enjoy it. This is basically a teaser for the game so that you know what the game actually talks about. I hope it works. Here we go. So this is a video, and I thought it's important for the community, as you know, we are scientists. So if it is a problem that impacts all of us, it's something that we have to collectively do. And we am very happy that when we developed this game, it became one of the finalists in the International Educational Games competition across the world. So I would really like all of you to go to the Jigyasa or my homepage and play this game, popularize it so that we actually can contribute to this social issue, which is AMR. It's not just a scientific problem anymore. So as Mohit was saying, I work with a very, very large group of people who are not always sitting in my lab. So I do a lot of crowdsourcing projects, and I have worked with some brilliant kids. They are doing excellent work right now. These two kids happen to be my favorite because they published in international peer-reviewed journals from my lab when they were still finishing their school. So they were very, very young at that point in time, but extremely talented. And it actually, what I could see in them as talent was later endorsed by the fact that Pooja became the Gates Cambridge Fellow, Ashma became the Goldwater Scholar, and they both are on full scholarship for their master's and PhD programs. So this is a message to the students out there. We will be discussing more in the panel, is that you don't need really an age or a degree to do science. Passion is what should drive you for doing scientific discovery. And I have, like my experience with school kids, it's extremely rewarding in that context. A recommendation, which I'll again highlight in the panel, is that be like a Swiss knife, interdisciplinary. Today, if you are doing biology, learn at least one programming language. Very strongly recommended. The kind of data that is being generated, it will be extremely difficult if you don't know data analysis. So do that. And with that, I would like to conclude my presentation for today by thanking my lab members both in India and France. And I thank all the funding agencies who have supported our lab over the years. I would also like to thank my institute. And I'm also happy and very proud to tell you that we are among the top 4 institutes worldwide who actually contribute to biological databases, open source biological databases to the world, and they are highly cited. So this is a proud thing for India because we are actually in line with EBI and NCBI. So I'm really, really happy to share that with all of you. We do some 3D printing, not bioprinting in the lab. So if some of you are interested in doing that, you're very much welcome to come here and use this device. And of course, there are open PhD and postdoc positions in the lab. So if any one of you are interested and you saw a brief of what my lab does, if you need to more, we can chat and take it forward from there. So with that, thank you so much again for having me here, and I'm happy to take any questions.
Okay. Thank you so much, Dr. Anshu. That was indeed a very interesting talk. And yes, we have a bunch of questions that we would like to ask you, but as we are running out of time, I would be emailing you these questions and then get back to the participants with the answers. And it's time now that we move to the academic panel discussion. All right, so from COVID-19 disease to Alzheimer's disease, so far we have learned about the various active fields of academic research in India. But as a student interested in pursuing a career in academic bioinformatics research, few questions might arise, such as what is the future of bioinformatics and data science in India? What are the various challenges and opportunities that the field might face? I would now like to invite all our panelists and our moderator for the session, Dr. Mohit Mazumdar, to please turn on their cameras. And now it's time that we have the academic panel discussion on future of bioinformatics and data science in India, challenges and opportunities. As I had introduced Dr. Anshu and Dr. Suchi, I would now like to take this time out to introduce Dr. Sagar. Dr. Sagar is working in the area of computational biology from last 15 years. He has served as various academic and administrative positions in academic institutions. He has published 30+ research articles in peer-reviewed national and international journals and has one patent as well. He has received 49 grant from BSD, served to conduct research on asthma and allergic diseases. He is a life member of Alzheimer's and Related Disorders Society of India and Biocruise Society of India as well. He has delivered 10 invited talks in national and international conferences and seminars. We are pleased to have you all among us. And Dr. Mohit, I would now like to pass on the stage to you to take the session. Over to you.
Thank you, Sonalika, and it's an honor to be here on this academic panel as a moderator with all the educators here amongst us. So one of the main objective coming to this session and learning about the different application of bioinformatics and leading to the fact that the very important fact is about the interdisciplinary nature of this subject, bioinformatics, and with this substantial growth and growing influence of bioinformatics in health and environment and the society, how we can utilize this, how we can integrate this into our education, how well we can integrate this, what are the challenges, and where are we with all of this in India, and specifically what are the new opportunities that we see in India in terms of the new educational policy, and then the challenges and the benefits of bioinformatics being a subject, or the research behind the bioinformatics could be something that could be done online. And like as Dr. Anshu said, that many of her projects are between labs and it's collaborative in nature. So how different it is from traditional research versus bioinformatics and the collaborative nature of all of this. And then some of the promising areas, because we have a huge community of students who are biologists, who are coming from a background of biotechnology and other life science backgrounds, where they would help with the promising areas that an expert is seeing in India and Asia, where there are opportunities, which would bring us to the last point, would be the job opportunities in India and academia, and like for students from different backgrounds who are completing masters, some of them starting at a high school level versus someone who is doing masters and PhD in bioinformatics and starting from there. So these are some of the questions that our team thought would be helpful for the students based on their feedback, as well as based on what we have been observing as a bioinformatics training and educational company, working with some of these students who are joining us and working with the expert and developing some of the projects while learning a lot of skill sets. So with that introduction, I would want to invite all the panelists to have to be a part of this academic panel discussion to help find answers, or to have that discussion to give their opinion. And with that, we would want to start with this question of is there a need for data science education for life science students? Now, to define this question, because bioinformatics, as we know and as from our experience, has been evolving really rapidly. And that's what we have observed in the presentations as well, that the integration of new research, there are thousands of articles getting published, there are new algorithms which are coming up, and there are new data types that we are now now looking at. And during Dr. Anshu's presentation, we saw that there are new updates which are really important now to cover. So how can we do this effectively? And is there a need for education and revision of those curriculums? So maybe we can start with Dr. Sagar, who's joining, and he's a seasoned educator. So welcome, sir. And what are your thoughts on bioinformatics and data science education in India, and especially your experience in Amity University, Mumbai?
Thank you, Sonalika, for a nice introduction. Is I'm audible properly?
Yes, yes.
So as we see that the data science requirement in the life sciences, actually it is a must. If we see that in the biotechnology and other courses are there, now the experimental things, that is equipment things, are tremendously developed. That is, if you know, that is earlier 1 decade before, Where people are doing everything in the labs with the help of test tubes, they're doing assays and so on. At that time, their limited data is there, limited outcome is there. If suppose someone doing an experiment on a protein, they just going to purify the protein and then they just doing an assay. Along with that, if someone doing a work of genomics, in that case also they just going to amplify and deal with only single gene molecules. But now there are the tremendous advancement is there in these techniques. For example, protein purification is there or genome sequences is there. So there is a large amount of the data is generated. If you see that if single run of ion torrent machine, they just give you 300 GB of the data. And then it is very much important to analyze this data, how you just process this data and analyze this data. Now, the efforts of experimental biologists is reduced in some extent, but they need to learn this data science, advanced data science, because the data which is generated by these techniques, that is NGS is there or mass spec is there. So what data output is there? That is huge data is there, and you need to process the data and you have to come up with some useful information out of it. For example, if you run the mass spec, then also you will get the large amount of the masses. You have to analyze which are the important molecules or which are the protein expressed by a particular cell. Mostly we talking about the cancerous cell and healthy cell. So suppose you perform the mass spec analysis, then to identify these masses and proteins, you need to learn this advanced data science. Then and then only you just able to handle. So for that you need to learn the Perl requirement is there, Python requirement is there, R requirement is there. So these languages will helpful to you to process the data and associated tool which is able to analyze what components are there, and then you just come up with a particular protein or particular gene which is expressed or expressed in that diseased cell and which is not there in the healthy cell. So if you see, there is a perception in India, that is the bioinformatics. People are looking at bioinformatics, that is they just only give some artistic images and for publication only. Means people are only in that area, that is only docking simulation, and we just creating a picture for publication itself. Means if you see the approaches of the experimental biologists, they just want only you just to do the docking and give it to us, then we will add it and we will publish the data. But these things are now changed. That is all companies have developed a lot of pipelines are there, and even the normal biologists able to do this docking and simulation stuff. Now the need of the data science, that is people need or student need to learn these advanced languages as well as advanced tools to process and analyze big data generated by these genomics and proteomics machines which currently people are using.
Thank you. Thank you so much for your insights on that. So to summarize it, I think you're saying that, and also like that could be seen is that the wet lab technology has evolved and the data generation is now easy now compared to before when we were focused more on single experiments generating. Now we are discussing and we are generating high throughput data, which is simpler to generate than before and also less cost effective, I think. And the requirement is now more for the analysis part, because the data generation capabilities have increased, the technology has evolved. So that's how data science is now becoming a very important tool to handle this data, to get meaningful insights from it. That requires coding, some of the coding challenges, then someone has to be maybe learn more about how to apply maybe than a hardcore coding. But to be able to apply that, basic coding is of course is needed, and some bioinformatics related coding information would be needed for the students to dive into this field, which is getting now maybe the way to access this is becoming easier with the companies bringing this pipelines, and also labs such as Dr. Anshu's are generating those resources to be utilized by the students.
Yes.
Okay, that's something very insightful. I think maybe now we learned about that why data science is important from a bioinformatics perspective, and how also bioinformatics has been seen before by the by biologists, where they can see very few applications. And as the data has grown, I guess, so tremendously, now we are getting all those insights, and that awareness is somehow coming into picture. So the next question that I would want to ask Dr. Anshu is that how do you tackle these challenges or integration of this information into students' curriculum and the courses? Like, are the university adopting to these new technologies and approaches and integrating them, or there are other opportunities that the students are getting to learn about this through some maybe internships or something like that? So is there a structure, or is there a way that the universities are kind of incorporating this into their course structure?
Right. So Mohit, in the new education policy, as you know, the way the course structure is to be for undergrads is that after first year, if they wish to move fields, they should be able to do that. So recently, I have designed a course for the new education policy for undergrad in data analysis for Madhya Pradesh. So I'm on the board of studies to basically design that course. So the problem is not designing the course. What I have also told them parallelly is that who is going to teach it? We don't have enough people here in the country who can really take up these courses with the students. So course design is not the challenge here. We can write anything on paper, but the ultimate challenge is to put that to practice. Now, national laboratories are not equipped to handle the size of students that we currently generate in university systems. We cannot do that, despite the fact we try our best. We can handle probably a few thousands, but we are not talking about numbers in thousands here. So, and we need that kind of a workforce to really understand it. So then everything boils down to the fact that how motivated these students are to learn these skills. So I think the job that the universities can do here, even if they cannot teach the students the hard skills, if they can convey the message that these skills are critical, it will allow these kids to actually start taking up assignments in that domain. Because I think data analysis skills, if one has to learn, it has to be the best learning happens when you do it partly as structured teaching, but majorly as project-based learning. Reason is it cannot be structured to a very large extent because, as you rightly said, it is an evolving field. And it is evolving so fast that by the time you bring a structure into practice, it will change. So we need to give them the basic fundamental understanding of what the field is, rather than start teaching them only very specific methods. They should be able to adapt to new methods. And that is where training on what is fair data, what are ontologies, what is basically, when you say reference human genome, what is reference? And I think all of this can be taught by life science faculty in universities. You don't need any kind of data analysis skills to do that. So I think the way to approach this problem is to have to train teachers to convey this message across to the students. And then you will actually see the real impact of the new education policy also. If we do it the more traditional way as we have been doing, we'll not be able to scale it up. And that is, I think, a more logical, practical way of approaching this problem.
Yeah, that's a great point, actually, which kind of simplifies this for many faculty who might be struggling with.
Can I add something to what
Yes. I
Dr.
was coming to
Anshu
you.
just
Please,
said.
please go ahead.
So as she rightly said that, it's obviously the manpower which is missing, but at the same time, I think at the school level, when we are having 11th and 12th, what I have observed is, the boards have actually given them choices of taking either biology or mathematics. So many a times, the students are not taught mathematics. And when we are talking about data science, you cannot do without mathematics. That's a very big, I think, negative that I can see. And for that, it's very difficult to teach them. So the people who are actually coming from with the biology background, they are actually because they are scared of mathematics, and without mathematics it's very difficult to get people and train them as well.
Right. So Shuchi, I was under the impression that people are allowed to take both, because when I did my 11th, 12th, there was no binding on not taking maths and biology together.
Yeah, I agree that even I was of the same impression, but then this is my personal experience, I would say. So one of my very close relative, actually, she told me, I was like forcing her that don't do that. But then she said that no, the school is straight away saying that either you go for biology or go for mathematics. So when the students used to come, so I used to take up first year students, when they used to say, I never believed it. But having a experience of this kind, I saw that no, that's not the fact. So I think you cannot just segregate biology with mathematics when we are talking about data science and something which is really the need of the hour, we can say.
I just want to add here that maybe there is a silver lining, which is statistics. So statistics, I think by utilizing statistics, and we do have those in the curriculum in most of the biotechnology and all those allied fields. And we think that maybe data science is an advanced statistics that we are utilizing. And maybe if it is only application based, not like coding based, because when many of the bioinformatics challenges are to develop maybe some of these pipelines, workflows, but many of the biologists are just trying to find ways to analyze it and get to the biological part of it, more deep dive into the pathways and look at the mechanism and understand all those things. Maybe they can utilize the data science as a tool rather than data science, go in depth of data science. What do you think?
Yeah, I think what you are referring to, Mohit, is something which in gaming terms. See, I'm also a gamer, so I always go back to the gaming concept. So it is called as onboarding. So the learning curve cannot be very steep. So you need to first onboard, and then there is a concept of scaffolding where you really get people involved in the process. So I think before the anybody, not just school children, anybody coming into data sciences, there has to be an onboarding step, which has to be a small, learning curve. And then you start going on from there. And that is why I think project-based learning is important, because you have a question in mind. There will be some basic interest, because normally what I see in a lot of universities, because I actually go and teach at a lot of places, is that the programming is taught by computer science faculty. There is no context of biology. Students fail to make the connection between why, where do I use what? So I think that is where you need interdisciplinary faculty who can bring that context. And then if that is not possible, do a project-based learning. At least the question is clear in their mind. So they at least understand what they're doing. I completely agree with you.
I think this makes sense in the context of the presentation also, that Dr. Anshu's presentation covered very basic understanding of where these technologies can be applied. So maybe a keyword based, like you're saying that people need to be more, like students need to become more and more comfortable with this basic part of these, like reference genome, you said ontologies, you said fair data. So these are very common terminologies that could be a part of the basic training. And after the basic training, when they are comfortable, it would be more about project-based training, So that would be a proper pipeline. Maybe after the basic understanding, you do a project-based learning and choose your own specialization, choose your own maybe application of bioinformatics, that in metagenomics, I'll do this, in transcriptomics, I'll do this, and in structural bioinformatics, I can do this. So that clarity, if is there, and you also said that if that is something that a life science faculty can easily teach, can easily help the student community understand some of those facts to get them more interested and more exploring by themselves and get that enthusiasm, which is right now missing, maybe due to not being aware, and also some fear of mathematics and algorithms and all of that.
See, I think, Mohit, I'll just take 10 more seconds, is to also understand that till the time the research faculty get convinced that data sciences are important, the message will not trickle down. And I think the AlphaFold has been able to make that mental impact, where experimental biologists are able to appreciate why data sciences are so critical. I think before that, there was partial acceptance. With AlphaFold, that acceptance has gone multiple folds. So I think this is a right time for us. It's like when the iron is hot kind of a situation. So right now is the time when you can really make them. That message has to trickle down. And I think that message, if only comes from data scientists, people will not accept it. It has to be the entire community speaking the same language.
So you're saying we were looking for more validation, maybe a few AI-based drugs, and then we will be more accepting it. Yeah, which is, I think,
more acceptance for the in silico part of the work, rather than just allowing that wet lab, believing the wet lab result more. Yeah, very true.
All right, I think that concludes. This is a great point, and I think we can now maybe move to the next point, which brings us to the challenge of how, like, you think that integration of this hybrid training, which is like many university, they believe, and many institutions and many students, they think that maybe going to the university and working is the right way of learning. But during the COVID, many universities face this challenge of training students online, and then they were able to do this successfully. But do you see an integration of both of them? Because I saw that latest announcement by our Prime Minister, and he was emphasizing on that there should be an hybrid model. So what are your thoughts about this hybrid model, and is it possible? Because in bioinformatics and data science, yes, that is possible, but in an educational system, is it something that is going to happen?
So I make some points. That is when in the pandemic situation, so we turn from offline to online suddenly. And it is difficult for first one semester. We have found a lot of difficulties that students not able to understand anything when they join, and a lot of issue related to the biotechnology subject. Those are molecular biology or microbiology and so on. The students theoretically they understand everything, but the issue with the practical courses, not able to understand anything. And also in virtual world, you need your proper attention when the lecture or anything is. If you just sitting in front of computers for time being, and if you not listening in properly, you not understand, and you need to revise and you read it again. So that issues are there in the online system, but we are escaped from that for computational biology, because we successfully conducted all the practicals, all the theory lecture, but also that is the students, when in your presence, when they are learning, then it is some different impact is there. If everything is online, it is a little bit difficult, student not understand. And we also evaluated the student after examination. So the score of means passing percentage of the student in lockdown period is of university around 95 to 98%. And then after now physically they are attending, and in this semester it is suddenly dropped till 70%. So huge difference, 30% difference is there. Why these people's means many peoples are qualified this? Because MCQs are there, and open book exams are conducted by the colleges and universities, and that's why there are the what we can say misuse of this done by. And even though the knowledge level of the student is not that much, means if you just when as a teacher you just recall the previous thing, because the syllabus designed in such a way that you need to remember the earlier semester thing, then you just able to go for the next subject. But when you just try to recall the back subject, they're not able to understand what actually they learn. So it is very difficult to teacher to teach each and every subject in online. Only the thing is that if they are doing the specific courses on their own, means the university system is different. But if the students are doing a courses in online mode, paying the money for the course, then they have tremendous attention. But that attention is not there when they're learning in the university system. And that's why a little bit problem. But finally, the system is online or offline, doesn't matter. That is, that student or that participant should have attention towards the lecture or practice. And in national education policy, I think they have implemented a lot of things, that is in-out, multiple in-out they have made. So it would be better, I think, that a student can come in anytime, went out anytime, and as per his or her choice, he can opt the subject and so on. Because if you know the 12th passed student, they actually don't know what subject is this, actually what I have to do, even though it is imposed by the parent on them. That is, you have to learn this and that. This subject is good and this subject is bad. But student come to know after 2-3 years, when he is in last year of graduation, he come to know which subject is good one and what he like, and according to that they able to change. And that's why this in-out system in NAP is good for the student. At the last stage they can switch and they just go into the interested subject.
All right. Thank you so much. I mean, that kind of is in between. And I think COVID-19 has helped us maybe through these challenges to us. And now we have this experience of working online and saw those challenges. Now there are some good things that we can take from there, and that has been taken up already. And there is an integration and there is more flexibility and customization that could be more personalized. And that personalized training is maybe answer is one of some of the answers required for maybe better training and better outcome for every student, because we also get like good students, but there are some students who are not been able to do that in such focused way which they should have. So I think great point, and that's something that is a great thing for moving forward. That brings us to our last question. Yeah,
Okay,
please.
may I add one thing here? So see the hybrid mode that the Prime Minister is talking about. So there are two ways of doing that. First is we can actually have DIY kits. And this is not something which is hypothetical. We are already making a lot of DIY components and sending to the students, and they can actually try it out at their home. So they can actually learn to do experiments hands-on. Second is we are also generating a lot of, this virtual reality content, where you actually have that haptic sense of controlling things and doing the experiment sitting at home. So obviously the hybrid method will actually involve both. But yeah, but this is something where the content is increasing day by day, and DIY kits are also increasing. I would strongly recommend people not to do this, but if you really need to generate a mutant fly or to do CRISPR, believe me, every single component of that is available on Amazon. So you don't need any lab to generate a mutant fly as of today. You can do that. This is a very heavily unregulated domain that requires a lot of regulation, but to tell you that if you really want to do experiments, possible. So we need to put that to good use now.
Yeah, that's great to know that DIY and VR is making to the education where this has a maybe huge future and impact, and also maybe business opportunities for startups and students who are graduating now, and they would be the ones who can maybe do this more in their field. So yeah, great
I want to
thoughts.
add up one more
Yes.
point to this hybrid mode that all of us have gone through. So it's not only just the students, like it is saying it from the point of students, even with regards to teachers also, it becomes very difficult to convey your message because it's not, we cannot see the expression of the students. And, so that also makes, it demoralizes us also to some extent because we don't know whether what we are saying, are they able to grasp it or not? And whether we are just talking to walls. So that again makes it difficult. And with regards to when we are talking about bioinformatics or any computational experiments like work that we are doing, we are also unable to see what they are doing is correct or not. It might be they have got some result, but is it the right one, or are they going by the right way, is what which is very difficult to monitor also. So it's both sided, and once the person who is delivering also becomes monotonous, then obviously whatever interest is there, that also goes away. So hybrid does restrict even us to get to some kind of innovative measures to help them enjoy learning.
Yeah, so you're saying that, so I think many institutions made the video compulsory so that they can see the students, but it's not possible when you're looking at a bigger class. It's just does not make
They will
it
say we have connection issues, that's the
similar.
common
Yeah,
thing that we
similar
actually come
problem.
across.
And another challenge that you have mentioned is also to track progress, like how to properly track progress of the students who are doing these things online where you don't see them actually completing, or maybe how they're completing it, and if they're learning all of that or not. And then all of this is kind of like maybe at one time you see the expression, and as a teacher you understand that this is what the student has got, but that is missing, and which is a demotivating factor. But yeah, I mean, pros and cons, some of the things that we have learned is that by using technology, this experience can be improved far better, and that's where we are heading. So the use of technology and then use of mentors and the requirement of mentors in this field is something I also like to point out. That is also a requirement, and those students who are now getting educated and trained could be those resources. So there is this need of capacity, student capacity based that is required for the industry as well as for the academia. So those with those points, I think we can maybe move to the last point. We are running out of time, but about the promising areas that please, I think if all of you can just tell two of the promising areas where this biotechnology and bioinformatics and computational biology is heading. So precision medicine is one such area that Dr. Anshu has already recommended. And I would like to ask Dr. Suchi and Dr. Sagar also to recommend students that maybe these are the promising areas where the industry will be requiring students, the academia will be requiring students for research.
Yeah. So like promising area, obviously one is precision medicine that we're talking about. Personalized medicine is something which is really a promising area that is there by using bioinformatics and the omics technologies. That's because by studying a particular person's complete DNA, I think we can actually find out the things that are specific to that person. and that would actually help us to be giving a personalized medicine. In fact, using the data that can be generated, say simply like we are talking about taking all the data from blood sample and urine sample, we can actually analyze and generate models where we can see what is one thing that is increasing in a particular group of people and not in the other group. So comparing those kind of information that is available. Now, if you see it practically, when there are loads of people who are getting their blood test done every day, urine test done every day. But that data is really very important from the research point of view, which is actually going waste at present. So that's one area where we can actually think of exploring, and that would actually help us to generate quite a few models. So there are certain parameters when if it fits the parameter, okay, this model can be used, and that would help the doctors to prescribe medicines and also help them to guide them as to the right lifestyle that they could lead for helping them in a good way, so they can lead a good life accordingly. So that can be obviously with regards to various diseases that
All right. I think electronic health records and analysis of that data you're saying is very important and could play an important role.
So from my point of view, there are a lot of opportunity actually in the market. People are searching for data scientists. If you see that machine learning is there, artificial intelligence is there, NGS data analysis is there. So, or R is there, those people are able to handle the Python is there. So those people who have expertise, if you just put a single search on job.ac.uk, you will find there are the thousands of positions are there that people are waiting for skilled manpower who have expertise in this particular area. That people's biologists are there. The issue is that biologists are there, hardcore biologists, they don't know the computational thing. And computational biologists, computational people are there. They are good in the computation, but they don't know biology. So bioinformaticians or data scientists are the connecting link. And obvious, these people know both, and they have lot of opportunity. Right? That is, people's are one example I will give you. That is in one hospital, they just collecting the data, and their requirement is of the person who is able to process the data, medical data, and present in the graphical representation. And they're searching for the person who have experience in R package, okay, for process the data and represent. And they say that any amount means package they will offer. So that is the opportunity which is already present outside, but people need to learn the Python or machine learning or artificial intelligence and NGS data analysis. A lot of opportunities are there. And now the university systems are also implementing this, but there is also limitation. For MSc biotechnology, how much subject you incorporate of data science, rather than means you need to focus more on biotechnology, and that some additional subject you can add. But those are interested, they can learn these additional courses from, I hope you are offering the courses in data science and so on. So a student can join and they can learn the data science or specific component from other institute as well. So this is all about the opportunity which are there. And I hope that if the student develop the interest in the computational biology, it is very flourishing and now the advanced field. But the thing is that you need to update yourself every time, because the thing which are there in last year, it is tremendously changing next year. So you need to keep you updated, and then you will survive in this area. So thank you.
Thank you. Thank you, Dr. Sagar, for those insights and important skill sets for students to take home, take notes of data science, machine learning, and R and Python. So maybe if Dr. Anshu, you can conclude shortly. We are just out of time for the next student poster presentation.
So I think what Shuchi and Sagar has said as very, very critical components. So what they actually, students got a lot of keywords. They may add network medicine to it because it's like, combining all of that together. So if you have the right keywords on Google, you will be able to fetch the right information. I think searching is also a very important skill set. Don't underestimate that. If you don't have the right keywords, you may not be able to land on the right resources. So I think all the faculty gave you a very good set of keywords today. Go for that, and a request to Dr. Sagar, if you can list sites where there is a consolidated list of what the current requirements for industry and academia are. I think students can take a look at that and take cues from there to start practicing.
Yes, sir.
All right. Thank you so much, all the panelists, for being a part of this Omics Research Symposium. And we are really, really happy to have you all and learn so much from these sessions. And I hope that the same goes for all the participants who have been part of this discussion. And we have also this being shared live on YouTube, where we have a lot of users watching this. And this will be also shared for those who have missed this session. So thank you again for being a part of this and helping us all learn about these different aspects of bioinformatics in education and both in research. We will now have the poster presentation session, and we'll be happy to have you around. But again, thank you so much.
Thank you for your invitation for this panel discussion. And I think you need to extend a little bit more time, because to discuss on a lot of many points, we need at least 20 minutes to discuss on single. National education policy is different one.
Yes,
So
yes,
all the
I
points.
think.
So, okay,
Yes.
thank you. That is it is well managed and well maintained.
We'll connect
thank
again.
You for
That's definitely on the agenda. Thank you so much.
Yes,
Thank you. Thank you so much for the invitation. It was really nice having this kind of discussion.
Thank you.
Share a lot of points that could be helpful to all of us, and as well as the students. Thank you.
Perfect. Thank you.
Thanks, Mohit,
now it's time for the
creating this network. So thanks for that. I otherwise would never have met probably Dr. Sagar and Dr. Shuchi. So thanks for doing that.
It's great to connect, and I think we can connect and help each other in some of those aspects and collaborate maybe. Thank you so much.
One of the advantages of hybrid mode, where we are sitting at different places and we can still connect.
Yes, and that's the advantage all the students can also have, connecting with the mentors, and I think it's a great platform together we can provide to the students in India. So thank you again. And we have some students waiting to show their presentations, and it would be an exciting part of this research fellowship, research symposium. So now I'd like to invite Ms. Srigori Krishnan, Krishna Kumar. She is going to help the students go through this entire experience of showing their posters that could be evaluated. Over to you, Srigori.
All right. Thank you so much, Dr. Mohit. So far, we had an exciting sessions that talked about the various active areas of research that are happening in the field of bioinformatics and data science in the academic industry in India. And we also discussed some of the what are the various opportunities that students can access and where to access these opportunities and how to access them. Keeping these in mind, there still remains one of the challenge that students face, that is, they don't get the opportunity to showcase their project after completion. So that is why the Omics Research Symposium encourages students worldwide to present their research project and network with peers and experts in the field. So the topics of the symposium is Omics and Bioinformatics, AI, Machine Learning and Data Science, Astrobiology and Space Omics, Computational Biology in Biotechnology and Pharmaceutical Industry. The submission was open to all registered participants from high school, undergraduate, graduate, as well as citizen science projects. And out of the 25 plus abstracts that we have received, we have selected 12 of the finest projects for further rounds of review by the judges. Now, let me take a brief moment to introduce you to the judges who will be reviewing the student projects. First, we have Dr. Ramesh Kumar. He is the assistant professor at Reva University. We are also joined by Dr. Harpreet Kaur. She is the postdoctoral researcher at NCI NIH. We are also joined by Dr. Raghavendran Lakshmi Narayan. He is the research consultant at Pmi Biotech. And Dr. Abhishek Sengupta is the assistant professor at Amity University. And finally, and last but not the least, Dr. Julia Panov. She is the associate director at Tauber Bioinformatics Research Center, Israel. The presentation will be judged in 4 categories, that is introduction, methods and results, discussion and conclusion. Finally, if the students are able to showcase a quality-driven project within the time allowed, the best posters will be selected on the following different categories, that is best technical research project, promising research direction for future potential, and most innovative approach in an established domain. And the guidelines for the competition are simple. The participants will be given 5 minutes to present their poster. Once the time is up, you'll have to stop your presentation and move to the next candidate. So kindly make sure that you complete your presentation within 5 minutes. With that, let me invite the first participant who will be presenting their project. That is Dr. Parak Sehgal. She is the professor at Rangta College of Science and Technology, Chhattisgarh, India. And she and her team will be presenting her project on characterization of the hypothetical protein sequence of Naegleria fowleri using bioinformatics approach. So over to you, Dr.
Parak. Yeah, ma'am, am I audible?
Yes, you're audible.
Ma'am, I'll be a moment, ma'am.
All right, sure. Could you please share your screen?
Just give me a second. Ma'am, is my screen visible?
Yes, it is visible.
Okay, thank you so much. Just
your time starts
now. Okay, ma'am. Good evening to all the judges. Myself, Dr. Parak Sehgal, and I'm representing Rungta College of Science and Technology, Bhilai. And my colleague is Pratigya Das, Research Associate at the Biotech. We have chosen a topic to work on characterization of a hypothetical protein sequence of Naegleria fowleri using a bioinformatics approach. Now, this particular amoeba is causing a disease which is called as PAM. Okay, so PAM is primary amoebic meningoencephalitis. This disease is basically caused when somebody goes for swimming in the open kind of water environment. So this particular amoeba is inhaled via nostrils, and this can cause a lot of issues and can be very infectious and can be fatal for one person's life. So particularly when it is taken inside, now there are 3 lives in the life cycle of Naegleria fowleri. There are 3 phases. So one is cyst phase, another is trophozoite phase, and there's a third one. So in these 3 phases, the trophozoites are the most severe one, which causes the infection because they can multiply themselves with the binary fusion method, and they can cause that much of infection that a person can die in 7 days. Okay, so while looking to this particular slide, we can see if cerebrospinal fluid, the diagnosis is cerebrospinal from the cerebrospinal fluid, that gray to yellow color is changed, and when 1 ml of blood sample is taken and it is being checked, so it is found that the red blood cells number is very high. So person can be suffering from this particular PAM disease. There are various type of treatments known for this. So this is a life cycle I've already told about cyst, trophozoite, and flagellate phase. So we can see here the division of the cyst to trophozoite phase, which is the infectious one. Okay, so in this particular project, I have tried to find some details about the hypothetical protein which is not available in the database. So I have tried to find all the information about it using various bioinformatics tools. And there is something new which I have discovered in the project while doing the calculations with the bioinformatics tools about Nagella floweri, which causes this disease. So we can see the list of the bioinformatics tools I have used. I will tell you what new thing which I have discovered when I was doing the project. So this is the approach I have used. This is the hypothetical protein, and this is the ID number of N floweri, that particular protein which about which the information is very less available in the databases. Its structure is not there, its detail is not there, its protein-protein interaction was not available. So by the approach, I was able to figure out about this. So this N floweri, this particular ID was, I have got this from the UniProt database, and I have tried to perform the BLASTP, by which I was able to figure out the list of 10 sequences similar to this particular ID, and that was easily available. And there were around 82 of the sequences, but I took the first 10 to understand what could be the maximum similarity with its co-similar kind of species from the same family. So the first 10 sequences I have taken N floweri 5, and the next thing what I was to tell you was that there is another species called as N gruberry. Okay, so Nagella gruberry. I have taken 5 sequences of that and 5 of floweri just for the supportive study. So after I generated the multiple sequence alignment of these 10 sequences N floweri and N gruberry, I was able to see the conservation of the amino acid pattern in the sequences that supported the data that they belong to the same family. And then I have tried to discover the kind of family they belong to, the same family. And there was some patterns, common patterns were seen in the ProSite database. And something which was different was in the string database. When I wanted to see about the protein interaction, I was not able to figure out anything for N floweri. But when I took the next 5 sequences of N gruberry, I was able to figure out some similarity in the co-similar sequences of N gruberry. So this was something new which is not yet reported. So I figured out this. And after the structural characterization, when mod base I have used, there was no structure which was modeled for N floweri. And I was able to make it with the help of Swiss automated modeler. The results I'll be showing soon. And more information about this particular sequence I have figured out in Amoeba database, which shows that this particular hypothetical protein is very important in some kind of KEGG and metasec pathways, which I'll be showing later. So this is very much important in doing some metabolic activities. So we are going to the results section from the UniProt. This is the ID I have retrieved for Nagella floweri. And this was all about the project, where this was the main idea to figure out the details of it. These are few sequences which I have taken for N floweri and gruberry together just for the supportive study. This is the multiple sequence alignment. So I can show you the conservation over here. The sequences are very highly conserved amongst these 10 sequences. And I was able to figure out from the PFAM and the CDD database that they are all belonging to have the HSP70 domain. So this confirms that it is also having HSP70 domain like rest of the sequences. Then from this particular analysis of CDD database, it confirms that they are belonging to the same clan, 36495. They're having the same accession number and the same superfamily, PTZ 4x09. Then from the Interpro databases, these are few of the patterns from the domains I was able to figure out. IPR numbers are given. So I was able to figure out that they are belonging to the same family and clans. Then I was able to figure out some patterns which were similar. So I can show you a heat shock protein, heat shock HSP70 patterns are present. So they are having around 5 sequences. When N floweri was there, they were around 15 hits I have got from ProSite. This is the string result. So this tells that when I have used N floweri, there was no result. And just to support my study, I have tried that if N floweri is not there, N gruberry can be present for protein-protein interaction, which is just similar and belongs to the same family. So I was able to see that there are co-occurrences. There are some literature-based reports available. These are those ID numbers which I've shown you in the BLAST results. So they are having some or the other relation between them. Then using the CISP model, I was able to figure out that with this template number of 4ANI1, it was able to generate based upon sequence identity that the structure can be like this for the hypothetical protein, which is not available in the database. And very much important, it is based upon sequence identity. It means that this model has been generated based upon the place of the amino acid. So this is based upon the sequence identity. Then
sorry,
using
Dr. Parekh, could you please stop up?
Yes, ma'am. Yes, ma'am. Okay. So this is the last result where I can show you the amoeba database has shown that these are some of the important pathways, like it is involved in the purine metabolism and thymine metabolism. So overall conclusion of my project is that whatever information was available for N-Floweri was very less to interpret. In India, there are only around 15 cases reported. And with the help of N-Gruberry data, I was able to support my studies that some information can be said that Gruberry and Floweri are similar. They belong to the same family, and the results can be checked that way. Thank you so much.
All right. Thank you so much, Dr.
These are my
Parekh.
references. Thank you, ma'am.
The judges, do you have any feedback to give to Dr. Parekh?
I'll just show my poster as well.
Judges, do you have any feedback to give to Dr. Parekh about her project?
Yes, hello, hello, Dr. Parekh.
Yes,
And
ma'am.
it was nice work. So I have one actually suggestions maybe when you're choosing like 10 sequences. So
Yes, ma'am.
instead of choosing 10 sequences, it would be better if you use some criteria like maybe percentage of similarity. So maybe 90% similarity of the sequences. So maybe that would be the better criteria rather than just choosing the top 10 sequences. So because when we comparing some sequence, so we should consider its similarity or dissimilarity score. So my suggestion is fine for this work if you're just doing for the publication perspective. It would be much better like if you consider that. Yeah. Rest of thing is fine. Like it is interesting work. Yeah.
Okay, ma'am. Thank you so much for the suggestion. Ma'am, just I would like to say something because the idea behind this project was not to figure out the similarity initially. It was just about to figure out only the information about the hypothetical protein. So that criteria I may have missed as per you have said. So, but yes, for the publication point of view, for the next I will consider this. Thank you so much for the suggestion. Yes, ma'am.
All right. Thank you so much, Dr. Parekh. Could you please stop sharing the screen?
Yes, ma'am. Yes.
Thank you. All right. So let me invite the next participant, Mr. Kashish Kundu. He is a postgraduate student of Bioinformatics and Computational Biology from Amity University. And he'll be presenting his project on in silico analysis of transcriptional responses to UV and IR radiations. So over to you, Kashish.
Thank you so much, ma'am. Yes. So good evening, everyone. Myself, Kashish Kundu, and about my poster. In skin cancer, many genes are involved in known DNA damage response pathways for cell cycling and proliferation, and also in apoptosis, DNA repair or in other stress responses. However, majority of these genes are involved in unexpected pathways with functions in signal transduction, RNA binding and editing, protein synthesis and degradation, energy metabolism, and cell structure. I was interested in knowing these responses, hence I chose a particular dataset to work on. The analysis was on the dataset was done on Bioinfo server along with a few other tools and platforms. This part of poster represents the wait a second. Yeah. So this was the introduction which I gave about my poster. Yes. So this part of the poster represents a methodology that was used along with a flowchart illustrating the the pipeline that was followed. Firstly, the dataset was identified on NCBI using GEO dataset database. The database was downloaded and filtered, and grouping of the sample was done. Of the samples was done. PCA and other statistical analysis were performed manually as well as using GUI platforms. Later on, pathway analysis was done using Links tool and PantherDB. Links database, that is library of integrated network based cellular signatures, takes up input as gene IDs, fold change information, and an optional field for p-values. Protein analysis through evolutionary relationships, abbreviated as Panther, it links protein sequence evolution to the development of protein activities and biological functions. Separated files of genes are accepted as inputs, and as output, graphical representation of gene expression levels are plotted against the pathways they are expressed in. From here, further biological interpretation was made. From the top probe sets, the IR responsive genes like ATF3, MAP2, MEF2C were involved in stress response. Genes like PVT1, MMYC, NEK2 were involved in cell cycle and proliferation. There were many genes with involvement in signal transduction and apoptosis. For UV responsive genes, similar results were found with entries in stress responses, signal transduction, energy metabolism, and apoptotic pathways. From the results recorded, it can be interpreted that abrupt activities of cell cycle and energy metabolism may cause IR as well as UV-induced skin lesions. Apoptotic cycle showed an increase in the expressions, which can be inferred as immune activity to tackle the lesion development, or it can also indicate the breakdown of essential cellular components that were aided in prevention of further proliferation of abnormal cells. The outcome proposes that protein translation is additionally hindered by halted transcription of tRNA synthetase genes. Now, here you can see the PCA result, which was drawn out, and this shows the PantherDB result along with the bar charts of the genes that was expressed. Now, using PantherDB, these two tables shows us the threshold that I took, that is the 9 gene from this table, and I studied these pathways. I haven't covered all these pathways because instead of going into an extensive research, I was more focused on the highly expressed genes, and hence I went for these pathways.
Kashish, sorry to interrupt you. You have less than 20 seconds now.
Okay, yeah, I'm concluding. So the conclusion section gives us a brief overview of the interpretations of the analysis, and the bar chart represents the probe set information of induced and repressed genes of IR and UV. And for future perspective about this project, now I'm looking into the method comparison of this dataset that was that I chose along with the method that I used, and I am looking forward for its validation now. Thank you, ma'am.
All right, thank you so much. Does the judges have any feedback for Kashish? Dr. Raghav, Dr. Harpreet, do you have any feedback?
Hello, Kashish. So I have just a few things. So it would be better if you just also show like how many samples have you used. Maybe I missed that. So that it should be like statistically correct. Another thing, when you actually this is just for the I mean from the analysis perspective, I'm not going into the deeper science. So another thing in your principal PCA results and your pathway results, so it would be better if you maybe do it based on the sample group, because when you're drawing this PCA, it is difficult to understand like what is actually which group is there or which is not. I guess you just assigned it
No, I
colors
did.
or something. Yeah.
Ma'am, actually, before I have the result for both before grouping the PCA, actually I was looking if there is any similarity or any plot interpretation which can I take, which I may find without grouping the data. Once it was grouped into IR, UV, and separately for UV and IR, I have inferred that information also there.
So it would be much better when you're doing even in the first group before grouping. You can take actually, I mean, you can color your sample based on either this radiation type, because it's difficult to interpret anything out of this, like whether these groups are distinguished from each other or not. So that's something that you need to maybe must do this or reanalyze it. So because it is difficult to interpret at the moment. Yeah.
Okay.
Yeah.
All right,
yeah, that's all.
All right then. Kashish, could you please stop sharing the screen?
Yes, ma'am. Thank you, ma'am.
All right. So next, I would like to invite Mr. Emmanuel and team who will be presenting their project on visualization of the chromosome 1 genomic data from the African, American, Asian, and European populations. Over to you. Emmanuel, are you there? All right, then. So let's move on to the next participant, Ms. Anmika Ghosh. She will be presenting her project on transcriptomic analysis of hippocampal subfield CA1 in Alzheimer's disease. Over to you, Anmika.
Hello all. So I'm sharing my screen. Is it visible, everyone?
Yes, it is visible. Could you please zoom in? Or thank you so much.
Yeah, so I can easily zoom in here. Okay. So hello everyone. So today I'm going to present my project topic, transcriptomic analysis of hippocampal subfield CA1 in Alzheimer's disease. So what is Alzheimer's disease? Alzheimer's disease is a neurodegenerative disorder affecting mainly the elder population. Affecting mainly, while it could be observed in the younger population, but its prevalence is more in the older generation. There are several hypotheses that exist behind this Alzheimer's disease. However, some of not all of them are really accomplished or really found enough evidence. So here is a comparison between the healthy brain and a brain that is affected by severe Alzheimer's disease. And here is another diagram that shows hallmarks of Alzheimer's disease, which include accumulation of amyloid plaque proteins, neuroinflammation, mitochondrial dysfunction, autophagy dysfunction, and cholinergic insufficiency. And last but not least, a neurofibrillary tangles. So here is an overview of the methodology of what we have done. So for the sake of project, we have used this data set from gene expression omnibus. The pre-processing of the data was carried out with the help of Microsoft Excel 2019. After pre-processing of the data, we have quantum normalized our data and labeled it. Followed by this, we done principal component analysis, and we detected X119 as an outlier and removed it. Then we ran D6-2 and insert enrichment analysis. After determining statistically significant genes, we validated our results. So here is an overview of our results section. So as we can see here, this is the result of the principal component analysis. So the PC1, it accounts for 23.01%, PC2 accounts for 18.19%, and PC3 accounts for 14.75% variance. X119, as mentioned earlier, is detected as the earlier, is detected as the outlier, and we have removed it and proceeded further. So here is a volcano plot showing the upregulated and downregulated genes, and the right side shows the positive upregulation and upregulation, whereas the left side shows the negative upregulation. Here is the gene ontology analysis for our data set. So activated and suppressed pathways are separated by two separate boxes, and the gene count is reflected by the size of this circles. Gene ratio is mentioned at the bottom of the boxes, and here is the mention of the p adjusted values. And this is the CNET plot of our genes and gene ontologies. So both these are the genes that are, the fold change is negative, so they are basically down regulated, and there are some gene ontology terms, again they are also down regulated. Here is here is the result of our cage enriched pathways analysis. So here we can see again that activated and suppressed pathways are separated by two boxes. Gene ratio is mentioned here in the bottom, and here is the like we can see the p adjusted values. The red signifies the most significant ones, the statistically significant ones. Okay, so for the sake of the validation, what we have did is we took the gene list of the table supplementary table S5 of the main paper that is associated with our data set, and we compared it with our results, the output file that we got after this analysis. So we mainly went for the statistically significant values, statistically significant ones. So our results shows that there is 0%, there is no overlap between this two list of genes. So therefore we concluded that therefore we concluded that that it might be due to the fact that our project is very much focused on the hippocampal subfield CA1, whereas the main paper was taking into consideration other hippocampal subfields. So that is what I can say about the conclusion section, and here is my references. And last but not least is I would love to acknowledge my research mentors, Mr. Dr. Raghavendram Lakshminarayan sir and Elia Brodsky sir, for helping me out to carry out this research project. I would also love to thank all the members of the Pmi Biotech. Thank you.
All right, thank you so much. Does the judges have any feedback for Anamika?
Yeah, I can ask this time maybe. Thank you very much for the presentation, and just a few questions about your presentation. So you mentioned that there was an outlier in your data set, and that you excluded it. So did you try to find out and understand why it is an outlier? Did you go back to the data and try to understand why the sample is an outlier? Maybe some normalization was done differently, because the main paper, as I understand, did not mention any outliers, correct?
Yeah, the main paper. Actually, to be honest, I back searched about the sample. It's 119, but I could not find anything really significant to tell actually. But that's, I could not find anything.
Okay, yeah, just you know, just a remark that usually an outlier is something that you should probably go and really dig into and try to understand why it is an outlier. Maybe the problem is not with the sample, but with the analysis. So maybe, you know, analyzing and looking at the data a little bit differently would yield different results. So also you mentioned the downregulated genes and upregulated genes and the pathways and everything you know, those graphs that you talked about. What I'm kind of was a little missing is your interpretation. So are those pathways or genes mentioned in literature? Have they been found in Alzheimer's disease before? You know, some of your input on, you know, on the interpretation of the results. So that was a little bit missing for me. And also, yeah, the last graph where you have the Venn diagram and you show that you have nothing actually in common with the main paper. Maybe maybe I didn't, you know, I didn't have time to go into the data set itself, but you probably took only part of the data from this big data set, I'm guessing, right?
Actually,
So
we have considered the statistically significant genes only, that is p-value less than 0.05. So it was mentioned in the supplementary table S5 in the main paper, that is the last one, and here is actually my 181 gene. So I compared that.
Right. So you said that none of the genes actually were the same. So I was wondering, probably they had more samples and maybe did different differentiation, or was it the same? Because if you had nothing in common, that's a little, you know, concerning, I guess. that's just interesting.
Actually, I take it in another way. Like that paper, the original paper, what they did was related to all hippocampal sepals and also EC or entorhinal cortex. But when I did not find any like overlapping gene, I went back to the data, right, the GSC file, and what I found out that the data submitted in the GEO or Gene Expression Omnibus database, that is very specific to hippocampal sepal CA1 that I have mentioned. So I am very positive about my results actually, because I think my result is specific for CA1.
Okay, no problem. Just wanted to.
Can I interrupt? Actually, I was expecting Animika to tell this. And the comparison that she did was with the differentially expressed proteome from proteomics data analysis from different regions of Alzheimer's disease patients. So that's what the paper actually listed, and that's what she took to compare her differentially expressed genes list that she got from hippocampus. Yeah, that's why we did not, or she did not find a lot of common genes between the differentially expressed proteins analysis from proteomics data, which
I see. This is proteomics.
Yeah,
okay.
Yeah, averaged out data from several regions of Alzheimer's disease patient from one specific region, that is hippocampus. That is differentially expressed genes that she found out.
I see. Yeah, and I think this is pretty much known that there is little, actually, a lot of times there is little interaction between the proteomics and the gene expression. So yeah, this makes sense. Thank you.
Yes.
That's all. Thank you. Thank you for all the
Yes.
answers.
Thank you, ma'am, for valuable feedback. Thank you all.
All right, thank you so much. So now I would like to conclude today's poster presentation competition, unfortunately due to time constraint. So during the March 2022 Symposium, we brought together a panel of experts from Africa to discuss about the challenges, opportunities that bioinformatics education in Africa faces. So with those points in mind, now I'd like to invite for this symposium, Mr. Adekunle Adewoye, to start with August 2022 Symposium. Over to you. Mr. Ericule, could you start with the session?
Hi, please collaborate to meet up the visualization team. We are here to present also. I was unable to mute my mic the other time.
Yes, due to time constraints, we are having to wrap up. So your posters have already been sent to the judges and will be reviewed, and the results will be declared tomorrow. Thank you so much, Amanda.
All right, thank you.
Hello, good afternoon. Can you hear me?
Yes, I'm able to hear you. Could you please share your screen?
All right. Sorry about that. I needed to get permission to share my screen earlier on. Okay. Please, can you see my screen? Hello, can you see my screen?
Yes, but yes, now it is visible.
Okay, great, thanks. Good afternoon, everyone. I'm happy to be with us. And once again, I want to welcome all participants to this Omics Research Symposium with the thematic focus on bioinformatics and data science research and training. Beginning with a session by the Omicslogic Asia, I have no doubt that the entire symposium will be an enriching and stimulating scientific discourse over this 2 days period. So moving on, here within the Omicslogic Africa section, we will in the next an hour and a half be looking at the Omicslogic Africa programs and the scope of bioinformatics training within the African continent in the past 12 months. We will equally be taking 2 important keynote presentations by 2 notable scholars of repute. Firstly is Professor Akinde Babatunde, who will be doing justice to the topic Advancing Biomedical Research with Omics Data and Computational Biology in Africa, talking from insights from our researches. And then that will be followed by Dr. Mariana Dibiyi, who will be speaking on opportunities for bioinformatics and computational approaches in scientific research in Africa. And importantly, we will be taking on a panel session afterwards to discuss on bioinformatics, education, training, and research in Africa. Before then, let me quickly give an overview of the introduction on Omicslogic Africa programs, and and as well as partnerships and some of the outcomes we've achieved in the past 12 months. So moving on, allow me to introduce myself. My name is Adekunle Adeluoye. I'm a faculty with Lee City University here in Nigeria, and I'm an associate with the Medical Laboratory Science Council of Nigeria. I'm also a member and facilitator with Pmi Biotech Incorporated, and doubles as the program director for Omicslogic Africa and Eureka. I am therefore pleased to introduce you all to Omicslogic Africa programs during the next 10 minutes. Equally, permit me to quickly acknowledge here the Omicslogic Africa team working hard to advance the Omicslogic training across Africa. So the Omicslogic set of bioinformatics and big data analysis training programs of the Pmi Biotech Incorporated, which has been globally proven by peer reviews and curriculum experts in partner institutions, is being inventively explored in closing the needed gaps with regards to bioinformatics education and training globally. And of course, this is of great importance for us here in Africa in terms of building these scientific capacities among students, researchers, young scholars, young professionals, and faculties across the African continent. And so our postulation rather with Omicslogic Africa that is anchored by Eureka is that by increasingly advancing the knowledge of bioinformatics and its use in obtaining scientific insights from sequence data, there will be a significant growth and development of the bioinformatics community here in Africa and its considerable implications for translational science from understanding of gene functions, as well as its role in biology, agriculture, and medicine. I mean, just a variety of fields for addressing challenges that are peculiar to the population here within the continent. And so the goal of Omicslogic programs, therefore, is to provide this training in bioinformatics by enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research that is guided by mentors and peer examples. And so the Omicslogic programs boast of everything a participant needs to get started and grow in the bioinformatics and big data analysis space for scientific research and practice. Our programs combine online interactive sections and self-guided study materials and practical assignments for an immersive experience proven to have been very effective in our well-known and respected Omicslogic bioinformatics training programs. So while the growing interest in bioinformatics and big data analysis continues to grow here in Africa, many are still far from being able to gain this skill set for utilization in their scientific research and as well as practice. But we are glad that with the Omicslogic Africa training programs of Pmi Biotech Incorporated, these gaps are being bridged for students, faculties, and researchers across the African continent. And so I'd like to quickly share here a few of such programs which over the course of the past 12 months we have organized, to bring bioinformatics training with project-guided learning and resources to a cross-section of participants across the continent, ranging from beginners and students, many of whom are new to bioinformatics, to postgraduates, as well as researchers and faculties wanting to develop skills and research capacity to explore the world of omics sciences and using bioinformatics and big data analysis to answer research questions. So this high training, of course, includes data science for biologists, introduction to bioinformatics, as well as with bioinformatics and computational biology. And also we've gone into bioinformatics for biomedical drug discovery. These are some of the programs we've run lately. And equally, at least this program is the Omicslogic training for students and researchers, which is a basic bioinformatics introductory workshop that was recently concluded. And this program actually had over 200 participants who participated and gained the requisite skill to get introduced into the bioinformatics space. Equally, we recently conducted Omicslogic Genomics workshop, which we just concluded, and it's amazing the outcome we got from these programs, as we have currently a number of participants moving on currently into our research fellowship program. So this training essentially prepares these participants. And some of the training program participants who have gone from being beginners in bioinformatics to learning requisite skill sets that they can apply into their own personal research and practice includes Stephen Matthew. Some of the, of the things that these participants have learned and equally acquired a number of expertise in a cross section of domains, learning domains on our learning platform. Equally, we have Ujubi Sola, a pharmaceutical microbiologist who has seen her practice and application of key bioinformatics concepts in her practice significantly enhanced. And in addition to that, we have Giselle Flodre from Cameroon, who now sees herself well prepared to take on research and journey into the marine biology study space, leveraging on key skill sets from our Omicslogic bioinformatics trainings. So suffice to say that we have also forged notable partnerships with institutions towards integrating our hands-on, mentor-driven, globally proven training methods and resources into university educations across Africa, both at undergraduate as well as postgraduate levels, just to help scale rapidly the growth and capacity of bioinformatics researchers across the continent. And so a few of such notable partnerships include, partnerships with Lee City University in Nigeria. We also have a partnership with Crescent University, as well as Osun State University. And much recently, we had a working relationship with University of Yaoundé, that is in Cameroon. Similarly, Omicslogic Africa team has participated at notable regional scientific workshops, such as the Nigerian Bioinformatics and Genomics Network, where we had a poster presentation on the topic, experimenting with 21st century e-learning design for bridging bioinformatics education gaps in Africa, a test case with Omicslogic activity rubrics. And moving on, I'd like to quickly mention our next training program that is around the corner, which is another introductory bioinformatics workshop built to take place between the 7th and 13th of September. It's an introductory beginner level workshop, and the goal is to bring a number of participants into the omics, logic, as well as bioinformatics and big data science skills and knowledge space. And so this program will be happening soon. And shortly after that, we also intend to run another Omicslogic Metagenomics workshop. And I will encourage participants to please stay on the lookout for these programs. So moving on, in conclusion, the African continent has been regarded as the cradle of modern humans, and also the African genomes known to contain more genetic variations than those from any other continent across the globe. Thus, it presents a rich focus for scientific studies. Bioinformatics has become definitely an indispensable tool for management of this hugely generated data in modern biology, medicine, and related fields. However, infrastructural gaps and other challenges with educational system within the continent remains a cause for concern. Hence, the need for innovative approach to advancing bioinformatics education and training across Africa. And of course, Omicslogic programs of the Biotech no doubt occupies a strategic role in scaling the bioinformatics education and training needs for today, as well as in the future to come. And so we hope to continue to play a significant role in this space. So on that note, I would like to mention that a panel session will be coming up shortly where we discuss in detail about the bioinformatics education, training, and research in Africa. I want you all to please stay tuned and join that section. So thank you for listening. You can reach us at african@pine.bio in case you have any inquiries or follow-on questions. At this point, without taking time, if you have questions, please, I will not hesitate to attend to that. You can please drop them in the chat box. But at this point, we want to move on. And it is my hope our next speaker is available online already. Hello, Professor Akinde, are you there? Hello, Ayanfe, are you there?
Yeah, I'm around, I'm here.
Okay, please.
Professor Akinde is having a challenge presently. He's having a challenge connecting presently. He's writing me that he's been having issues connecting. Yeah, that was the last message I got.
All right. So then perhaps we move on to the next speaker. That will be Dr. Marion Adebiyi. Is Dr. Marion Adebiyi online? Hello, Dr. Marion. Hello, Dr. Marion.
I doubt if Dr. Marion is available.
Okay, perhaps we have to move on and take a panel discussion and then come back to these presentations. I remember Professor Akinde mentioned he was having challenges connecting earlier on. I told him to connect via the incognito mode. Perhaps he's having challenges with that still. And so without wasting time, I think we should move on to our panel discussion and while we engage to see that these speakers are on hand to take on their sessions afterwards. So over to you, Ayanfe.
Yeah, thank you so much. Thank you so much, participants. It's so lovely to have us all here. I can see Dr. Francis over here. Hello, Dr. Francis, are you there? And then I can see Risi. Hello, Risi, are you there? Can you say hello?
Yes, please. Can you let me allow them to?
Hello, yes.
Okay.
I'm here.
Okay. I can also hear you. Wow, that's so lovely. That's so lovely. Thank you so much for joining this section. I want to also confirm if our consultant doctor is also around, the person of Dr. Shonibare. Hello, Dr. Shonibare, are you here? Okay, in the absence of that, I believe that we will just start straight away with the panelist section, then go back. Okay, Dr. Shonibare is around. Hello, sir, you're welcome. Can you hear me, sir? Okay, you are actually We can't actually hear you. We can't actually hear you. Dr. Shonibare, can you say something again? We can see you, but we can't hear you. So I am asking you to unmute, Dr. Shonibare, or maybe reconnect. We can't hear you.
Okay. Hello.
Okay.
Hello.
Now
How
we
are
can
you?
hear you.
Can you hear me now?
Yes, we can hear you.
Okay.
Wow,
okay.
That's so lovely. That's so lovely. I want to welcome everyone to this section, and we will be having a lovely discussion this evening because we will be bringing the scopes at Africa, and we will be checking around our continent, seeing how and what is actually going on in Africa. So the topic for our panel discussion today is bioinformatics education in research in Africa. We have here on the panel Ms. Risi Wizonga. She is a scholar and she has actually had a trans-African or an intra-African education. It's so lovely to have you here with us, Ms. Risi Wizonga.
Thank you.
Yeah, then we also have Dr. Francis. Dr. Francis, can you say hello?
Yes, I can hear you, everyone. Good evening to everyone.
Yeah, so we have Dr. Francis Sue King. He's a faculty at the University of Bio in Cameroon. He's actually representing West Africa and is an associate researcher at the Biotechnology Center of the University of Yaoundé. Yeah, so lovely to have you here with us. Thank you so much.
Thank you, sir.
Okay, I will also be introducing Dr. Samuel Adedayo Shonibare. He's a consultant, a practicing physician, and a clinical microbiologist, especially, and a specialist doctor who works with the clinical microbiology department at the National Hospital in Abuja, Nigeria. Hello, doctor, can you hear me?
Yes, I can hear you clearly.
Yeah, thank you so much for joining us at this section.
Yeah, pleasure.
Yeah, then we also have, as part of the panelists, our program director, the person of Adekunle Adelowoye. He is our program director at Omicslogic Africa and Pmi Biotech Incorporated USA. He's also a faculty at the Medical Laboratory Science Department in Leeds City University, Nigeria. He's also a CEO and founder of Digital Health Tech Project, Pre-Diane and Screened here in Nigeria. I'm so honored to actually introduce you, Mr. Adekunle Adelowoye. You're welcome.
Thank you very much. It's a pleasure being with you.
All right. So some people are wondering, who is the person speaking right now? So I am Ayonfeoluwa. I am the Community Engagement Manager for Omicslogic Africa Engagement as a whole. And I am a practicing molecular biologist and geneticist. I am a postgraduate of bioinformatics in the Institute of Cytology and Genetics in Novosibirsk, Russia. So lovely to actually have us here. So going straight to the talk, I would love to start by saying that despite the poor capacity development for genomics research in Africa and the effort to empower African scientists to get involved in genomic research, particularly those research that involve African populations, we had consortium coming up, the likes of Human Hereditary and Health in Africa, the H3 Africa Consortium. Then we had other initiative that actually came up, such that we could have databases and co. I would love to throw out some questions in a bit. Here in Africa, before the year 2010, bioinformatics and genomics training was largely done by small groups at very few institutions, and they heavily relied on visiting experts in Europe and America, and some of our African experts had to go to Europe and America to get this experience firsthand. However, we have actually seen how the effort to change this has actually surged so far. We saw the African Society of Human Genomics rising up. We saw the Human Hereditary and Health Consortium in Africa. We saw them also coming up in 2011, gearing towards empowering the Africa that actually have the bioinformatics education, the African researchers that are properly trained, and the African researchers that could actually engage proper bioinformatics research in Africa. So fast forward to today, we also understand that individuals that do not have access to formal graduate trainings at these big universities like we used to know, we understand that there are actually some challenges that the present day researchers actually experience. So I would love to call on Dr. Francis. Dr. Francis, I would love to ask from you. So far so good. I remember that recently you had a training in bioinformatics in your country, and you tried as much as possible, and in your institution you tried as much as possible to work towards bioinformatics training. Can you just share with us the challenges that you actually met during this training, and what were actually the constraint? What was the success you were able to have?
So thank you, Mr. Alabetito. Thank you to all the panelists, and it's a very great pleasure to be amongst you this evening and to discuss about the challenges of bioinformatics in Africa. And thank you also for introducing that great workshop that we recently organized in January 2022 at the Biotechnology Center of the University of the Yaoundé 1. And talking about the challenges and even the success of that training, I will start by saying that talking about bioinformatics in Africa today is no more a challenging talk. It's no more a tricky aspect, because it's something that people are getting involved. And the interest has been also increased, as you just introduced from your initial speech. And regarding that special training, it was a little bit striking because it was a first count of such training to be organized in our institution. And we even went too far by training people on NGS, which is a very, very new aspect in the research domain in Cameroon. So definitely, having people of a very good background to get involved in such training was a very great challenge. You have people from different backgrounds, people who have never worked on molecular biology and who want to get involved into bioinformatics. You have biologists, you have engineers, you have. But definitely the first challenge was to get them, to bring them together, or to try to have people with the same background in order to assess that at least we move together during the training. And this was achieved because we mainly focus on those who at least has a good background in molecular biology, even if they are going to work on bioinformatics and they don't yet have bioinformatics background. At least we are certain that they can have a very good background in molecular biology, knowing that if they are acquainted with biological data, it will be easy to talk with them about those data and how to handle those data. And we finally got something very interesting from those that we selected, because the final number of participants selected for that workshop was 20. And I can say that yes, the workshop was not to bring them or to build them as experts of bioinformatics, but the workshop was just to get them acquainted with the introductory knowledge of bioinformatics. And I think the goal was set. But as soon as you run a such training, and as soon as you introduce people to a new discipline, you should ascertain that they continue building that skill to be able to have the final required skill that you plan before you are training. And that's why during that workshop, we bring in PanBio or Omics and Omicslogics to be sure that immediately after the training, those participants will have other platforms, other learning platforms, other windows where they could be able to continue getting much training about bioinformatics. So definitely it was a very good experience, and we really need such experience again. We need such training again in Africa and in Cameroon in particular to get other involved to introduce other into the discipline of bioinformatics.
Yeah, thank you so much, Dr. Francis. Yeah, that was so detailed because you were with the, I mean, you were from your first-hand experience looking, I mean, experiencing bioinformatics training, even as a trainer in Cameroon, was actually a great task for you. So
Exactly.
I really appreciate those inputs. I would love to call on someone in the field before I call on Risi, yeah, so that she can share our experience at Kenya, Babuan experience with us, how she got involved in bioinformatics and how she was able to cross over. But before I call on Risi, I would love to call on Dr. Shonibare. Dr. Shonibare, are you there?
Yes, I'm here.
Okay, so the quick question I would love to ask is still related with this. The question is, what do you think is actually the obstacle that is actually preventing the public health sector, for example, in West Africa, using Nigeria as an example? What do you think is actually hindering them from really engaging molecular research and molecular diagnostics that will actually bring into use the expertise of bioinformatics knowledge?
Yes, like you said, I'm a clinician. I'm a medical microbiologist, and I want to look at discussing with Omicslogic about health research and diagnostics and in treatment. So I just recently got introduced to Omicslogic about 3 to 6 months back, and I've been following. And like I will want you to know that in my country, Nigeria, we're actually into molecular research. A lot of centers now have molecular laboratories, but we are very, like you said, we are at the very baby phase of bioinformatics. A lot of centers have not keyed into bioinformatics. And my center is service oriented. Be that as it may, that it also enjoys research in diseases and diagnostics. We wouldn't mind, but a lot of hindrances, a lot of encumbrances are there in my country. Right now, you know that most of our training institutions, most of our education institutions are home. They have been on strike for the past 7 months. And it's only those institutions that are not public that are running. And then because of the challenges, which is global, the economic meltdown and everything, going into researches, especially molecular training, molecular research, is seriously challenging. Although it is not undaunting, it's not something that cannot be surmounted. And that I believe that, like the first speaker from Cameroon said, Africa is in the teething phase. Africa is learning. And I'm sure that Nigeria will also catch up quickly, because as I've been following the presentation this morning, I've been hearing people from Osun State, the Osun State University. I've been hearing other people who have logged on to the Omicslogic platform and are already doing well at catching up. I heard the first speaker talk about what they do, you understand? And then the training that they've been giving people, even spreading far back as Cameroon, spreading back to neighboring nations in Nigeria. So I know I can see a future for Nigeria with Omicslogic and bioinformatics. So that's what I have to say for now.
Okay, thank you so much. Thank you so much. We can actually draw a summary that that even if the government isn't actually helping to push this to work, people are still engaging the knowledge, which is actually a very, very good one. So I'm going to call on Risi at this point to actually share with us. So Risi, from a first-hand experience, I understand that you actually had some intra-African education. What is actually the gap between, I mean, in molecular studies, bioinformatics studies, between Kenya and Zimbabwe, and what has the experience been so far for you?
Okay, sir, thank you so much for the opportunity. So my introduction to the field of bioinformatics actually started in Kenya. My background is in biochemistry and molecular biology. So I had my internship at the Kenya Medical Research Institute. And at that point, my boss at the time had some projects that required a bit of genomics and bioinformatics knowledge. And by virtue of that, he organized a lot of knowledge transfer seminars whereby you would have people coming to teach us different things. So back then, I wasn't confident enough that I understood bioinformatics as such, because when you said bioinformatics, you'd have to be in places like ILRI where they offer bioinformatics based internship, or you would have to be in the ENBIT program because it's one of the major programs that offers bioinformatics training in Africa. And with these few centers, it means that you needed to know someone with knowledge in terms of bioinformatics, or who knows a sort of training, a sort of institute that is offering that kind of training for you to be able to be exposed to this kind of knowledge. Transitioning into my master's, I'm currently doing my master's in genomics and precision medicine at the African Institute of Biomedical Science and Technology in Zimbabwe. So ABS as an institute, they have a bioinformatics and genomics cohort, and they are also part of one of the nodes for the H3 BioNet program. So transitioning from Kenya to ABS, despite my program being genomics, you cannot afford to ignore bioinformatics, because I think where we are going in the life science field, knowledge in bioinformatics is very, very crucial. So part of my course here at ABS is in bioinformatics. I had to do a module in bioinformatics. So that was like in class sessions, and then in addition to that, we also had to take up some other online classes in FutureLearn, where they do like bioinformatics training for biologists, because you need to merge both into one. And then being a node for H3 BioNet, I am actually a beneficiary of most of the trainings at H3 BioNet, and it has been a continuous process, because the more you learn, the more you're curious, you want to know more, and that's how I ended up at the Omicslogic Research Symposium. I've been attending a lot of bioinformatics research symposium because I want to be familiar with the kind of research that is currently going on. And through this, the gap has been that there's no systematic training, in that you need to start with this part and move to this part. So it just happens that in your journey as a student, when there's need for bioinformatics training, that is when it comes up. But I don't really think it should be an excuse, because with getting knowledge from bits of bits, I grew up to a position whereby currently, by attending what we call codathons, whereby you meet and you're grouped into different kind of groups, then you get to run a project. One such program is by the African Society for Bioinformatics and Computational Biology. So I joined it last year in October. I was a participant then, but then based on how they were running things, I decided to pitch a project which I'm currently leading. It's called the GNBS Explore project, whereby we are trying to explore the feasibility of using genomics skills, bioinformatics in newborn screening. So I would say that it is really an uphill task for a student in Africa, but you still need to understand like what is driving you and be able to connect with the right people who know the right resources that you need to continue building your career, because most of this knowledge is in informal setting unless you actually enroll as a bioinformatics student full-time. You shouldn't like limit yourself to say that since I'm not training in bioinformatics, I don't want to get that kind of knowledge. So that is the gap I would say is there in brief.
Yeah, thank you so much. Thank you so much. I actually love those submissions that you actually brought because they're actually actual for a lot of persons. And how actual are they? For some persons, they're looking at having a systematic way of learning, but may not actually come to them like that. Some other persons actually need to do bioinformatics at a particular time of research, and they have to catch up with the reality of what bioinformatics is. And the accessibility of trainers and so on were things that you subtly mentioned over there, and that will actually take me to the next question. I hope to call on Mr. Dekunle after I throw this question to a lecturer that is over here. And the question is, today, majority of bioinformatics teaching is at postgraduate level, and student tends to enroll in these programs from different backgrounds and terms of formal training received. I mean, in terms of formal training received. And these trainings, both the lecturers and the student, they actually have to face their own challenge of catching up with the know-how. So the question is, Dr. Francis, the question is to you. What do you think is the biggest challenge of owning bioinformatics skills for trainers and for students and those in the field and those that are actually working? Yeah, so that's the question.
Okay, thank you, Mr. Betetutu, for the question. That's a very, very tricky question because the problem is there. And I think we all know the problem. We all know the problem. Bioinformatics is practical. Bioinformatics is practical. And most of the time, those getting involved into bioinformatics have never practiced bioinformatics. And I said initially, they are from various backgrounds. They don't know what is decoding. They don't know what is programming. They finish with their undergraduate level. They reach the graduate and the postgraduate level, and they generate data and they want to analyze the data. So definitely bringing together those individuals from various backgrounds into the same discipline, which is bioinformatics, is very, very difficult. As a lecturer, imagine that I start teaching bioinformatics at the postgraduate level. Means that someone who started his undergraduate in biology, in biochemistry, in mathematics, has never joined those skills together to make the discipline of bioinformatics as he will start at the postgraduate level. So for you or for me as a lecturer is already a great, great challenge. And this brings in the question of what or how is our national education policy being established? Because a new discipline like a bioinformatics, to get involved, this policy had to develop global guidelines to establish new bioinformatics degree program at different institutions for us to ascertain that those getting involved into the bioinformatics disciplines for the practical session or at the practical level, which is generally the postgraduate, when they generated their own data, they already have a very good background to get involved into the discipline and to master easily what they will be taught. I will take an example of one of my lecturers from Kenya, from Ilri. He told me that he started decoding at the age of 10. He started decoding at the age of 10. And me, when I started with I started getting involved with bioinformatics, I was already 30. And I want to master the skill he's using to analyze the biological data that I generated during my PhD. Imagine the difficulty, imagine the challenge. The guy was just playing with the code. The guy was just playing with the programs. And when you are looking at him, it's like he's coming from a new planet, a different planet, which unfortunately is not really easy to accept. But definitely it's a real situation, it's a true situation because of that system, which unfortunately did not allow us to start or to grow up by getting familiar with those basic background. And bioinformatics today is something that, as a biologist, you can't move without it. If you are dealing with biological data, if you are generating data, you need bioinformatics to analyze those data. So at this step, I think we just need to bring it our government, to bring it our policy, to interpret them, for them to understand the needs of this new field of study, to allow those interested to start at the basic background level, to get familiar with the discipline in order to become the good bioinformaticians that we need as trainer of the new generation. Because today, I started in biochemistry, I continue with molecular biology, and I'm getting familiar with bioinformatics. Definitely, my background or my skills, my experience will never be the same as the one of my instructor who started at the age of 10. So this is one of the aspects that I think we have to bring in, and that has to be considered by our policy.
Yeah, thank you so much for that submission. It's actually very actual. Yeah, because our realities in Africa isn't the same realities with other places. And now that you mentioned about one of your instructors that started coding at the age of 9, I remembered working in an academy where I had to go join some basic Python class over a year. Even though I was a teacher in that academy, I had to go join secondary school. I mean, from 8 students, I learned some of those coding, and I actually didn't experience it a lot until I was already my postgraduate. So that's actually a bridge. Yeah. So I will be throwing a similar question.
Yeah. Even maybe I face some. Before we move in, another challenge is the method of teaching. You know, most of us get involved into bioinformatics during workshops or maybe during short-term internship. And most of the time, those workshops and internships have been carried out abroad. And we do it for a very short period of time. And when we come back, the facility we were getting there are no more available in our institutions. And definitely this also impacts our training experience and also our long-term training capacity. So definitely, this is also something that has to be addressed. If I'm getting trained, how long? What sustainable measure is putting in place to ascertain that as soon as I'm starting, my training will get continued till I acquire the best experience that I need. So thank you.
Okay, yeah, thank you so much for that. Yeah, because it's actually, there's power in continuity, like Risi actually shared. So I would be throwing almost the same question to Risi, but I will be putting it in a different way, because Doctor just said that there is a systematic way of teaching about informatics. So let me ask you, Risi, have you recently joined? Joined a training program, and you were actually stuck in the process of training. What part of bioinformatics training seems hard for you? Yeah.
Okay, thank you so much for that question. So indeed, majority of the trainings that I have joined are usually at the basic introductory level. But then when I started working on this particular project, the genomics screening, I tried to join different training that now, you've been learning these skill sets, but you need to apply them. So at the application phase is when you actually realize, oh, so they used to say we call for variants, but I can't actually call for variants. Like, how do I go about it? So you usually get stuck when it's now time to actually apply the skill sets that you have gotten. So the training might be nice, structured, well structured, you are following it, but if you don't have actual data to actually apply the skill set that you have learned, then it becomes a problem.
So what you're trying to say is that, okay, sometimes we have the right access to these things, but we still need some hands-on practicals that that we can do by ourselves. So that's actually the application phase of the training. So for the application phase of the training, many times people get stuck, not just about the coding now, but about they already have access to the database. To the database and to the data they actually need. But at the same time, it's actually getting stuck. They actually get stuck in the process. Wow, this is actually true and real. And I know some questions are actually boiling in the minds of some persons. I want you to start to write your questions down so that we can start to answer it. Before we round up this session, I would love to call on Dr. On our consultant doctor to ask him. Hello, Dr. Shonibari, are you there?
Yes, I'm here.
Okay, lovely to still have you here with us. The question I want to ask from you is to understand what is actually the bottleneck really affecting professionals in the field from accessing bioinformatics knowledge from your own perspective. What do you think is actually the bottleneck that actually affects professionals, maybe genetic counselors, maybe clinical microbiologists and co, and these other professionals that we have on the field? What do you think is actually the bottleneck that's actually stopping them from actually getting access to this information?
Thank you very much. The major challenge is in the curriculum of training. Everything is dynamic. Now, bioinformatics has come in. I can tell you that when we started training, there were no molecular techniques, in medicine. And then gradually molecular techniques came in, and then we were drafted into doing molecular, researches. Although this has been far in other climes, but it gradually got into our system. And a lot of us in our training got involved in molecular techniques, molecular analysis of our, our samples, molecular techniques in diagnostics and things like that. And then we gradually get started imbibing it. And then molecular techniques cropped into our practice. A lot of our practice now, we use molecular techniques in diagnostics and things like that. And so bioinformatics will gradually get in when it gets integrated into the training. The next crop of professionals, as they come in, as they are trained, if the Nigerian government and the educational institution gradually put in bioinformatics into our regular institutions, so that these things are readily available and people get into it as they are trained. So as the training comes up, they get into bioinformatics and get to know that this is a useful tool in diagnostic and research. And it gradually creep into our system and creep into our practice. I am looking forward to a kind of cooperation among researchers so that those of us who are in the field, who are clinicians, will be able to, our diagnostics will be better. We'll have people we can rely on to talk to, say, okay, you can make my diagnosis better. Right now, we do a lot of molecular diagnosis. We want to go into bioinformatics too, since it's a new area of, research and diagnostics. We want to collaborate. We want to join people to do this. So the bottleneck is that for now, bioinformatics is relatively new in our environment. A lot of people have not caught up with it. By the time the research institutions started catching up with it, by the time the training institutions start catching up with it, by the time the postgraduate medical colleges catch up with bioinformatics as part of diagnostics, a lot of doctors, a lot of professionals, a lot of physicians like me who are research-oriented would go into that. And a lot of the research centers in Nigeria would also, when we get into bioinformatics, when these things get into our training, into our curriculum, into our practice, we'll gradually. So what I'm looking forward to as part of this workshop, this Omicslogic workshop, is a further introduction, a further foray into our institutions, so that a lot of us can, get introduced to this and get involved in it.
Thank you so much for that. And that's actually very, 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 very true. In recent times, I actually covered Nigeria this year, meeting with heads of department and major stakeholders in the different faculties. And I was so surprised that bioinformatics wasn't on the syllabus, even for the common biomedical students. So it's actually something that is actually very, very important, that even the medical schools, the medical students, the biomedical students will actually start to engage bioinformatics. Okay, so at this point, I would love to call on Mr. Adekunle. Mr. Adekunle, are you there? Hello, Mr. Adekunle, are you there?
Yes, I'm here.
Okay, so the quick question I would love to share with you is, you have actually in recent time you have actually engaged training individuals in the field of bioinformatics, and the question is, what do you think are actually the challenge to why the trainers, the students, and the lecturers, what do you think are actually the hindrance to these people opening up to bioinformatics training in Africa? Because they need to actually open up to it before they can actually get the training. So that's the question, sir.
All right, thank you for that question. I would say that, like the speakers that have spoken earlier on have said, there are a number of challenges when it comes to Africa also catching up with the speed of and use in omics sciences generally. But nonetheless, we have seen from Africa that, particularly here in Nigeria, that more and more people are getting interested in learning these skills. All right. How be some of the challenges we have noticed from our works, in recent past, ranges from, issue of economic power to be able to access this training. to assess these, these courses, these programs, also to some of the technical challenges that participants have during these programs. All right, you will agree with me, most of, we conduct most of our studies, our programs, researches, we do them online. And we've seen situations where participant begins to complain about even good internet accessibility. Okay, and this is quite a huge challenge. Equally, most of these programs come at a cost, and because of the economic power also, a number of persons get to complain. They have the desires to join these programs, but then they have no choice but to drop back at some point. And that's why we've seen more and more people join our free webinars and maybe workshops. Some of the workshops that are sponsored under the Pmi Biotech Scholarship, all right? And then to get the follow-on build-up training, because what we do with Omicslogic is a pedagogical training, building up the skills from the very basis. Okay, so you see them drop away from such follow-on trainings. But of course, I want to sincerely acknowledge here, what Omicslogic is doing here in Africa, particularly the support of the Pmi Biotech International Office, to ensure that the cost of these trainings is made available with a lot of partial scholarships and the rest for participants from Africa. All right, that's quite very, very helpful. But nonetheless, these are some of the challenges. We will talk about the technical aspects, talking about the internet. I've seen people even on some of our workshop programs complaining that their internet is not strong enough. Not necessarily that they are complaining their internet is not strong enough, but you find them having difficulties accessing some of those pipelines and platforms. And you get to let them know that it's about their internet network. They have to change locations, change their source of connectivity. And we've seen that get resolved, and then they move on. Then lastly, that I'd like to mention is, bioinformatics also comes with, a bit, I wouldn't say a bit, a good knowledge of coding skills, okay? And then we have seen that, particularly because they're getting introduced, yes, many of them, like Dr. Sunibari mentioned earlier on, while in school, they were never introduced even to molecular techniques, let alone bioinformatics, okay? And so while going through these programs, they're getting to see new concepts and learn new things. And especially when we now get to the coding aspect, a lot of them get lost and get discouraged. But that is why also I want to acknowledge what we are doing with the Omicslogic platform, the learn.omicslogic platform, because participants can get to, get introduced to these new feats, these new concepts, starting from the bytes and molecules, the basis, and then they build on that skill set. So with tenacity, definitely we've seen people move from that very basis, being a novice with bioinformatics and moving on to become an expert and even conducting their own researches now currently in their research fellowship. So these are a plethora of challenges we've been having, but thankfully we are trying to bridge this gap with our set of programs, and it is our hope that we can contribute to this until we have most of our institutions partnering with programs like ours and or reviewing the curriculum to accommodate these changes. So I think until we get to that stage, we'll keep making the progress as it's maybe in bits and pieces.
Yeah, thank you so much for that. Thank you so much for that. It has actually been very, very enlightening from this part, because bioinformatics actually has and promises a lot, and this section has actually opened our minds and our eyes to actually see the challenge. I would love to give us all the ability, a closing, a privilege to actually share a closing remark on one thing you think will fix bioinformatics training in Africa. So just round up, give us a closing sentence about one thing you think we fix bioinformatics training in Africa. So I would love to call on Risi.
Okay, thank you so much for the opportunity. So from my part as a student and having experienced bioinformatics in Africa, I would say we need to start small, maybe reach them when they're young, like don't just train postgraduate level. Can we try from high school level, like keep telling them there's this thing called bioinformatics, keep introducing them to coding, and also the way I have understood it is you don't need to be in bioinformatics, a bioinformatician to learn bioinformatics, because even in the life science field, you'll have to apply bioinformatics. So if we encourage more people, more students, like you don't need to be really, like once they understand that you don't really need to be a bioinformatician to get the bioinformatics skills, then I think more people will be open to learning. And then if you like it, then you can go on ahead and do it full time. So that would be my parting remarks, like let's get them young and let's encourage them to keep learning.
Thank you so much. Get them young, encourage them to keep learning. They just take it to our primary school, our secondary schools. Yeah, I love that. I love that. Okay, over to you, Dr. Francis.
Yeah, so I think one of the most important thing is that in addition to current bioinformatics training capacity building environments, like the H3A Bionets, like Omicslogic, like the ERI, we need additional flexible, sustainable, and high quality bioinformatics training environment. And I think that filling this gap will help us to ensure that the biological data derived of sample generated in Africa will finally be analyzed only by African bioinformaticians.
Wow, that's actually very, very powerful. No one will actually care for our Africa like we do. So we actually need to become the expert that we need to be able to solve the problem that we have. Thank you so much for that. That's so short and very, very, very powerful. Okay, I would love to call on Dr. Shonibare if he's still with us. Dr. Shonibare, are you there? Hello, Dr. Shonibare, are you there? Okay, so we'll give the final word to Mr. Adekunle Adeluwoye. Just give us a closing sentence, on how to go on with this.
Okay, please. Can you please repeat that?
Yeah. So the question is, can you give us a last advice or a last take home to the solution that you think we need in our bioinformatics training in Africa? Just a closing sentence.
All right, thank you very much. I think personally we need to begin to engage more and more the policymakers, the educational policymakers, in this continent about bioinformatics and the need to integrate this into the curriculum. I want to believe so much that we can actually scale the accessibility and the use of bioinformatics in our educational space. With that, of course, most of our engagement personally as a group, as a team here, have been much with the universities individually, and it has been quite a welcoming one. So I think at this point, with the various programs out there, H3 Bionet and the rest, Omicslogic, we might need to also begin to engage policymakers and ministries of education to let them see the need to actually take on bioinformatics actively into our high institutions. That would be that from my end. Thank you.
Thank you so much, everyone, for joining us. It has actually been a teachable moment for a lot of us, because a lot of persons have actually been thinking, how do I cross from this page to this page? How do I actually face this challenge, and how do I overcome this? The truth is that it is not new. We actually have to cross and jump over our own way, to build our own ladder our own way. And like we've heard from the persons on the field, that We need bioinformatics in our medical schools. We need it everywhere. We need it for the biomedicals and for the sciences, for every aspect of what we do. We need a curriculum that is changing, and we need you to actually be the change. You need to take it to the schools and so on. Thanks so much, everyone, for joining us. I would love to call on Dr. Adekunle for him to continue, because we have the representative of Prof on the ground now. Thank you so much, everyone.
Thank you.
All right, thank you very much for that. Thank you, Risi. Thank you, Dr. Francis. I want to thank Dr. Shonibari as well. So at this point, I want to welcome now Dr. Hello, can you hear me? Hello, can you hear me?
Yeah, we can hear you.
Okay, great. So at this junction, I want to welcome the representative of Dr. Professor Akinde. I am aware Professor Akinde is unavoidably able to join this meeting. However, I think an arrangement has been made to have someone stand in for him. So do we have Dr. Shii here? Hello?
Yeah,
okay, yes.
It was here some minutes ago. I think he's here.
Yes, he's still with us. I think I need to grant him ability to share his screen. So please kindly unmute yourself.
All right, done that.
Okay, lovely to hear you.
Thank you so much, Mr. Ayanfe. Abi Dr. Ayanfe, sorry.
Yeah. Lovely to have you here, sir.
I'm glad to hear your voice too.
Okay, please, you can go ahead to share your screen. Hello?
I'm so sorry for the delay. Please, I'm so sorry. I'm sharing my screen already. Can you see my? You have to enable me from your hand.
Yes, please, go ahead. You can go ahead.
You've not allowed.
Yes, please.
You see my screen? Hello? Can you see my screen? Hello? Can you see my screen?
Yes, we can.
Yes, we can see your screen.
Hello?
Yeah, we can see your screen, and but it's not in presenter's mode.
Can you see my screen?
Yes, we can see your screen, and you are breaking up. Your network is breaking up. Yes, we can see your screen, but your network is breaking up.
Oh, sorry about. Can you hear me now?
Okay, yes, we can hear you now.
All
yes,
right.
We can hear you now.
I'm so sorry. Okay, I'm so sorry. I'm Olani Oluwaseyi Paul, representing Professor Akinde, which is on another university assignment that is very urgent His end is really here, but we are so sorry for not bringing him in. However, I'll be stepping in for him.
Okay, okay.
All right, thank you. Yeah, according to him, the topic given to him is advancing biomedical research with omics data and computational biology in Africa, insight from our researchers. I hope I'm audible enough. Can you hear me?
Yes, we can follow. Yeah, we can hear you.
All right. All right, that's good. Okay. Now we cannot proceed to this. We cannot proceed in this presentation if we did not understand the significance of biomedical research. Biomedical research is the area of science to
I think we are actually losing you, sir. I think we are actually losing you.
research it
I think we are actually losing you.
Maybe we can move further with the US panel. They're also waiting with us.
Yeah, I think, hello, Dr. Shea. Hello, Dr. Shea, your internet is quite bad. We can't seem to follow. I think we may have to move on to the next. I mean, to the next presenter. Your internet is actually breaking, and your voice is not actually coming through. So we may have to move to the next presenter. I don't know, Dr. Shea, if you can actually hear me. Yeah. Yeah, I think we can't actually hear you. I'm sorry for that, but we may have to move to the next presenter. Hello, Jasvi, are you there?
Yes, hello. Yes. I'm
okay. I believe the US team can just take over from here because we actually lost them because of the internet issue over there. Then the other presenter cannot actually access the link.
Okay, no problem. So we have Grant with us. Maybe he can take over the stage now. Hi, Grant, are you there? Okay.
Good afternoon and hello. Kind of running on the speed up here. So glad to be with you. Can everybody hear my voice okay?
Yeah, we can hear you.
Very good. All right. So thank you all for your patience on those little things that happen. We are running a little bit ahead because of that, but we want to go ahead and get started. Thank you to that global community. We heard some really exciting updates from India and Africa, and we're now going to see how bioinformatics as a field is evolving here in the US and the Americas, as well as I said in the general Americas. Welcome to the Americas section of the Omics Research Symposium. Why are we here? Well, we know you have an interest in how omics are being used around the world. And over the next several hours, you'll hear from world-renowned experts in various omics fields, from the broadest uses of omics to areas of incredible drill-down niches. So stay tuned. My name is Grant, and I am your host for this afternoon. And I'd love to see where everyone is from. So if you would take a moment to drop a hello, I'd love to see it in your native tongue, because I'm sure we have some international people on the call. Drop a hello in your native tongue and maybe your country or locale in English, if you would. Drop that in the chat box. I would love to see that, if we could get some responses there. I know I saw some people from Africa earlier in that mode. But can we get some responses in the chat box? That would be really fun. Nice. Nigeria. Habari, is that your name or your locale? Great. India, Mambo. This is what I was hoping for. This is always amazing, isn't it? To see places from all around the world. Anybody else want to jump in? We've got, what, 60 people or so on the call. It would be fun to see who all 50 people on the call. Nigeria again, Malaysia. Relocated to the UK. I hope that's going well for you. Cameroon, Zimbabwe. This is fascinating. Thank you all. You know, at Pine, and keep going, please. Don't stop because I'm talking. At Pine Biotech, we are really interested to know the makeup of our audience. So we thought you might be also. I've got a little poll set up, and unfortunately, I don't have the official Zoom poll, but we're going to put a little poll in the chat box. And we would like to see some more information from you. So this is the poll, and you could just copy and paste one of those, or if you happen to have two or more things, like if you're both an MD and a PhD, if you wouldn't mind dropping that in, we would love to see what the makeup of our audience is and where you are at in your career. We would be fascinated by that. Who can chip in and give us some information on your current status? All right, pursuing a PhD. That's wonderful. Master's? Rissy, thank you. PhD is a lot of work. My brother got a PhD years ago, and he always called it piled higher and deeper. I don't know if that's still the case with getting a PhD, but that certainly seems like it is. Pursuing, good. We've got a lot of really high achieving people. Microbiologist, great. Pursuing a PhD. Nice. Well, thank you all for that. While we're looking at the poll results, if you're not yet registered at learnomicslogic.com for your free learning portal account, I'd like to encourage you to do that now. I'm going to put a link in the chat box. I don't want to interrupt the poll, so you guys just keep dropping it in, but that is the web address, and you can, again, it's free, and it sets you on a path to learning about this fascinating body of research and discovery. So if you sign up now at learn.omicslogic.com and click the register for free button, that'd be great if you could do that. I'm going to close that poll now. So thank you for those of you that jumped in on that. A lot of people really going for higher levels, and that is wonderful. And those of you that are maybe still in high school or something like that and did get a chance to chip in, it's a great field to be looking at. Since you're so willing to help, I'm going to do another one. What is your area of interest and data types? So I'm going to put a lead in here for you. So where are your interests? Are you still exploring? Are you into genomics, transcriptomics? Where are you at in your path? Where do you want to be? What is your interest? What is your data type? Genomics. Okay, thank you, Rissy. Thank you for participating in the poll. Another genomics. Fascinating. Nice drill down there. Epigenomics, cool, nice. These are really great results. You know, if you're wanting to start your career in this broad field, this is valuable insight to what other people are doing, excuse me. Anybody else want to chip in on that? We got a lot of genomics exploring. Cyrilli, if I said your name right, that's great. Epigemia, nice. All right, cool. So what will we hear this afternoon? And feel free to continue with the poll. What will we hear this afternoon? Well, in the US, data science has seen more adoption and more variety. So we're going to hear about some exciting ways omics data and data science is used, from algorithm development to training and applications. The need for more user-friendly tools to facilitate discovery and discussion around reproducible data analysis. Traditional use cases. We're going to look at some increased complexity as well as the availability of data. And then we're also going to look at new applications for this data. Traditional as in research and clinical, and then today as in wellness, beauty, and even supplements. Now, our first introduction is one of the masterminds behind this symposium and Pmi Biotech, a leading big data organization whose corporate vision it is to enhance human health and well-being by enabling biological research and discovery with relevant data, solutions, and support. But before I officially invite Ilya to speak, I want him to pop in and see the final results of our next survey, which is going to be kind of related to what we just did, but a little bit different. And I'm going to pop this in here. What is your application area of interest? Where are you going to take this? Now, some of you put that in above as you listed your area, but if you can answer that poll, we would be interested in seeing that also. Infectious diseases, neuroscience. What else do we have out there? Oncology, fascinating. Good for you guys. That's really, really fascinating. So Ilya, what do you think about these results? What are you starting to see?
Thank you, Grant. Yes, first of all, it's really exciting to see such a diverse background, such diverse interests, and I'm sure that today's session will be informative because we'll speak about how all of these different types of data converge. And as you mentioned, interesting new applications that maybe some of us have not thought of, but this is definitely exciting.
Cool. Well, great. Thank you for that response. And thank you all for playing. And since you guys have almost met him, I'll do an official introduction. One of the tech masterminds behind the symposium is Ilya Brodsky. Ilya is the co-founder, one of the co-founders of Pmi Biotech, and serves as CEO. His background is in applied mathematics and data science, and is currently completing his degree in business and management from Tulane University. He's facilitated and managed collaborations with researchers at Stanford, UCSF, Louisiana State University, Tulane, and other academic and pharma R&D teams. Mr. Brodsky oversees the strategic development of research projects and manages the team responsible for bioinformatics analysis, training, and collaborations for biotech. Ladies and gentlemen, let's kick off our North American session and our Americas session with Ilya Brodsky. Ilya, did I get all that in there right? Did I get it said right?
Yes, I think that was perfect. Thank you very much.
You're welcome. It's all yours.
All right, I'll share my screen and welcome everyone. This is going to be another kind of a transition into the rest of the speakers. We have an exciting set of topics and speakers that we'll be hearing from today. And so I am really excited. This is the third time we are doing this group meeting, this symposium. Really, it's supposed to be more of a discussion. So as you've seen up until now, we've heard from our representatives in both India and Africa. I've really heard some great insights about how education and research are converging, what are the needs and interests in these different regions. And I'm hoping that today we're going to learn more about some of these topics here in our Americas section of this today's session. And then tomorrow we have a great lineup of speakers again. So hopefully you will join that discussion as well. So just kind of like a high level overview of some of the insights that I've heard so far. It was really interesting to hear about the kind of greater interest and the need for hybrid education, where we see both online and in-person education. And especially in the domain of bioinformatics, where we see such growing amounts of data and growing complexity of data emerge, the need for biotechnology and life sciences degrees to incorporate bioinformatics and data science training into that teaching. So I think that is really relevant to what we're going to be discussing today. In Africa, we saw some of the big challenges that still the continent experiences with infrastructure, with ability to not only hear about bioinformatics, but use bioinformatics. and actually apply, I think, bioinformatics methods to areas of research that those participants came and spoke about. So we heard about your interests in environmental sciences, biotechnology, infectious diseases, non-communicable diseases, right? There's such a wide range of applications for bioinformatics. And so I'm hoping that today we'll kind of learn about some new ones, but that kind of presents a challenge, right? How do we actually learn bioinformatics and align that learning with those topics of interest? And how do we not only kind of follow the same technique that is one type of an approach for everyone, right? How do we personalize and match the training with the interests that all of you are pursuing? So we heard a lot about that, and I also am learning about it, and it's really interesting to be able to find collaborations as a result of this discussion, because there's so much I think that we can benefit from by joining forces and working to address these challenges together. So another important announcement that we heard today is the launch of the India Partnership Program. And Dr. Mohit Mazumdar, who is based in India, spoke about our recent developments in developing these long-term relationships with universities and building out a whole pipeline for students not only to be exposed to bioinformatics, but actually learning to answer biological and research questions with data, and working together with faculty as well as mentors from different institutions to advance publications, to advance research projects, and also to get experiences in industry. And we also heard about the research projects that different students are working on, right? So we heard about just a few examples. We have a lot more projects that are available on our portal. So Grant already mentioned, our portal is learn.omicslogic.com. And on that portal, you can see the training that we provide, but you will also see some of the projects that some of the graduates have completed. Thank you, Ajasvi, for sharing that link in the chat box. You can go and kind of check out some of those new and updated projects. Of course, the benefit of developing a project isn't only to complete a bioinformatics project, but it's also to be able to demonstrate how you can think critically, how you can apply your data science skills to a new and challenging topic, and how you can actually be presenting the results of your research or your analysis in a meaningful way. And I think to facilitate that, we've included some of the judges today, but we've also seen judges previously that came from a background in industry and academia and all kinds of applications of bioinformatics. So really, it's an opportunity to develop your profile and stand out from the crowd. Now, we also spoke about another challenge that I think is varied across the continent here in the US. And we'll talk about some of those challenges outside of these major biotechnology hubs in the US. But we also will hear about bioinformatics in Brazil, and we heard about bioinformatics in Africa and Nigeria. And we have people here from South Africa, from Malaysia, right, from India, from all over the world. And so another major challenge in all of these domains is ability to find mentorship and opportunities to join a research lab or even an internship or a job, finding a job. So recognizing this challenge, we want to really connect this ability to showcase what you have done with the opportunities out there. And so we've launched a job listing site where over the next few months, we will be interacting with the research labs, with the employers to make sure that we can build out that pipeline. And that all fits back into that long-term partnership program where universities can join our community, bring their students to learn about the basics of bioinformatics for absolutely free, develop an interest in a specific application, learn from mentors that are experts in that domain, and eventually showcase their achievements, their projects, their skill sets by demonstrating their projects and applying for those opportunities. So if you're here from a university, if you manage students, if you mentor students, we would be excited to speak to you after this symposium is over. So another important aspect of the projects that we saw in the project competition that we are holding this time, as well as what we've done in the previous times, is we're going to be looking to hand out prizes for these 3 categories. So we have the category of Best Technical and Research Project. Our judges are right now evaluating the projects that were submitted over the past few months. We have the Promising Research Direction and the Most Innovative Approach. So these are prizes that are going to be awarded tomorrow. If you have participated in the showcase, please join, but also all of you that are supporting these students that are showcasing their research projects, we invite you to join tomorrow and really see who won the prize, and also see some of our past participants that have won prizes and what they did after they got those prizes, right? Because they went on to join different programs where they continue their study or have used it to advance their research careers as well. Now, we already spoke about the significance of industry participation. And so as a part of this long-term partnership program, we're really working hard to develop relationships with companies that both hire and offer resources such as infrastructure to help advance bioinformatics and help prepare bioinformaticians of the future for the careers of the future. And so we are excited to now work together with a number of collaborators. Some of them you will meet over the next 2 days. But those include the Pharma Academy. They include companies like Google and Camp4 Therapeutics and others. And all of these are areas where bioinformatics is in very high demand and a significantly growing industry. And I think you will be able to see that by looking at the diversity of the job listings on our page. And I also want to mention that if you are here and you have a job opportunity or an internship, or some kind of an experience that you would like to share with our participants, please do submit a job, which is available on that page as well. So what are we going to hear about today? Today, we're going to be talking about advances in enabling technologies. Omics Research Symposium is obviously based on these different types of omics data. So we're going to be hearing about how the diversity of instruments. We have now Illumina, Oxford Nanopore, 10x Genomics. Recently, we heard about Ultimate Genomics, right? So the diversity of data that is being generated, the decreasing costs that are being associated with the sample preparation and sequencing, all of this is driving the need for complex data analysis, cleaning, and integration. And importantly, in different domains, we will hear about how it varies by application type. So we'll hear, for example, about applications in oncology, applications in forensic sciences, and tomorrow we will hear about other applications, including, for example, in beauty. So really a lot of interesting applications that we need to learn about, and the choice of data type, the choice of the sequencing platform, as well as how to manage and analyze such data is what we're going to be hearing about. Now, I'm a big believer in user-friendly tools, right? We all understand the significance of knowing mathematics and knowing statistics and machine learning and also knowing coding languages. Ultimately, you cannot be an expert in everything. And I think that this expectation from bioinformaticians to be both very sophisticated engineers, data engineers, and expert biologists is just sometimes unrealistic. And so to make bioinformatics more accessible, more kind of user-friendly, we need to make sure that a lot of these tools can be used by people with less experience. And so I'm really excited to hear about some of the updates that we're going to hear from Dr. Julia Panov, who's going to be speaking about the analysis of bulk and single-cell transcriptomics data, and from Elizaveta Taranenko, who is going to be speaking about the analysis of metagenomic data on the T-Bioinfo platform. So these are going to be exciting sessions where we will hear some of these important updates, and not only hear about them, but hopefully right away be able to use some of those as well. Now, I am also excited to announce that we are going to be hosting a bioinformatics hackathon. So we've tentatively set the date for November 17 to 22, where we are going to be hosting a hackathon for omics, wellness, and health. And the major target will be for students from around the world, as our participants here today, from Africa, from India, from the US, from South America, to find each other, build a team, and come together to build an application. And so the goal, and I hope that you take this perspective as you hear some of the speakers that are going to be speaking in just a few more minutes. This is an opportunity to identify a really clear application of omics data in wellness and health. And so I hope that after today's session, we will share announcements about application process, the different resources that we're going to be making available. That will include computing resources, data sets that you can start working with, as well as some mentorship and guidance that we will be making available for those of you that are interested in this opportunity. And the winning team is also going to be getting prizes and recognition for the application that they build. So let me briefly now describe some of the speakers that will cover some research topics that I think are very relevant to what we are doing here today. We're going to speak from Dr. Nikolai Oskolkov, who is a senior bioinformatician at the Department of Biology at Lund University, Sweden, and Science for Life Laboratory. He is going to be speaking about machine learning for multi-omics data integration. Now, this is a very interesting and hot topic in bioinformatics, so I hope that we will kind of see the significance of these challenges that new data types bring, but also to the need in being able to make sense of how does this data actually describe biological phenomena. We'll also talk about another person who is applying multi-omics integration or multi-omics techniques to the area of forensic sciences. And that's where Dr. Sarah Zapico, who is an assistant professor in the Department of Chemistry and Environmental Sciences, is going to speak to us about her current research, but also how she got into this whole field of bioinformatics, because her background is more on the biology, biochemistry side. And I think that this program, the Omicslogic program, in part was instrumental in her being able to incorporate bioinformatics into her work. We will also, sorry, there's a mistake on the slide, but we will also hear from Dr. Kasturi Kannan, not Julia Panov as it's written on the slide, but Dr. Kasturi Kannan, who is an assistant director at the MD Anderson Cancer Center. So really excited to hear from him about his experience in applying systems biology to oncology. And I think we'll learn a lot about his area of specialization as well. Now, we're also going to hear from a clinician, Dr. Luis Sakamoto, who is a pediatric oncologist in Brazil. And he will be speaking about precision medicine in pediatric oncology and what is the state of the art and perspectives. And I think it's important to kind of see how these different applications are maybe different by location as well. Now, before we continue, I want to remind you that we offer code or no-code bioinformatics paths for learning and mastering bioinformatics. And so we hope that anyone here today can participate in at least some of what we make available by visiting the Omicslogic portal. We welcome everyone to subscribe and register there for free, create a profile, and as this becomes more relevant in your area of research, to start benefiting from some of the resources, project examples, and mentorship opportunities that we provide. So we hope that really these kinds of programs can enable research across all of the regions that we have represented today. We offer big data analysis tools where you can make sense of complex relationships between phenotypes and genotypes. We offer mentorship with experts and peer examples that you can learn from. And we're also offering training that is both asynchronous training that you can leverage as a as a faculty member or even a teacher, and as a guided program that you can incorporate or even offer as a trainer yourself. So one of the important things that we are moving into is opening up the Omicslogic platform to other participants. So before we move on, I just briefly want to take a pause and kind of show you how you also can participate in this today. So let's go to learn omicslogic.com. And you can follow along. So really, I'll show some of the benefits of what you are able to find on our portal. I'm going to log out. And the way to sign up is simply to go to this link. And you can either sign in with an account that you already have or create an account like this. And so I am going to briefly log in. You're welcome to log in as well. Once you log in, the benefit is that you actually get access to all of these resources, including the Bioinfo platform, where you can try out different kinds of demo pipelines and really explore this whole path from beginning to the end. So we're just going to take a couple of minutes to take a look at this. We offer here courses, programs, and projects, and I briefly want to show what do all of these different things mean. So let's say you're interested in mastering Python or R, or you want to learn about single-cell RNA sequencing, or you have an interest in a specific project, like maybe COVID-19. So this is going to be an easy way where you can find any kind of a resource with curated data sets and case studies that are based on publications. You can also look for things like machine learning in R, and maybe you want to learn how to visualize and analyze data using complex machine learning methods. So really, there's a lot of different opportunities here. And a simple way to get started is just to click right here on Getting Started and see a collection of courses that are only around a specific topic. Now, once you are here, you also have a personal profile. On this profile, you can monitor and share your progress and achieve levels of expertise that later on you might be able to showcase if you're applying for a specific position. So here you will track the coursework that you are completing. You can also take mentor-guided programs that are actually led by an expert in that field. And importantly, you can develop an independent project. So developing an independent project is something that is, I think, critical to your mastery of bioinformatics. Not only are you going to be able to show that you've understood the challenge, the problem, found a data set, prepared a whole plan for analysis, but you'll be able to showcase what you have achieved at the end. So if you click right here on the projects tab on top, you'll be able to see some of the exciting projects that others have worked on over the past few months. You can see projects in all kinds of domains, from oncology to infectious diseases to metabolic diseases, neuroscience, and others. And so, as you can see, we also have some features here that you can comment on other projects or like them and highlight them. And that really makes it a much more engaging platform for everyone to participate in. Now, you might be asking, well, but how many people are actually using this platform at the moment? And so you can always check the activity on the portal by simply going to the Achievements tab. And you will see that, it's 11:47 right now. Just at 11:46, somebody already learned about bioinformatics, how to get started with bytes and molecules. You can see that somebody just signed up very recently at 11:16, right? So here you can really see a real-time flow of participation, and you can also comment and like this as well, as long as you are signed in. So what the Omicslogic portal offers you is a a comprehensive community and set of resources that you can leverage to learn and apply bioinformatics. Now, I also mentioned that we have this high priority for making bioinformatics accessible and user-friendly for anyone that is going to be a part of these training courses. And that's why we have the T-Bioinfo platform specifically designed to help you master analysis of next-generation sequencing, mass spec, structural biology, and all kinds of phenotypic data types. So on this platform, which, by the way, you get a free account with if you complete the registration on the Omicslogic portal, here you will get access to complex pipelines that you can try to analyze data. You'll be able to interactively create pipelines that produce results right away and learn as you go through this process to really not only be able to perform the analysis, but understand what goes on at each step of the way. In addition to that, you will be able to compare and contrast how selection of different algorithms defines the outcomes of your analysis and what are the types of results that you will end up getting in the end. So you will see here right away, we have some output data sets that we can explore. In addition to that, you will be able to store your pipelines for later use, and you'll be able to see how this data could be explored interactively, something that we will talk about in today's session in much greater detail, but actually play around with your data and really understand how can this data be informative for your analysis and for the types of things that you would like to understand. So really, not only are you getting the direct access to high quality resources that you can learn from and utilize to build upon to make your own research project, but also have a set of easy to use tools that really make this whole data analysis process much simpler and user-friendly in the end. So with that, I do want to briefly mention some of our initiatives in the US. I'm excited about just this year, we have started a series of pilot projects with an organization called the National Association of Idea Principal Investigators. This organization brings together members from 23 states and Puerto Rico and includes universities like University of Delaware and University of Hawaii and University of Puerto Rico and many others, where students are kind of outside of these major biotechnology hubs. On the West Coast, on the East Coast, we have many universities that specialize in biotechnology and have been able to put together impressive ecosystems that bring clinical research, biomedical research, biotechnology, companies, funding, etc. And so these 23 states and Puerto Rico represent areas that are underfunded and do not have access to such high quality resources. So we've been able to partner with this network and offer virtual summer programs such as machine learning for biomedical data, analysis of infectious disease data, oncology, as well as our research fellowship program. And we're excited to see participation from both research technicians, students, and faculty across many of these areas that were able to participate and complete successfully some of these programs. Now, the next step as we complete some of this basic training is to start opening up this platform to other universities and students around this network. And so what we'll be focusing on tomorrow is hearing from some of the members of this community from different locations, how they see the challenges associated with training in bioinformatics and data science for those who come from a life science and medical background, and how can we actually build a more consistent, high quality, and effective resource to advance biomedical research across these regions. Now, briefly, I want to also mention that we will be starting our next cycle of registrations for the research fellowship program. that is designed to help researchers and young researchers and students develop their bioinformatics skills as they kind of immerse themselves into research using bioinformatics. And so the goal there is not so much on skill, it is more on soft skills and critical thinking. How can you pick out your own data set? How can you technically define your research question and really apply your skills to answer that question with real data? We have heard some great examples today, but it is a very structured and clear process that we have designed over the last few years to make sure that anyone without any bioinformatics background can actually perform such analysis and be successful at it. And so we'll hear a little bit more about the need for such research experiences tomorrow, but I want to make sure that you're all aware of this program and you are able to participate if this is something that you would like to do in the next few months. All right, finally, my last exciting announcement for today is that Pine Biotech, with our Omicslogic Bioinformatics Education Portal, has been nominated as the finalist for the EdTech Cup. And this is hosted by StartEd. This is the largest EdTech accelerator in the US. And they brought together some experts in education technology and EdTech, as well as venture capital from leading firms that invest in such companies. And we hope that we can connect with them to really allow many more students to not only participate, but also really advance their careers and succeed in this field. So over the next few weeks, we'll be sending out information and announcements, and we really hope that we can get your support in succeeding in this competition and getting enough support to really advance to the next level. So again, thank you for joining today. I'm excited to hear our speakers, and I will pass this on to Grant to talk about what's coming up next.
Thank you, Ilya. I was just about to hit you with my timer and I didn't need it, so thank you for that. We appreciate all that information. Thank you for the symposium and the great lead into our time together today. It was also really fun to see more about the platform itself. So that was really great. All right, so how Omics works in our lives across many niches and specialties, more than we can list. Machine learning is a current hot topic and one we can always hear about in movies. Right? We all know about the machine learning thing. Well, one important trend we've seen over the past few years is multi-omics. The basic idea is simple. We see different platforms and patterns in genomics, transcriptomics, or epigenomics. Are these different patterns describing one complex biological phenomenon? And if so, what does the combination of these signatures in various omics data mean together? How we integrate the various features. Do we just use a database to connect mutations in a gene with expression levels in the same gene, or is it more complex than that? So our next speaker, Nikolay Oskolov, has a PhD in soft matter, theoretical physics, and statistical physics. In 2016, he joined MBIS, which is the National Bioinformatics Infrastructure in Sweden, as part of a bioinformatics long-term support team. Nikolay has extensive experience working with genomics, with more acronyms and exhaustive subject matter areas of interest than I have time to list right now. And he's also got some programming experience, just to throw it in case you need some software languages written. He'll be talking mostly about the integration of biomedical data, but also has research interests including single-cell genomics and evolutionary research. Nikolay, I can't wait to hear what you're going to be speaking on today. The floor is yours.
Hi everyone. Thank you very much, Grant, for this wonderful introduction. I'm going to share my screen and I hope you guys can see it. Otherwise, please let me know. Yes, so hello everyone. I'm really excited to be at this symposium, and it's a very nice to meet all of you guys virtually. My name is Nikolai Skolkov. I'm a bioinformatician from Lund University in Sweden, and I'm also employed by an organization which is called SciLife Labs, which stands for Science for Life Laboratory, and I belong to the bioinformatics platform of SciLife Lab, which is called NBIS, another acronym, the National Bioinformatics Infrastructure Sweden. And today I'm going to share our experience working with multi-omics data integration from a machine learning point of view. So I emphasize specifically that we are going to look at data integration from machine learning perspective, because this is how we do it. It's not the only way to do it. There are different ways, but I'm going to share our experience that established in our team in the National Bioinformatics Infrastructure Sweden. I hope it will be interesting and informative for you. Yes, so this project, this idea, we started talking about it a few years ago, maybe around 2015, 2016, because many labs in Sweden, many academic institutions in Sweden realized that they had plenty of heterogeneous biological data. And there was there was an idea whether we can combine somehow those different biological data types to put them together to maybe discover somehow new biology. So it was a vague, fuzzy idea. At that time, not so many people knew how exactly to do it. What are we going to do? But since then, I think we grew a lot in our understanding. And we even now teaching a course on biological data integration. I believe a link to the course material will be shared with all the participants. So what I'm going to present here, it's an introduction on how we, what exactly we understand by data integration, and how we evaluate it. So I will introduce a few concepts without going too much into the methodological details. So first of all, some terminology. I'm going to talk about a particular type of data integration, which is called vertical integration. Indeed, there is a little bit of confusion here. So some people introduced the terminology horizontal integration, and there is a concept of vertical integration. And those two concepts were nicely explained in a paper of Ricard Argelago and colleagues from Oliver Stegle lab in the University of Cambridge in the UK last year. So basically, they explain in this paper that there is a horizontal integration when you work within one omic, within one data type, and you simply combine statistical observations from different samples, different cells, different individuals, different labs, but you work within the same omic. For example, gene expression. So you have 20,000 protein coding genes. Their names are the same. You're just collecting samples from the US, from Sweden, maybe from Asia. So that's one type of integration, which is called a horizontal type of integration. I will not be talking about this type of integration because it's it's a known problem in bioinformatics, which is called the batch effect correction. So if you worked with, for example, gene expression data or methylation data or metabolomics, maybe you definitely know about this problem that you have to harmonize somehow your data to to basically to combine data sets coming from different labs. So I'm going to focus on the vertical integration because here we have a conceptual difficulty. I would argue that horizontal integration, it is a challenge, but people worked a lot in this area, and we made a lot of progress. So we have a pretty good understanding of how to do it. While with vertical data integration, here we are working, we are collecting multiple layers of biological information for the same biological cells, samples, individuals. So maybe we are collecting epigenetic information, genomics, and transcriptomics taken from the same biological cells. So how to combine these different data types? So there is a conceptual challenge here. What exactly we mean here? How exactly we are going to do it? And having said that, before we start talking about integration, more specifically, we have to realize one important challenge of working with any, basically, to my experience, any biological data. It's important to realize that biological data is high dimensional very often. And what does it mean, high dimensional? I'm doing analysis, so I'm an analyst. So for me, biological data, it's a multidimensional array or a matrix, simply put. And the matrix has 2 dimensions: the dimension of statistical observation, N. So this dimension can be, so it's basically your samples, cells, maybe nucleotides. So it's pictures if you are working with microscope imaging. And the second dimension is a P dimension. So here you're measuring certain variables in your experiment. It can be maybe genes, it can be proteins, maybe CpG sites if you're working with methylation. And you're measuring maybe methylation of a CpG site for 1 cell, 2nd cell, 3rd cell, etc. So those are your statistical observation, but the dimension of features, those are your CpG sites. And you can think about this matrix as of n data points living in p-dimensional space. So this p dimension, the dimension of features, it actually builds the dimensionality of where your statistical observations live. So you have p dimensions, and there are n data points living in this p-dimensional space. And the thing is that in order to do a robust statistical analysis, you have to sample this p-dimensional space really well. Otherwise, you do not satisfy assumptions of your statistical analysis. So you actually would like to have the number of data points n to be much greater than p. But in reality, we have the opposite limit. In reality, our biological data are almost always have this dimensionality p much greater than n. So if you're talking about maybe RNA-seq, you have 20,000 protein coding genes, and how many statistical observations you have, how many samples. If it's not single cell genomics, I would argue that maybe you have 100 samples, 1,000 samples, maybe, but not 20,000 really. So you actually are working on this p much greater than n limit. The sad thing is that when you combine different data types, when you combine biological omics, you actually, as I said, you're doing this vertical integration. So you keep n the same, but you are increasing p. So maybe P1 corresponds to genes. So it's a gene expression. So the first matrix can be gene expression, and the second matrix is methylation, measured for the same biological cells. So same biological cells means that N is the same, but P is actually increasing now when you put two data types together, because you had 20,000 genes, and then you add a few millions of CpG sites. So you actually your merged data becomes even more high dimensional. And why is it bad? Well, it's a problem because of the thing which is called the curse of dimensionality. What is the curse of dimensionality? So we are talking about robustness of statistical analysis for data integration. And so by statistical analysis, you can mean different things. People sometimes think about traditional frequency statistics with p-values, hypothesis tests, and power calculations. But there are also other approaches to do statistical analysis, such as Bayesian statistics or machine learning and deep learning. And the choice of statistical method, it actually it's actually defined by the dimensionality of your data. So it's very much about whether P is greater than N or N is greater than P. And so if you're working with this high dimensional limit, as I said, P much greater than N, that's a typical limit for biological and biomedical data. You sort of, sometimes people do traditional frequency statistics. I would rather recommend to do it in a Bayesian way because of sort of different assumptions. But what can happen if you have this high dimensional biological data and you continue with naive sort of frequentist statistics analysis? So here I just give you one example of manifestation of the curse of dimensionality. There are multiple examples, but that's probably most obvious. So this is a typical problem in typical frequency statistics analysis, which is called linear regression. When you have a linear dependence between your data, X matrix, and your phenotype of interest. For example, if you think about differential gene expression, or if you are thinking about genome-wide association studies, this is essentially what you are solving. You're solving this problem, a linear regression problem. And this regression problem can be solved in a closed form in this way. Basically, what you see here in the middle, this is so-called inverse variance covariance matrix. And it can be shown that in this limit, in the high dimensional limit, so remember your biological data are high dimensional. And if you do data integration, you increase the dimensionality of your data even more. So it can be shown that in this high dimensional limit, basically you can't inverse, you can't compute this inverse variance covariance matrix. You get into singularities. So that's a manifestation of the curse of dimensionality. So basically you have to always remember about this pitfall. When you do data integration, you have to watch for dimensionalities of your data sets. So that was a warning. Fortunately, we have an area in life sciences where the curse of dimensionality is sort of less of a problem. And this is single cell genomics. And a few years ago, it became sort of obvious that single cell genomics technologies such as 10x genomics can leverage hundreds of thousands of cells and even millions of cells. So at that time, a few years ago, we were talking about 1 million cells. Then very quickly, people started working with 2 million cells, 5 million cells. So we are talking about 5 million statistical observation. So here we are into the opposite, actually, limit, where you have N much greater than P. So for single cell genomics, this curse of dimensionality is less of a problem. But if you're doing data integration using bulk, sequencing, next generation sequencing data, you definitely would like to keep in mind the dimensions of your data matrices and avoid this pitfall, avoid the curse of dimensionality pitfall. Yes, and in single cell genomics, there are more and more technologies popping up basically every day that can deliver so-called multimodal data, meaning that you can measure multiple biological types of information from the same cells. So here I list a few of them, such as SCNMT, CiteSeq. The recent one was the 10x genomics multiome, really high throughput technology delivering ATAC-seq, open chromatin region information, and gene expression information from the same biological cells. So it's really exciting, the development. And that's why there is a lot of methodological development in this area regarding data integration. So now I will formally introduce of what exactly we mean by omics integration and how we evaluate whether it worked out, whether the integration worked out. So what exactly I mean by omics integration? So it can be illustrated with this simple cartoon. What you see on this plot, that's a number of statistical observations, a number of data points. Maybe cells or samples or individuals. One point is maybe one individual. And these individuals or samples, they belong to 3 different classes. And imagine that you have, you measured 2 types of biological information from the same cells or samples. Maybe you have 2 omics. So your omic 1, maybe RNA-seq, gene expression information, and your omic 2 maybe methylation. Let's take methylation. And so basically what I'm plotting here is that, so imagine that you take your gene expression matrix and you perform a principal component analysis, you do some dimensionality reduction of your data, and you take just first leading principal component, and you plot it as x-axis here. And then you take methylation data, you perform principal component analysis on your methylation data, you take PC1, your top leading principal component from the methylation data, and you plot it as y-axis. So for both types of biological information, they are available. So methylation and gene expression, they are available for the same cells, for the same samples. So in case there is some sort of so now imagine that you project your data points onto the x-axis and try to separate those 3 classes: the black, red, and green points. So they're very well mixed. There is no way to separate them using just one omic. The same is valid if you project your data points on the y-axis. They are very well mixed and they can't be separated. On the other hand, if you now put those 2 omics against each other, if you plot your data points the way I plotted them here, you can see that you can almost linearly separate those 3 classes, the data points belonging to 3 classes. And this is the beauty of data integration. You start discovering some new clustering, new patterns in the data that were not visible. These patterns were not visible for individual omics, but they become visible once you combine your data. I hope that's clear what I mean here. So the idea is to see patterns hidden in individual omics. And so that's all can be done providing you have some sort of covariation between your omics. So there should be some link, some sort of connection between your gene expression in this case and methylation. There should be some correlation. And there is a nice tool which is called MOFA, which we are sort of teaching our participants of our course, that can visualize how well your different data types are connected, correlated. So here there are sort of this heat map on the bottom, it demonstrates, it highlights sort of linkage between 3 different data types: single-cell bisulfite-seq methylation data, single-cell ATAC-seq, open chromatin region data, and single-cell RNA-seq for the same biological cells. And so these colors here mean that you basically, so the idea behind this MOFA, it's a factor analysis. Sort of you split your data into so-called orthogonal latent factors. And it turns out that within the first latent factor, you observe a connection between single-cell ATAC-seq and single-cell RNA-seq. So those colors here, they mean that there is some covariance, some correlation between ATAC-seq and RNA-seq. And bisulfite-seq and RNA-seq, they are connected, they are linked in the second factor. So that's a way to have a quick overview of whether we can proceed with data integration, whether it makes sense, whether we have something in common between different biological data types. So that was about what I mean exactly by data integration. It's sort of an ability to see something new, some new biology, some patterns that were not visible in individual omics. And now how I evaluate whether, so if suppose I performed some integrative analysis, so how do I know that it was successful? And the way we do it here, we are looking, we are using the concept of prediction. So we are looking at a boost in predictive capacity. So let me explain this a little bit better. So imagine that you have 3 different omics: methylation, gene expression, and maybe some phenotypic morphology data for the same biological cells. And these 3 different data types, they have very different statistical distributions. So it would be very naive just, to concatenate those 3 data matrices without taking into account the difference in the underlying statistical distributions of the data. That's why we need integration, proper integration. And here in the parentheses, I sort of visualize some accuracy of predicting something about the cells or samples. That's just a cartoon, so the numbers here are made up just for illustration. Maybe we want to know whether the samples or cells they came from they are good cells or bad cells. And we compute an accuracy of predicting whether it's a good cell or bad cell for each individual omic, and we achieve this kind of accuracy displayed in the parentheses for each individual omic. Now, what we want to see when we've done data integration is that the accuracy of classification, or whether it's a good cell or bad cell, whether it's a sick individual or healthy individual, we want to see that the data integration accuracy increased. So there was a boost in the accuracy of prediction. And this is actually why I'm saying that I'm explaining this data integration concepts from machine learning point of view, because prediction, it's a sort of very, very, very important concept in machine learning. Now, how exactly put these 3 different data types together? Here we have to dive into the methodology. There are different ways. So we explaining all these different types of data integration in our course. Briefly speaking, there are 3 directions. First is you can convert your data to some common space where the data kind of forgets about technological differences. So you convert them to some common space where it's safe to just concatenate those data matrices. And this is maybe neural networks, similarity network fusion, or UMAP can do this very easily. Second way is to explicitly model data distributions, and this is typical for all Bayesian ways of data integration. So if you have different shapes of data distributions and you can model them, you have some idea what was the underlying data distribution, then you can sort of combine these different data distributions in a Bayes rule. So if you're familiar with Bayes rule, that's basically joint probability of observing all your different data types. So you multiply sort of your different data types and combine them as a Bayes rule. And then more for what I mentioned previously, Bayesian networks are great at sort of integrating data through explicit modeling of their distributions. And then the third way is sort of extracting common variation. So you don't really account for difference in data distribution, but you sort of you factorize your data, extract common variation, and factorize it, and sort of try to learn something, some common factors, common axis of variation between your different data types. So I'm not explaining here details, I'm sorry, because then it will take more time, but you're very welcome to sort of come back to me if you want to know more. And as I said, so here I'm talking about data integration using this machine learning mentality. And maybe it's useful to sort of emphasize why I'm sort of talking about machine learning here is because of the prediction. So as I said, we are using prediction as an ultimate criterion of successful data integration. And that's very different actually from traditional statistical approaches. I would say, that's my opinion, it's not written in stone, but I believe that if you sort of work and if you do traditional statistical analysis, you are maybe mostly interested in detecting candidates, some drivers of your biological process. Maybe if you're thinking about sort of genome-wide association studies, this is a Manhattan plot where you can sort of detect genes that have something to do with type 2 diabetes, but those are separate genes. It's because you're testing them one by one. And the consequence of this this type of analysis, this traditional statistical analysis, that you sometimes can end up with a lack of prediction. For genetic studies, this is called the case of the missing heritability. Maybe you're familiar with this if you have experience in genetics. That's a very well-known problem there, and it literally means that genes delivered in this way, so you can get a list of genes that should have something to do with type 2 diabetes, but if you bring these genes to the clinics, you can't predict, you can't use this genetic signature in order to take a blood sample from an individual and predict the inclination, the risk of this individual for type 2 diabetes. It doesn't work, unfortunately, because of this, the case of missing heritability problem, which means the lack of prediction. So, and in machine learning, you optimize prediction from the very beginning. Maybe you're familiar with this paper, was a very famous paper a few years ago, where people sort of demonstrated how neural networks can be used for making very valuable clinical predictions. So really bringing machine learning to the clinics. And as I said, you here following this machine learning mentality, you really start optimizing prediction from the very beginning. And in statistical approach, you are not really interested in prediction, so to say, in the beginning. So that, I hope I delivered my message about the difference in the approaches. And by the way, even if you start with prediction in machine learning way, you still can have a look at the drivers. You can sort of roll it back and explain why your model can predict. So if your model can predict, you still can understand what are the genes, what are the drivers that provide this good prediction. But this is delivered by machine model as a byproduct. As I said, you from the beginning you start with prediction, and then you can of course try to interpret it and try to take a closer look why your model can predict. So now very quickly, I'll give you a practical example. So this was a theory. This was sort of what essentially we are doing in many different integrative omics methods. Now, just give you one quick example how well it works in reality. So here I'm using a multimodal single cell technology, which is called SCNMT-seq. It's one of the early multimodal technologies that delivers RNA-seq, bisulfite-seq, and ATAC-seq for the same biological cells. And as I said, it's quite naive to sort of just merge, just concatenate these data matrices. And one way to overcome this problem of different statistical distributions of the data would be to, as I mentioned previously, to convert this data to some common space, which can be done through graphs. So you simply, now one point here is one cell. And you're simply, so the edges here, they simply mean connections, correlations between cells. So here, regardless of the initial data distribution, you convert your data to some non-parametric representation where the graphs, they actually don't remember, they don't know anything about the technology that produced the data. But once you converted your data into graphs, you only keep the pairwise connections, pairwise correlations, pairwise links between your cells. So now, once you converted your data into graphs, now you can sort of use a concept which is called graph intersection. So it's demonstrated here on the left bottom corner. So when you intersect graphs, you simply keep edges that are consistently present in all omics. So looking at these three graphs, you can say that those two cells, so it's highlighted by red, the edge between these cells is highlighted by red. So these cells were connected here in single cell RNA-seq, and they were connected in single cell bisulfite-seq, and they were connected in single cell ATAC-seq. So they will be present on the consensus graph. So by doing this graph intersection, you build a consensus graph. And sometimes you can actually see new clusters when you combine gene expression with epigenetic. So this is if you want to build the graphs manually, but you don't have to do it actually, because there is a great tool, UMAP. And usually people use UMAP for visualization of high dimensional data. But you can also use it for data integration, because essentially on the first step of UMAP, you convert your data into graphs. And then there is one just line of code you can use to overlap different graphs. And here I'm demonstrating it for technology, single cell technology, multimodal technology, which is called CITE-seq, which is sort of RNA-seq and protein level information from the same biological cells. And this is how it looks like if you sort of overlap these two different UMAP plots. Sometimes you can see that this purple cluster here becomes more transparent. So here it was sort of smeared out and was not really detectable for clustering algorithm. But combining gene expression with protein level information, you start seeing new cell type, and perhaps this cell type has new functionality. So gene expression does not always separate functionally different cells. Gene expression alone might not be enough to sort of discriminate between cells with different functionalities. So I hope that's a little bit clear. And here I would like to wrap it up and thank you for your attention. This is our team, the National Bioinformatics Infrastructure Sweden, and I'm from Lund University. I really thank you for listening. Thanks. Grant, can you hear me?
Oh, maybe you can hear me now better. Can you hear me now?
Yes.
All right, very good. Yeah, the old microphone off trick. Thank you, Nikolay. That was fascinating, and thank you for helping us understand multi-omics data integration and the challenges that are being addressed in this way. To me, it sounds incredibly complicated, but I know there's others out there that know it better. It's also interesting at the same time, fascinating. I'm sure that many of you are working. How specialized and trained do you need to be in order to use these complex methods?
Is the question. Yes.
So, I mean, how specialized do you need to be able to use these complex methods? Is it pretty deep?
Yeah, so bioinformatics sort of implies some mathematical training, some fair understanding of mathematics and programming, I would say. So I would say that yes, so biology plus mathematics plus programming, this is pretty much what builds a good bioinformatician. Sort of it depends on the type of analysis you're doing. Sometimes you need to be a better biologist. Sometimes you need to be a better programmer. But what I was explaining, so since I have a mathematical background, I see the data in this way. And I hope you can also find it informative for your research.
Very good. I think a lot of the people on the call like to know that kind of stuff. So that gives us a nice background. Thank you again. We appreciate your time. Thank you.
Thank you very much.
Well, Julia, I'll let you undo your microphone like I just didn't. So in a totally different Omics universe, our next speaker, Dr. Julia Penov, is the Associate Director at the Tauber Bioinformatics Research Center in University of Haifa, Israel. At Tauber Bioinformatics Center, Julia, together with the Tauber Center team, are working to develop a bioinformatics platform for analysis of omics data. The platform is constantly developed and expanded to include state-of-the-art algorithms and approaches for the analysis of various types of omics data, including single-cell transcriptomics, spatial transcriptomics, metagenomics, and whole genome sequencing, along with a whole bunch of other stuff. Julia, it's a pleasure to have you with us. I'll let you roll. Thank you.
Thank you very much. Let me just share my screen. Let's see how I do this. And let's see, can you see my screen or also my notes?
We can see your screen just fine.
Great. Thank you. Just get ready for this. Okay. So thank you very much for the organizers, Ilya, Sonalika, Mohit, Raghav, for this opportunity to speak today. And thank you to the audience for your time and attention. I hope it will be worth your time. I'm very excited to share with you some of the improvements and the extensions that we at Tauber Bioinformatics Center, together with Pmi Biotech, have been working on for the last several months. Today, I will focus on the transcriptomics section of the platform, which includes the bulk RNA-seq dedicated to analysis of raw RNA-seq reads all the way up to differential expression and pathway analysis. A separate section on the platform for analysis of already pre-processed expression tables, which many times are deposited to the NCBI database and so can be uploaded to the platform and analyzed, and the post-processing analysis of single-cell expression data, which can be either generated on the platform or is provided by the 10x Genomics or in the NCBI database, and also can be uploaded here to the platform. So before going into the extensions and the new developments, let me just remind all of us about the basic steps of the bulk RNA-seq analysis. So the first step is the pre-processing of data. which includes the quality control, filtering out the adapter sequences from the raw reads, and filtering of the duplicated reads, which are mostly generated at the PCR amplification step of library preparation. And the next step is the alignment step, where the user needs to decide which reference should be used. It can be either transcriptome, which are individual transcripts from the genome annotation file, or the genome, where it is possible to identify newly spliced transcripts specific to the data set you are analyzing, and so specific to the molecular events which you are investigating. After the alignment on the reference, we need to quantify the expression of genes or isoforms, and they can be presented either as raw read counts, which are recommended for further downstream differential expression algorithms, such as DSEC2 or EDGE-R or others, or in normalized counts, such as FPKM or RPM. And finally, the post-processing usually includes differential analysis, some PCA analysis, or enrichment analysis. And if you're familiar with the platform, then you know that we have two basic sections to the platform, or two basic steps of the platform. The data upload section, where you are asked about your data, whether you are uploading reads from your own computer or from the NCBI database. Also, the user is asked about the reference, whether this reference genome can be found on our system. For example, here you can see that the genome is found in our system already, or the user is asked to upload his own reference. And another important step is the experimental, putting your samples into experimental groups. So you are asked to drag the samples into the correct groups so that afterwards differential analysis can be performed on these groups of samples. And the next part is the part where the user is asked to choose the algorithms and options. Again, there are many different algorithms and options for each step. And we try to help the user by separating each step into different columns of the platform, and also by color coding these steps. So pre-processing steps, which are optional, so are missed in this pipeline that you see on the screen, they are color coded in blue when pressed. Alignment algorithms are color coded in pink, and quantification of expression is green. And finally, the post-processing steps are orange. And when algorithms have been chosen and the pipeline is executed, you can turn off your computer because you will receive a notification email when the pipeline is finished. And if you have seen and worked with a platform, you know that when your pipeline is ready, you can download all your results from this download folder. And if we look at this folder, you will see many files, all of which might be important, but at first I think may feel somewhat overwhelming. So what do we have in this file? We have FastQC reports, which will tell us about the quality of our reads. We have alignment statistic table, which will tell us about the number of reads successfully aligned to the genome or the transcriptome. We also have the expression tables, raw tables for our genes and isoforms, and normalized in FPKM tables. In addition, we also have the DSEC normalized table with all the statistics, and the PCA table, and the pathway enrichment table. So to make it more readable and to try to help the user make order in the files, we now offer quick HTML graphs and interactive dashboards with most relevant graphs and tables. So this is how now the pipeline ends with links to the HTML graphs and with the external dashboard. For example, this first this first link will lead you to a graph with Monte QC, where you can check the quality of your reads and decide whether the pipeline that you chose was appropriate for your data, or whether you have to clean your data a little more than you thought was necessary before. So the other HTML link will show you, for example, volcano plot. And here also you can decide and get a first impression of your data and your analysis. And finally, the last button will lead you to an interactive dashboard. And the dashboard has, we think, has two main functions at the moment. First, you can more deeply explore your data, and it is interactive, so you can change the parameters on the dashboard, see what happens to your data when you change the parameters. So for example, you can change the thresholds of the volcano plot, or you can choose the top differentially expressed genes and see how they are expressed. You can also see your table and download your table. So this is one of the uses that we see in this dashboard. And the second use that we think can be useful for the users is that you can also share your analysis with your collaborators or other interested individuals. The dashboard is no longer dependent on the pipeline. And even if your collaborators don't have access to your pipelines or to the platform, you can still share the link with them, and they can download all the results, all the graphs, and all the tables, and they can explore your data together with you. Another potential use of these new developed dashboards is that many times, maybe you have run into this problem, you are running actually not one or two pipelines with the same data, but you are trying out many algorithms with different parameters. So for each data set, you might have 10 or 20 10 or 20 pipelines, and keeping track of all the tables and of all your graphs can be quite a challenge. So a dashboard or collection of these dashboards can also simplify things and can make order for the analysis and for choosing choosing the pipelines, the algorithms, and the parameters. So this was the dashboard for differential gene expression and RNA and bulk RNA-seq section. And we also developed the same similar dashboard for the single cell RNA-seq downstream analysis. But before going into the results, let's just go over the basic steps of the single cell RNA-seq analysis and maybe some of the challenges that this type of data actually presents. So in single cell RNA-seq analysis pipeline, we have three main steps of analysis, which are pre-processing, integration of several samples, and downstream analysis. So let me present each step using one of our projects where we investigated the changes in blood transcriptome in one of the marine tunicate organisms during hibernation caused by drop in water temperatures. So we had 5 single-cell RNA-seq expression tables generated from 3 active colonies and 2 hibernating colonies. So each sample consists of several thousand cells which are represented by about 18,000 genes. And the first step of analysis was normalizing the gene expression across all cells in each sample and selecting variable features or genes in each sample and reducing the dimensions of each sample using the PCA approach. And the second step of analysis was merging the different samples together. This is an important step, otherwise known as batch correction, and which was also actually mentioned by the previous speaker using, there are different algorithms of this batch correction of single cell data. So after correcting for the batch effect, we cluster the cells and project them on the two-dimensional plane, trying to sustain the local distances between cells. And for each cell cluster, we can find now genes differentiating this cluster from other clusters, also known as marker genes. And here in this particular project, we ran into some difficulty. And maybe some of you who are working with single cell data, and especially with non-model organisms, can relate. Actually, we found that only a small number of genes that we found as marker genes matched the known markers for tunicate blood cells. So we were quite at a puzzle what to do with these cells. And finally, without much success in assigning a particular name to the cell cluster, we still continued with our analysis. And we found genes that were differentially expressed in active colonies and in hibernating colonies. So this is how the actual analysis looked. First, we did quality control, and looking at the graphs generated by by some of these models that were by the pipeline, we can see that overall expression levels or counts in the hibernating colonies are lower than the inactive colonies across all cell types or all cell clusters. Also, the number of genes expressed in each cluster is much lower in hibernating colonies. And when we performed UMAP, looking at the UMAP projection of cells in the manifold, we can see that there are much less cells in the hibernating colonies compared to the active colonies. You can also produce a graph that will show it maybe a little bit more clearly if you have many cells. So here's the number of cells and the clusters, and you can see that the active colonies have a lot of cells almost in all clusters, while the number of cells in the hibernating colonies are very low. You can see here it's 4,000 and here it's just 75. Also, the density of expression per each cluster was very low in the hibernating colonies, with some clusters such as 11, 12 having no cells from the hibernating colony. The next step was finding the marker genes for each cluster and using these markers together with known markers of tunicate blood cells to assign a type of cell to each cluster. And again, as I mentioned before, this was a major problem actually for us. So for example, we identified 4 good marker genes for cluster 8, and these genes were highly expressed in most cells of cluster 8 and were almost not expressed in other cells in other clusters. However, when we tried to compare these genes to known markers of blood cells of this tunicate and to its close relatives, we could not find any genes that matched. So in usual cases, when you're working with model organisms, there are different approaches to finding the markers and to assigning cell types. These use machine learning algorithms and other single-cell atlases that are available for model organisms. But for non-model organisms, this is a bit more complicated and sometimes is not possible, at least for now. So finally, we could also find genes expressed only in hibernating colonies and almost not expressed at all in cells of active colonies. So we found 2 of these genes, gene 4248 and gene 2201. So I think I made quite clear that single-cell RNA-seq pipeline generates many graphs and tables, which are all collected in a folder in the result of the pipeline. And we tried to compress them. Each module is compressed into its own folder to make a bit more orderly the result files. However, still, we came to realize that it is still quite complicated to navigate all these multiple files, especially if you are just starting your way into single-cell analysis. And so we have also a dashboard where all results are collected and are interactive, and where the users can easily navigate through the graphs and files, changing slightly the parameters. And we find that this dashboard can be a huge advantage. It looks like, unfortunately, I don't actually have enough time to go over the dashboard, but I'm sure we will be able to put this dashboard online and put the video online, how you can navigate through the dashboard and what you can actually see. And again, I want to just mention that the dashboards can be shared after the analysis is done. And when you have multiple pipelines analyzing the same data set with different algorithms and different approaches, you can collect these dashboards in a more orderly way to remember and to come back to and reanalyze your data and share your research with others. So what we imagine for the future. And for the future, we imagine that these dashboards will gain momentum and will prove to be an excellent instrument for sharing biological data sets together with the results. For example, we already started some collections of data sets related to the same topic, and which can become a resource for researchers and where each researcher can just go right into the analysis, look at all the graphs, look at all the tables, download the code that comes with the pipeline. And we believe that these can really allow researchers to easily navigate all of the analysis of different models for the disease, for example, and may allow progress and quick interpretation of the underlying events, molecular events. Also, we are planning to make these dashboards more encompassing, including adding the option to download data together with all the code in R and Python, so that even analysis not performed on the platform can be easily performed by the researcher himself. And so we hope that the dashboards will become valuable tools for our students and researchers who use our platform, but also more wide community of researchers who can easily access these dashboards, these analysis, and these collections of data. So I want to thank again everyone on the Tauber and the Pine teams who were instrumental in all the developments I have been speaking about. Piotr Maishev, Avi Titievsky, Elizaveta Taranenko, Alexander Surdyaev, Vadim Sharov, Dr. Sofia Afanasieva, Dr. Leonid Brodsky, Professor Hanok Kafzan on the Tauber team, and Dr. Mohit Mazumdar, Dr. Radhav, Sonalika Ray, Ilya Brodsky. Thank you very much. And of course, thank you for your time and your attention. And if I have 3 minutes, so if there are any questions, I'm ready to take these.
All right, we've got a couple prepared for you. Well, first of all, thank you, Julie. That was very nice, and thank you for that information. What are the specific challenges that are needed to be considered when analyzing single cell data?
I think there are many different challenges.
We don't make it easy on you. Come on now. Go ahead, sorry.
Sure. There are many different challenges. First of all, there are many different technologies of generating this single cell data. And I think one should be at least aware of these technologies and use the algorithms that are appropriate for the technology that you're using. Another challenge is really what we call batch effect. So different samples have to be aligned with each other to be able to integrate them into one analysis. And another challenge is really annotation of the cell types if we're talking about some static tissue. And if we're talking about time course, then the challenges are even more, I think, hard to overcome.
Sounds like a whole nother topic to talk about.
Exactly.
We've got a lot of people learning here. So what are some of the different techniques, instruments, analytical techniques that you use to prepare an scRNA sequence data?
So I think basically the techniques can be divided into 2 types. There is wow, this is this is a very big topic, actually, when I come to think about it. And really, to be able to answer it, I would need another 10 minutes. So.
Okay. Very good. Well, we're about at a great transition point anyhow. So we really appreciate all that data and all of that information, and we really appreciate your time here. And so now that we know and have heard more about user-friendly solutions that make bioinformatics more accessible, I'm excited to see how researchers and students will utilize these methods. In fact, if I'm right, the platform you presented is already integrated into the Omicslogic portal. Is that correct?
Yes, I believe it is.
Okay, and that will allow students to learn and apply these techniques. So, Julia, again, thank you so much for your time, and we appreciate your being here.
Thank you very much. Thank you for the time.
Bye-bye. Our next speaker is Lisa Taraneko, and I hope I didn't trash your name too bad, Lisa. She earned a master's in genomics and bioinformatics at Siberian Federal University. So she is currently a PhD student in plant physiology. Her PhD thesis is about plant genomics, epigenetics, and genome annotation of oil palm. Wow. She is interested in all kinds of areas, loves working on mammals, studying genomics and transcriptomics. Another fascinating type of omics data is metagenomics. And we have been hearing about all the microbiome a lot, its role in health, disease, therapy, and even prevention. And now let's invite Lisa from the Tauber Bioinformatics Research Center to speak about metagenomics, data analysis, and how it can be performed in the Bioinfo platform. So welcome, Lisa. We really are looking forward to hearing about your area of interest. The floor is yours.
Thank you for having me. Can you see my screen well? Yep. Okay. Yep. My name is Lisa. Very happy to be here, and I'll be speaking about QIIME. It is a tool that we use to analyze metagenomics data. And metagenomics is really about microbial communities. We want to learn the composition of them and compare them between each other. Metagenomics is used in a variety of areas in agriculture, healthcare, biotechnology, we're everywhere. But this topic will be mostly about technical side of analysis on the platform, not the biological one. So on our platform, there is a possibility to, there is a possibility to use two types of data. Upload data. There is a manual upload when you have all the files downloaded on your laptop or computer, and NCBI upload. So the set of files needed for each is a bit different. In each case, you need metadata where you have the groupings of your interest. But for manual upload, you also have to have FASTQ files with your reads. And in case it is a multiplex data, you also need barcodes. So maybe you're already familiar with the concept of multiplex data. It's when in one run we have several samples and they're differentiated by the barcodes. So when this file is in QIIME, it needs these barcodes to separate the samples again. And in NCBI, we only need metadata, and this metadata is used to download all the FASTQ files that we need. So on the upload panel, you have to pick your type of upload, manual or NCBI, and you also have to pick if you have single or paired end reads, and select the file type if it's manual upload. For NCBI upload, this is a typical metadata file, and the critical column here is the first one, which has the SRA IDs, and they will be used to download all the data you need. Where to find this file? So you have to search the SRA archive and look for the study that interests you, and there will be this metadata button that will start the download of the metadata file. Just be careful because sometimes it downloads the file with commas as separators, but we can only accept tab separated files. So sometimes this file needs a bit of correcting before running the pipeline. So when the upload is done either manually or with the help of NCBI, we can start building our graph. And QIIME is separated into 5 subsections, and we start with pre-processing. So first we import data to QIIME, then we filter samples. We can filter out samples that are too few in the number of reads. And then we want to see the quality control, the quality of our reads. Then moving on to processing OTUs, that's operational taxonomic units. And here we have 2 algorithms, denoising algorithm, Data2 and DeBlur. And what they do, they create the they identify the operational taxonomic units, and they calculate the abundance in each sample. And with this table, we can proceed to taxonomy, where we classify our OTUs with existing database. In this case, it's Silva database for 16S RNA, where you have to pick your region of the region that was used for sequencing. In our case, it was variable region 4. And you can alter your classification, resulting data if you want. And then we can move on to metrics, where core metrics calculates all the alpha and beta diversity indexes. And then we can visualize alpha diversity with box plots. And for beta diversity, we will need to pick a column for testing beta diversity. And here I want to remind what beta diversity is about, because for alpha diversity, this is different measures that study the community within itself. But beta diversity compares the communities between each other. So here we have to pick some column that has a grouping of samples that we're interested in. In my case, it was the body side that the sample was taken from. And after beta diversity, we also can use interactive visualization. And there we also have to pick 2 columns to visualize. In my case, I picked body side and antibiotic usage. So when we are ready with our table and graph, and we ran this pipeline and it's finished successfully, you will see this external dashboard here. But actually, let me try and go to the browser. Okay, it's here. So this is the pipeline that successfully finished, and I want to check my results with the dashboard. So let's go here. You can see that in the dashboard, there are 3 areas of data. There's big bar plots, some NMDS, and alpha diversity box plots. And apart from this dashboard tab, we also have a table tab. This is the table that all our plots are based on, and you can have your OTU IDs and all the counts here for each sample. This is rarefied table, meaning that if you sum up all the abundances for each sample, they will be the same. You can download this data, and it will open you the raw look of this file. You can just copy it and paste it into text document. You can also download metadata or taxonomy, where for taxonomy it's the same logic. You have your OTU ID and all the taxonomic units. So, and also you can look what this OTU is associated with, what taxonomic, what taxa it is associated with. Here we have family, but again, you can look at different levels. So back to dashboard. This rarefied table is used to plot these bar plots. Let me make it a bit bigger. These bar plots that have samples and relative abundance, and you can play with this graph the way you want. You can, again, pick the different taxonomic levels. You can pick different coloring to your liking. Oh, this is not very good. Okay, let's stick to this. You can download a CSV of this table or anything. And if you're interested, for example, this sample, what tax it is, and what exactly the abundance is for this sample, for this taxa. Next is NMDS. This is something like a principal component analysis or principal coordinate analysis, also a dimensional reduction technique that tries to show the pairwise dissimilarities between the samples. Here it's also colors are controlled by these columns in your metadata. And if I want not 2, but only 1, I can pick the same color here and it will be like one group. Also, if we point at it, it will show what is the symbol and the color and coordinates in the 3-dimensional space and the sample name. And also the alpha diversity box plot. Here it only shows observed OTUs, and you can also group it by any column from your metadata. And for example, in body side, we have 71 observed O2s in gut and 165 observed O2s in palms, a lot more than in the gut. But this is the dashboard, but actually QIIME creates a bit more information than that. And if you want to look into beta diversity or more indexes from alpha diversity, you can go to download pipeline output files. And here you will see a zip folder for each job that finished successfully. And I'm interested in alpha diversity and in beta diversity, for example. So let me go back to my presentation. Sorry about it. Okay, we already finished this. Yeah, so here you have to get familiar with how actually QIIME operates. It has two types of data. It has QZA files and QZV files, and both are normally unreadable to us with our regular tools. QZA has all the tables in it, and so we modify it into TXT tables for you to look at. But QZV files are visualization files, and they're specifically designed to be viewed on the online browser of QIIME. And it looks like this, and you just take the file with QZV at the end and drag it here, drop it. And for example, let's look at beta diversity. Here, Bray-Curtis distance is used to calculate the distance metrics for the samples, and we colored them the same way with body side. We can see nice differentiation between the body sides, but both palms are together, which is also makes sense. Another distance metric, Jacquard, is used here, and they both are visualized in the Emperor plot, which is principal coordinate analysis algorithm. And we have two more distance metrics metrics that are used by QIIME. It's weighted unifractional and unweighted. Then if we move on to alpha diversity visualization, we can check out a bit more of the indices that there are. For example, faith index that shows us the evenness and richness of the community, but it takes into accordance the phylogenetic relationship between our OTUs. And here we again see that left palm and right palm are really rich compared to gut, but we actually can kind of see it in our bar plot. We can see that our gut samples, which are these, they are mostly comprised of three taxa. And for example, these are palms. We can see that there are a lot more different bacteria are there. So apart from box plots, QIIME also calculates statistics of all groups and pairwise statistics. And for example, here's gut versus our three other tissues. And we can see that there is significant difference with palms, but it's not significantly different from tongue, which also makes sense. And other metrics are the evenness, which shows how even each OTU is present in the sample. And just remembering the box plot I showed you, the palms are a lot more evenly present than the gut, where we have only three bacteria there. And Shannon index, which also shows evenness and richness. So thank you very much for your time and for having me here. I'm ready for questions.
All right, I am here. I just have to switch toys to help you out here. We do have a couple questions, and thank you for all that, Lisa. That was fascinating. So here's the first question. How has metagenomic data analysis been used in cancer research?
I'm not sure about cancer research since I'm really never did it in cancer research. But I believe that just metagenomics in general for human is used excessively to understand some physiological problems that cannot be explained by other techniques. For example, there are some diseases that genetically we kind of understand, but still the behavior of our GI tract cannot be only explained by the genetics. That's when a metagenomics step in and try to analyze the microbiome in our gut and explain this behavior. But in cancer, I don't know how it's used there.
Okay, sorry about that. It wasn't an attempt to throw you off. Just hoping you had an answer there, but thank you. Yeah, what are major challenges faced during analyzing metagenomic data?
Well, we rely heavily on the database to classify our OTUs. So I think this is one of the hardest parts, because most of the databases, they don't have ideal taxonomic information. Sometimes we end up with order of the bacteria, and that's the best that we can get. This is a bit troublesome. And also there is a difficulty with understanding when there is a contamination, because contamination mostly is bacterial contamination, and here we already are working with bacteria. So sometimes it gets tricky, but I think that classification is kind of the hardest part in metagenomics.
Okay, thank you. How about this one? How can one transition from statistical analysis to interpretation of metagenomic data analysis?
From statistical analysis?
To interpretation of the metagenomic data.
You mean statistical analysis, not a major in statistical analysis, just how to go from this Kruskal-Wallis test to understand how it's biological. Okay.
We'll go with that.
Well, you have to work with biologists sometimes because, well, if you really know the subject well, I guess you can interpret it yourself, but most of the time it really needs understanding of the processes and what we can expect, because a lot of things can go in a way that we do not expect, and that is wrong, because sometimes experiments go wrong, and we have to understand when we are wrong, where we're looking at something that doesn't make any sense. So I would pick a biologist in my team if I had a chance. And
phone a friend, right?
Sorry?
Phone a friend, right?
Yeah, something like that. Exactly.
Can microbial communities that you study be modulated with bacterial phages in a targeted way?
Sorry?
Can microbial communities that you study be modulated with bacteriophages in a targeted way?
We target them?
Yeah, bacteriophages. So can you target specific taxa of microbes with phages?
I'm not very knowledgeable in this area, but I think, yes, we can. But
and how would you identify such phages? Do you know?
Well, how we differentiate taxa by different differences in the genome in these variable parts. If we, I think that we can target our phages to these sequences, but I'm really not familiar with the process, how to do that.
Okay. How about this one? What are your thoughts on using the microbiome as a fertilizer in agriculture?
Well, I think I'm not sure, because there are different bacteria who, which work in the soil and which work in the gut. I'm not sure if the same bacteria will do the same job well. But we definitely use feces from other animals for soil, so maybe there is some idea in that.
Okay. Well, our apologies for putting you on the spot. We never know when the questions are going to come in and if they're all perfect for you. So thank you so much. And we will let you go for your afternoon. Thank you so much, Lisa. We really appreciate that. Yes,
Thank you. Bye.
Well, thank you again to Lisa and the team at the Tauber Bioinformatics Research Center. We're hoping to see more of this in the future as well. So I want to do a quick recap of what we've done and heard so far this afternoon. Ilya shared about the important trends in bioinformatics and highlighted the need for training, as well as the tools for user-friendly bioinformatics. And then we heard from Dr. Oskalov about multi-omics integration and the use of machine learning in this analysis. And then we heard from Julia and Lisa about user-friendly tools to perform really complex analysis. So you might be wondering, how are these types of data being used by researchers or industry, and what problems can be addressed by analyzing and integrating omics data? And to speak about that, we're going to introduce our next speaker. Many of us know the Smithsonian, but other than artifacts and a wonderful museum, we may not know about the research they do. I had no idea. Our next speaker, Dr. Sarah Zapico, is a researcher there, as well as having a day job, as it were, as the assistant professor at the Department of Chemistry and Environmental Sciences at the New Jersey Institute of Technology. Her research interests focus on the application of biochemical techniques to forensic science issues like age at death estimation and the determination of post-mortem interval, with implications in aging and biomedical research. Fascinating. It sounds like I'm going to watch an episode of NCIS, Sarah. The Symposium audience is waiting for more on your topic. Thank you.
Okay, thank you very much for this opportunity and the introduction. Let me share my screen. Can you see it?
We can share your screen. You might want to go to presentation mode in your slideshow menu.
Now, yes, it's fine.
There you go. Yep.
Okay, good. So first of all, yes, I am working in forensic science, but my PhD actually was in biomedical science in cancer research. But I always wanted to be a forensic scientist, so I changed during my postdoc at Smithsonian Institution. Because what I did and I am doing right now is translate what I learned during my PhD to forensic science issues. So I am going to go over the different issues we have. There are more, but where we can integrate the omics technologies. So here we have a crime scene that you can watch, for example, on NCIS. And one of the main things will be the identification of the perpetrator from evidence at the crime scene, or sometimes the victim, which is mainly what I do. I am not sure how familiar you are with the identification in forensic science, but we are using what is called in nuclear DNA length polymorphisms. These are repetitions of 4 to 6 nucleotides called short-tandem repeats. And these ones are normally in intros or intragenic spacers between genes. And the number of repetitions in each short tandem repeats, STR, vary between people. And the combination of this number of repetitions in each STR gives us the DNA profile that I will show you in the next slide. Actually, right now, the FBI has selected 20 core STR loss sites that must be run in all DNA tests in order to provide a common currency with DNA profiles to upload in the database that maybe you saw on TV as well. So this is, are not the 20 actually, but this is an example of an STR profile. What you see with the peaks here are the number of repetitions and in the top, the STR. Here, for example, the 21S11, this person is heterozygous with 32 repetitions in one chromosome and in the other one, 38. Here, TH01, you see is only one peak because this person is homozygous, 11, 11. And also, we check the sex through the amelogenic gene. XY means is a male. But what happens in these situations? Because what I show you is normally what you also watch on TV, the perfect profile, but it's not as easy. So many times we have mixtures, moreover in rape cases. Also in mass disaster scenarios, we can have degraded DNA and partial profiles. And it's when genomics, in this case, massive parallel sequence, plays a key role. First of all, I put here the forensic DNA signature prep kit from Verogen because it's the one approved by the FBI to upload in the database. So still, this kit, what it does is to analyze 27 global autosomal STRs, also YSTR, which are from the male chromosome, as you know, but are from the, for example, in cases of in rape cases, etc., and X-STRs from the female. Thanks to the coverage that I show you in the bottom, it's possible to better resolution in challenging samples, for example, degraded samples, and increase the resolution for mixtures, although sometimes it's still difficult. But still, the application of genomics, in this case to forensic science, could help in these challenging situations. But we always need a reference sample, so something to compare. It doesn't matter if we have the profile from the crime scene or the victim, if we don't have something to compare, a reference sample from the victim or a reference sample from a possible suspect. In this case, we can think about what is called forensic DNA phenotype. This means infer the sample donor's external visible characteristics, eye color, hair color, skin color from DNA, from the biological material at the crime scene, or from the unknown bodies. Also, we can include here ancestry and age. In this case, we use SNPs, single nucleotide polymorphisms. As you can see from the picture, are changes in only one base. And the combination of these changes are giving us the different colors, the eye color, the hair color, the skin color, and the ancestry. Here you have comparison between STRs and SNPs. And maybe you think if it will be possible also to identify a person using SNPs only. Yes, and as you can see here, we have the identity SNPs, but you require more SNPs, identity SNPs, to be able to provide an identification. And also, yes, phenotype SNPs and ancestry SNPs. So going back to the to this kit, this has two separate, I would say, kits inside of the kit. So the first one is the one I told you before with the STRs. The second one is this one with the 94 identity SNPs, 22 phenotyping SNPs for hair color, eye color, skin color, and 56 biogeographical ancestry. And here in the picture is the result of the software. But with this, you have to be careful because phenotyping and ancestry analysis are not allowed in some of the states. So moreover, in cases that you are looking for the perpetrator, because SNPs, in this case, phenotyping and biogeographical ancestry, constitute identifiable information. If you realize when I talk about STRs, STRs are in introns or intragenic spaces. So this one, you don't have, the information doesn't have any meaning, but this one does. So for that reason, in some places, some states, it's really difficult to use this, for example, for identification of the perpetrator, not as much for trying to identify the victim. And the other trend right now that I am sure heard a lot is the lineage SNPs and precisely forensic genetic genealogy. This is based on the looking for identical by descendant SNPs. So the SNPs we share with our ancestors, like here in this family tree. And here you have a table with the relations between the parents, etc. And how does it work? So when you do one of these tests, direct-to-consumer data tests, 23andMe, Ancestry, etc., now you have the option to decide if your data can be stored in the database or not. If this data is stored in the database, GetMatch or family tree DNA, then the forensic genealogists are able to access this data and go try to, for example, from an evidence of the crime scene, to see if you or whoever has a relation with the perpetrator, for example, from the crime scene. And these are the different, I would say, kits or microarrays, depending on the type of kit, depending on the type of kit. You see, they are overlapping. Some of them have more SNPs than others, but at the end, they do almost the same, but looking into different things. And this is the workflow. Basically, when the investigator has a cold case, because I am sure you heard by now, a lot of cold cases are solved thanks to forensic genealogy. So they have also to request permission to use the evidence from the crime scene for forensic genealogy. Then, depending on how the things are, there are some labs that they do the whole thing, analyze the SNPs, and then do the genetic genealogy, or others, they simply send to the labs, to analyze the SNPs and send to the genetic genealogy. So they do the microarray for the SNPs or whole genome sequencing, and then they send to the genealogists to do the genetic genealogy analysis. And based on that, then they can follow the trait to see if the suspect could be related to someone in the database. But again, we have another problem here, because forensic genetic genealogy, as forensic DNA phenotyping, provides investigative leads. To correctly identify the victim or the perpetrator, at the end, it requires STR analysis and comparison. So it doesn't matter that you have this in the forensic genetic genealogy. At the end, you need to compare the suspect STR profile with the profile from the crime scene. So again, we have another legal issue here. And I include this one here because we actually use for some cases, mostly degraded and difficult samples, is the analysis of mitochondrial DNA. The only problem it has is inherited from the mother. So all the relatives from the maternal lineage, they have the same mitochondrial DNA. It's not unique because you can, I would say, share, but kind of have the same mutations as other person. And here, what we analyze normally is the control region, which is HV1, HV2, and HV3. So you compare these, the mutations of these regions using normally Sanger sequence with what is called a revised Cambridge reference sequence. And then you compare and note the mutations. With these mutations, you determine the mitochondrial upload, where this, I would say, this person comes from the maternal lineage. So there is here another, this is also from Beroyen, Some labs also are using is the forensic mitochondrial DNA control region and whole genome kit. So they have again two options. You can only analyze control region, but the problem is if the person doesn't have enough mutations in the control region, it's better to get the whole mitochondrial haplogroup to get the whole genome. And this is an example of the results from one of the kits. And also this is the validation. You can see that they more or less get the same mutations from the control region and the whole genome kit. And we normally use this for this kind of samples that I told you, the greatest samples, difficult samples like bone, teeth, a hair without root, although now it's possible to get nuclear DNA from hair without root, but before it was more difficult. So normally you get mitochondrial DNA. Fingerprints, for example, because they don't have so much DNA, and burn remains or archaeological samples. So here we cover the genomics and including DNA profile, phenotyping, and genealogy. Going back to our crime scene, the other question is, how long ago the person died? And although on TV maybe used to is easy, actually it's more on one of the most challenging things in forensics. So maybe you are familiar with the terms rigor mortis, livor mortis, algor mortis. These are good, but for determination of post-mortem interval or PMI, but the main problem, they are good for the first days of death, and after that it's more difficult to determine the PMI. And also you can think about insect activity, but also has some disadvantage. And also it's not the same a corpse dying or decomposing here in New Jersey than, for example, in Miami. So one of the things to improve that is what we call thanatobiology. Is the use of advances based on DNA, RNA, and protein research for PMI estimation. And this is based on, at the beginning, the cells go to autolysis, but the cell nucleus remains intact for days after death. So we can apply cell biology methods for PMI estimation. And here we are talking first about transcriptomics. And I would like to show you, this is a study from my group. We analyzed the post-mortem interval in rat muscle, and but using actually RNA expression of different genes from autophagy and apoptosis signaling. What we realized was there is a switch in signaling in the first hours of death. At the beginning, there is an autophagy signaling because the cells are going to die, but try to mitigate the stress. But at the end, around 4-6 hours after death, there is the triggering of the apoptosis signaling, triggering the extrinsic and intrinsic pathway, and then releasing oxidative stress and other components that could try to mitigate that. So this was really interesting because of the switching signaling. So when I was actually a student for Pmi Biotech, I did, as a final project, I did this small study with previous data from rat retina. And if you are interested in the whole study, is in the YouTube channel, Application of Transcriptomics and Machine Learning to Postmortem Interval Estimation. And here I would like to show you the volcano plots that we developed. So you can see how from the beginning of the death, there are kind of changes in the expression of the different genes until the end, 24 hours is up to where they get samples. You see more genes are expressed. And what I really like, because is related to my previous research, is that when we analyze the pathways affected, here you can see apoptosis pathways, some genes were overexpressed and others downexpressed. So as you can see, this one more or less confirms our previous research. So we talk about transcriptomics, and now the next one, and also one of the trends right now, is proteomics, analysis of protein abundance in postmortem. There are more studies, but this one is more or less recent and also following with the mouse sapien. You can see how these are different animals, I will say, but they have similar outcomes in terms of proteomics, and also some of the proteins overlap, as you can see in the B. And in fact, they identify 2 proteins that decrease with the post-mortem interval in both animals, JPDH and EEF1A2. So this can be useful for analyzing the post-mortem interval in this case. So yeah, we have the proteome. And then another trend is the metabolomics, analysis of metabolites as a consequence of the decomposition process. And here, I would like to show you, this is from 2020, but I think it's really interesting because here they could do the study with human subjects without killing anybody. They did at the Texas State University Forensic Anthropology Research Facility, so body donations, and compare with the rats. And as you can see in the top, you have 2 human subjects. Over time, you can see how there is an increase in these metabolites. Some of them are amino acids, others are decompositional byproducts. And also in rats, you see mostly amino acids, free amino acids. So it could be useful in both rats and human subjects for PMI. So here we saw that proteomics and metabolomics can help for PMI estimation. The other approach for PMI estimation is the analysis of Thanatone microbiome. And this fits very well with the previous talk, because this is related to the analysis of changes in bacterial communities as a consequence of the decomposition process. So analysis here applying metagenomics. And here I would like to show you 2 studies from my colleague, Dr. Joa Tserias-Garriga. She did also these studies with human bodies from the Forensic Anthropology Research Facility at the University of Tennessee, Knoxville. And in this first study, she analyzed how the soil microbiome changed with the decomposition of the corpses. And as you can see in the 3 subjects, in this case called 3 donors, there you can see the same pattern. At the beginning, there are Proteobacteria, Firmicutes, Bacteroidetes, and others. At the end, mostly is Proteobacteria and Firmicutes in the 3 of them, with variability, because obviously there is some variability among the subject, but still you see the trend. But also what is really amazing is that in the second study, she analyzed the oral microbiome of the corpses. So see how this changes with the PMI. And you can see more or less the same trend in the 3 donors, but also you see at the end, the most common taxa or phyla is the Proteobacteria and Firmicutes. So these are really interesting studies, because also we can apply microbial analysis to determination of the PMI. And in fact, Dr. Tserias and I are going next week to the Forensic Anthropology Research Facility at Western Carolina University to continue with this research and the transcriptomic research. So here we talk about metagenomics for PMI estimation. And going back to our crime scene, and although I said this here, it will be one of the first things you try to do at crime scenes is identification of body fluids. Although you think it could be easy, it's not sometimes, because what it looks like to be blood, it could be ketchup, for example. So in this case, people approach this with proteomics analysis. There are different techniques, mainly MALDI-TOF or QTOF. and is based on the identification of proteins or combination of proteins characteristics of the body fluid. However, it's difficult to translate to the field because normally when we go to a crime scene, we try to detect body fluids using alternative light sources, and then when you identify that, you analyze on site. Sometimes if not, you send to the lab with, for example, rapid immunochromatographic tests, which are similar to the COVID rapid test that we use right now. So these are similar for blood, semen, saliva, etc. However, this proteomics analysis could be a confirmatory test for body fluid identification. And here you have a summary of the different proteins analyzing in the different main body fluids that you find at the crime scene. But I would like to show you like 2 examples. This one they did with Malditov. As you can see, they didn't analyze the whole spectra as previously, but you see the difference in the pattern between semenogenin, which is a protein from semen, versus alpha amylase, which is from saliva. You can see the pattern in each one in the mass spec. In this other study, you can see also the different proteins and the relative intensity or relative abundance. And in this case, the authors also, they analyze, pass through the QTOF samples from crime scene. So evidence samples, and even what they analyze, the different proteins match well with the body fluid. For example, semen, and also semen in different substrates, cotton, leather. So even with that, having the body fluid in a substrate, it doesn't matter because you can still identify the body fluid. So here we can include in the proteomics metabolomics the body fluid identification. So in conclusion, omics technologies can help to solve current issues in forensic science, like the identification of challenging samples and improving post-mortem interval estimation, among others. But omics approaches are slowly integrated into the forensic field due to legal limitations, as I described at the beginning. Future research will be able to expand these current studies, hopefully leading to a wider implementation of omics technologies. And as I told you, from our part, we are going to continue with research in transcriptomics and microbiome. As I told you, with Dr. Joe Garriga. I haven't talked about epigenetics, but I am using epigenetics for age estimation from teeth. But the integration of next generation sequencing technologies in this case is slowly as well. And also is using epigenetics for body fluid identification. Also, another thing I didn't mention, but is also still on the works, microbial forensics for human identification and body fluid identification. So here we need to include these, epigenetics and metagenomics. And I would like to thank first you all for listening, all the people involved in the post-mortem interval project, also Pmi Bio for the invitation and also for their help during the course I was doing. And one thing I am hiring, we are looking for a postdoc, biologist, biochemist, bioinformatics, someone who knows how to work with this data. It doesn't need to be a bioinformatics, but and also if you are interested in these topics. Thank you. You have any questions, I will be happy to answer now, or later you can send me an email. Thank you.
Thank you, Sarah. Forensics is such a captivating field. You know, we see it in TV, like I said, in movies, and yet so much more. Appreciate your time and knowledge for sharing with us. We do have a couple questions for you in our time that we've got left, so feel free to expand. How do forensic sciences benefit from the omics technologies?
So in general, for example, at the beginning, when I talk about genomics, in this case, because of the coverage of the omics technologies, is I would say is easier or more like it coverage more in terms of more for example, degraded samples. So the problem when we try to amplify the STRs in general, is that sometimes the primers are really but are not closer enough to, in the region. So integrated samples is one of the problems that you have. So with the coverage of the omics technologies, in genomics in this case, it's possible to cover this region several times, analyze several times, and try to be more precise and getting better profiles, for example. And then in post-mortem interval estimation, in this case, in general, transcriptomics, proteomics, etc., could help us the different studies described in improving the estimations.
Fascinating. So what are the skills required to establish a career in this field?
In forensic, okay, these are different skills. I mean, depending also on what you, now, it's funny because, you know, I am coming from biomedical science. I mean, I am biologist and biochemist. In Spain, I am originally from Spain. In Spain, we don't have so much in forensics. So I did my PhD in biomedical science. Still, I could change. So I did a master's in forensic anthropology and genetics at the University of Granada. And I changed to do my postdoc to forensic science. So I think the most important thing is you are interested in this field, you are willing to learn, and you have an open mind. You know, there are a lot of legal limitations in forensics, as I described, but I am sure doing research will help with so many things. For example, identification of human remains. So basically, yes, if you are interested in DNA, I mean, biology, biochemistry, yes, it's basically these are the skills.
You're gonna have to know a lot of that. I'm getting from all the speakers that it's just not, you gotta just go know this, you've gotta know a whole bunch of stuff, right?
Yeah.
Yeah.
Yes,
So
yes.
I'm gonna come back to real questions. I've got a question of my own. When are we gonna see you on TV?
I know.
Okay, back to real questions. So what are the current omics technologies used for data from forensic science?
So you mean in real forensic cases? It means because in forensic cases, I know for a fact that some people are using the, I mean, Biogen is not paying me for saying that, but it's because it's the one approved by the FBI. I know some forensic labs, they use the Biogen kit, the forensic signature kit. Also others, the STRs. Others use the mitochondrial DNA for sure. And also, because it's one of the things you heard a lot, the forensic genealogy. So applicable to forensic cases. Then in research, we are using all the techniques that I show you today. So one thing is research, but in terms of forensic cases, people are using the omics, the genomics forensic kit, then the forensic genealogy.
Okay. How did you get interested in this? What made you switch to this field?
Actually, I wanted to be a forensic scientist since I was a child. But again, the problem was in Spain, there were not so many opportunities. So yeah, I got my PhD. I mean, I am grateful for the time during my PhD. I learned a lot. And in fact, what you saw today related to the post-mortem interval, the RNA expression analysis was because of what I was doing during my PhD. But I really want, I know, and also some people will tell you that, yeah, the funding in forensic is not great, but still I think you are helping people. So I think this is the most important thing for me.
Thank you so much. We really appreciate your time here. I think, at least I think everybody's fascinated because of all the TV shows and movies, but it's still fascinating that all that can be determined in that way. So we really appreciate your time here, Sarah. Thank you for being on the symposium, and we'll bring you back here in a few minutes for the panel at the end. So stick around. We'll be right back to you. Thank you.
Thank you so much. Thank you.
You're welcome. Well, thank you again to Sarah. And before we move to the South America speaker presentation, I want to introduce Luis Carneiro. Lucas is our South America moderator and also a partner at Pine Biotech. And Lucas, if I can see you coming up on the screen here, there you are. No, there's still, there he is, there he is. We'll get you on the screen here. We're gonna have you go ahead and come out and have you introduce Omics training. It's all yours.
Thank you, Grant. Thank you all. First, I would like to thank the Academy and all the participants present here. It's with great honor that I would like to introduce and represent the participation of South America in this event. So I will share my screen with you all. Just a moment.
Lucas, you're muted at the moment. And we lost you. And we'll give Lucas a moment here to come back on. Dr. Sakamoto will be with you in just a minute. Oh, look, we're going to come to you early. It looks like Lucas has dropped out of Zoom. So we're going to go ahead and come to you, Luis, if you are ready for us. Well, let me see. We're trying to get him back in here. So give me just a minute, Luis. The fun of Zoom. Give us just a moment, please. All right, Luis, we're going to go ahead and pull the trigger here. I'm not seeing Lucas come back in, and we don't want to sag too long. So we're going to talk about precision. And we all know about being precise in language, or when measuring a board, measure twice, right? But precision is a relative thing. If we're going to the moon, we may measure in miles, and my woodworking is in fractions of an inch. But precision medicine and pediatric oncology is the specialty of our next speaker, Dr. Luis Sakamoto. Dr. Sakamoto is a medical doctor with more than 20 years of experience treating children and adolescents with cancer. God bless you, sir. His topic is the state of the art and perspectives relating to precision medicine and pediatric oncology. Luis, something tells me we're not going to be using my wood shop ruler.
Hello, hi everyone. Thanks for having me. Thanks for the organizer for the invitation to speak a little bit about my field, my work field, that is pediatric oncology. Well, I guess let me see if I can share my screen with you. Just a little bit. Okay. Let's see here. Are you seeing my screen?
Yes, we are. We can see your presentation just fine. Thank
you. Okay. Okay. are the topic that we will talk about today. I will, as I think there is few or even known attendants here that are used to deal with children with cancer. So I will bring you a little a few informations about pediatric oncology, and in the second part we will talk about how this bunch of information that is generated by the omics platforms are changing the way that we see the disease in children, and how we can use this technology to improve the efficacy of treatment, a efficacy that I will talk about this later, but a efficacy that is already really good when comparing to adulthood cancers. So let's go. Why I guess that there is no attendant here that is used to deal with children with cancer. It's because cancer is a rare event in childhood. In fact, when we compared the incidence of cancer in adults and in childhood, we can see that the numbers are very different, and we can conclude that cancer is a disease proper from older people. So it's really comprehensive, really understandable that there is no attendant here that used to deal with this type of disease. But the childhood cancer, although very rare, is in the high and middle income countries, is the second leading cause of death. In fact, cancer in childhood only only lost for the I'm sorry, just a minute. Oh, okay. In fact, cancer only lose for the accidents as a leading cause of death. And the major the most common types of cancers are that are related to hematopoietic system. So we have the leukemia that are the most common. In second place, lymphomas, and in the third place, the central nervous system's tumors. Although they are rare diseases, they are in the same way quite very curable diseases. If we see the evolution of treatment from the 1970s to 2010, we can see that there is a huge improvement in the efficacy of the treatment and a decrease in the mortality, especially for acute leukemias and brain lymphomas and brain cancer and lymphomas, especially non-Hodgkin and Hodgkin lymphomas. So this type of graph is it's a reason for all pediatric oncologists are very proud of this area of interest, because today we can cure a lot of more children than not cure. So why is it important to study this type of disease, given that we have a disease that is quite rare and it's very curable? The fact is, when we are talking about a disease that's very curable, we have to think to give back this person, this child, to the workforce, to the society, to the workforce. And as shown as a recent study that's published in Lancet Oncology, we see that cancer in the childhood is a very common cause of disability and morbidity in adulthood. So we are curing a lot, but at what cost we are curing? In middle and low income countries, however, we can see that this evolution of efficacy of treatment is not as good as in high income countries. We can see, for example, that the USA, France, Australia, European countries are far away and a lot better in terms of efficacy of treatment than Latin America and East Asia, for example. We have a problem with low and middle income countries that, in fact, we don't know exactly how we are treating these patients, because we don't have a system of register of cancer that is really really good. And we can see in this graph that the proportion of patients that are covered by these registers of cancers are almost 100% in North America, but in Latin America, for example, it's only less than 20%. So we are curing less, and even those with are not cured. I mean, we don't know that why they are not being cured. As I shown before in that graphic that shown the evolution of efficacy in pediatric oncology, all that evolution was due the improvement of supportive care and conventional chemotherapy. None of that evolution was due the new technology that we have today. And in the past, when we treated, for example, a leukemia, an acute lymphoblastic leukemia, all we did when we are characterizing that disease is to see the morphologic aspect of the cell and try to differentiate the cells from another kind of leukemic cells that are myeloid leukemia. So in that time, we have only the ability to differentiate myeloid cells from lymphoid cells. With the advent of the immunophenotyping, we had the capability to we were able to characterize more the cells in B cells or T cells, and to define subgroups of leukemic cells. And this is a methodology that is used nowadays to make the conventional diagnosis of leukemias. But with the advent of the new omics platforms, we are able, as you can see in this graph, we are able to define various subgroups of leukemic cells that have different clinical behaviors and different prognoses. For example, I would like to detail here this Philadelphia chromosome positive acute lymphoblastic leukemia that in the past has a really poor prognosis, but today with the advent of the tyrosine kinase inhibitors have a better prognosis, and with the advent of these Omics platforms, we were able to identify a group of patients that are similar to this Philadelphia chromosome positive patients, but in fact they don't have this chromosome altered chromosome. We now are able to differentiate, for example, acute myeloid leukemia from adults, originated from adults, from acute myeloid leukemia from childhood. As you can see here, the target study that is performed in childhood, as compared to the TCGA that was performed in adulthoods, demonstrate that we have a bunch of alterations that are totally different when comparing children and adults. As well, the genes that are more prevalent as altered in children are not almost never altered in the adults, and vice versa. Studying another type of neoplasms, that is medulloblastoma, that is a type of tumor that is quite common in childhood. It's originated from cerebellum in the fossa posterior of the cranium. In the past, we were able to classify only with the basis of histology, but nowadays with the advent of the Omics platforms, we are able to define 4 groups of patients, 4 groups of tumors that has quite different prognostic probabilities. Just to know, WNT tumors have a survival rate that is are really higher when compared to the group 3, for example, that are the group that have the worst prognostic and the worst survival rates. Another utility that we have for this new technologies is when we have some kinds of drugs, target therapies that are specific to some kinds of gene alterations, and we want to see if that alteration indicates the use of a specific drug. But to do that, we have to know if that molecular alteration is an actionable target. And what is the actionable target? Is a alteration, genetic or epigenetic, that can be used as a diagnostic or prognostic tool, as well can be predict treatment response. There are a lot of schemas that to define this if the target is in fact actionable, and I will not talk about this right now. But in general, we define 4 tiers of 4 levels of importance to define if that target is actionable. And the Tier 1 is that variants that have strong clinical significance, and that alterations implies in the almost in the obligation of use of that specific drug. The Tier 2 is when we have a variant that have a potential clinical significance, but we don't have for example, FDA approved therapy for that. Tier 3 is that alterations that have a non-clinical significance, and Tier 4 is just alterations that are probably passengers alterations that have no clinical significance. I brought here a clinical case just for an example, a case of a male with 2 years old, patients with a diagnosis of neuroblastoma. That is a type of tumor that originates from the sympathetic system. It was performed a NGS study that demonstrates that this tumor has a mutation G26S in ALK gene, and the problem is to define if this alteration is clinically relevant or not, and if it's relevant, if this patient is a candidate to the use of ALK inhibitors. And we can nowadays we can make use of some platforms that are publicly available. One of them is from the St. Jude cloud, and what we see here is that in fact there is a description of this mutation in ALK that already have been described in patients with neuroblastoma, but we can see here that the alterations that were described in the ALK related to neuroblastoma are in other regions that are not this mutation that was found in our patient. And the classic R1275Q that is related to a ALK inhibitor response is far away from this alteration that was found in our patient. Just a second, let me just erase this. When we made a search in another databases, we found that these alterations have no has an uncertain significant clinical significance, and in the cosmic database we found no potential target therapy that could be used for this patient. So in this case, the finding of alteration in ALK G26S has no clinical significance for the patient. So I think I'm done here.
Is that the end of your presentation?
I don't know. Yeah. Yeah. Okay.
All right. Thank you, Luis. Thank you for your incredible work. We all appreciate how important pediatric oncology is and how it can affect our families. So thank you again for sharing. And I'm sorry, I don't have me on the screen there. There we go. I'm not sure anybody wanted to see me, but there we are. It feels a little better. There's Dr. Sakamoto again. Thank you again. This time we're going to stay clear and bring Lucas back on the screen for just a moment. He had an intro a little while ago, and then we had a Zoom problem. That's the first time that's ever happened. So Lucas, I'm going to go ahead and take us off the screen and bring you back before we go to our panel discussion. It's all yours.
Okay. So should I start? Yes? Okay.
Yes, go right ahead, please.
So, okay. Let me share my screen. Okay. So as I was saying before, I would like to take the Academy and all the participants present here. It's with great honor that I would like to introduce and represent the participation of South America in this event. My name is Lucas. I'm an undergraduate student in statistics at the Federal University of Rio Grande do Norte, and I'm a moderator at Pmi Biotech. And I will start by talking about the plans and the perspectives for the contribution of Omicslogic program in Brazil, as well as the current scenario of bioinformatics and why to encourage the teaching of this area in Brazil. So we have about 2 programs only for the academic degrees of PhD and DSc according to the International Society of Computational Biology, and about just 6 universities that offer postgraduate courses focused on the area of bioinformatics with master and doctorate academic degrees. So in addition to these programs, there are graduate programs in computational biology that are offered by the Fiocruz Institute and the National Laboratory for Scientific Computing. It's not stable that bioinformatics is still a new science in development in Brazil, but also in the rest of the world. So some Brazilian states, especially in the Northeast, they lack this kind of knowledge, and there is an uneven distribution in the implementation of bioinformatics teaching among the Brazilian regions. So the use of tools and information obtained from the development of bioinformatics can bring significant advances to the field of education and Brazilian science. In that case, bioinformatics software is well accepted as an educational tool in different educational institutions in Brazil, because it allows students to associate the theoretical knowledge of biology and computing with the reality through the practice in a virtual environment. So with that in mind, Omicslogic training has been the focus of my work with biotech to reach the faculty and disseminate this kind of knowledge in practice among the students. And with that, to encourage these people in the citizen science of South America to participate in this community to contribute with the advancement of this area in the country. So I was talking about the precision medicine before the Dr. Luis, but let me briefly highlight a few important points about the topics of omics data and bioinformatics in South America. In 2017, the Brazilian Association of Personalized and Precision Medicine was created. There are small technology-based companies, albeit few, are now able to identify new markers and develop new tests that utilize genomics for medicine. In Brazil, this area is still underdeveloped in reality compared to other countries like the US, but there is a trend to expand and grow. So with that in mind, training for new students is critical for the success of this domain. Omicslogic training and its programs are designed to introduce anyone from a life science background to the concepts and logic behind omics data, bioinformatics, and their applications, And today I'm excited to share that we are translating the materials into Portuguese and Spanish, and we'll be making them available for free over the next few months. I'm excited about the potential for precision medicine and that we have so much data available to guide clinicians to adapt this approach to the needs of patients across Brazil and South America. So that's it. After that, I would introduce Dr. Luis Sakamoto to talk about what the future holds for precision medicine to become a reality for every patient, starting from the ones who most benefit, the children.
Thank you.
Have some time to questions.
Yeah, thank you, Lucas, for the introduction, and thank you, Dr. Sakamoto, for sharing these insights. I think we can go on and move on to our next speaker, Dr. Kasturi Kannan. So Grant, if you don't mind introducing him.
We'll do that. Thank you so much. This time we're going to stay in the same galaxy of cancer systems, but a totally different solar system, as it were. We'll also touch on machine learning, statistics, and large-scale data. Our passionate speaker is Dr. Kasturi Kannan, an associate professor and director of computational pathology program at the University of Texas MD Anderson Cancer Center in Houston, Texas. He's a data scientist with an expertise in cancer genomics, epigenomics, biomedical informatics, and image processing. Kasturi is also working currently on systems biology and spatial stochastic processes with its application to multiplexed immunofluorescent images. I'm not even sure what I just said, but I'm sure it'll all be clear in the next 30 minutes or so. Kasturi, the stage is yours. Thank you, sir, for being here.
Oh, thank you, Holmes. This has been a wonderful gathering, and it's my pleasure. I have known Ilya for quite some time, and I'm nice to meet him and everybody here. So let me start sharing my screen. Are you able to see my screen?
We can see your screen, but you might want to go to, there you go. Absolutely great.
All right. Okay, so let's get started without any further ado. Today, I'm going to talk about bioinformatics and cancer systems biology. In particular, I'm going to touch upon why bioinformatics has been critical for cancer systems biology. I have nothing to disclose or no conflict of interest. Before going into the topic, first of all, I would like to establish my credentials on this area in general about what led me so far to the current position here. Basically, I'm a mathematician. I got my master's in mathematics from India, and then I moved on here to study mathematics. In particular, I was more interested in topology, approximation theory, etc., etc. But my interest shifted towards computer science, and I graduated with a PhD in computer science, especially in computational physics. Until about 2008, I have no idea of what bioinformatics is or biology or bioinformatics. I should share an anecdote that, in fact, my mentor, she wanted me to pursue bioinformatics, and she was interested in protein folding. And at that point, I said, no, I'm not at all interested in bioinformatics. But it later turned out that I went to a medical research institute where I got some basics of biology, and that led me naturally to bioinformatics, working with the data sets, biological data sets. And I was doing genomics, large-scale systems, studying brain tumors at Memorial Sloan Kettering Cancer Center. And then that led me to the position at New York University, where I was an assistant professor. And I came to know about Ilya, and we had a good conversation, and I got introduced to him at that point. And slowly that led me to a current position at MD Anderson Cancer Center. I'm an associate professor and director driving majorly what is known as computational pathology. But that's not going to be the topic of today. I'm going to more talk about another exciting area that I've been working for about maybe 2 years now. In general, my expertise or it's varied. It's like a Swiss Army knife. I mean, in general, I would like to call myself as a data scientist. About 15 years, specifically focusing on cancer research. All right. So let's go to the topic. Let's go to the state of modern biomedicine, right? I mean, it's like a big elephant, and we all know the story that elephant and blind men, right? Some people touching their tails and thinking that's a snake, and some of them touch the skin and saying it's a wall, etc., etc. But nobody gets a big picture, right? I mean, it's so fragmented, and this fragmentation actually appears, especially when it comes to drug discovery. We have water everywhere, not a drop to drink, and data everywhere, not a drug. I think that's one of the famous quote that's been widely used now. But the key thing is that we all know that the medicine is very, very fragmented and bifurcated. And so we are having a hard time in reconciling, what directions to pursue, what are all the good targets. I mean, people go by single cell RNA-seq, or I mean, all the things have now come to the fore. A good thing is that we have discovering so much, the data is everywhere. But unfortunately, it's all difficult to put everything together to find a bigger picture of what's happening in modern biomedicine. So the key question as we dive into the topic is about nature versus nurture, right? At the end of the day, we can think of genetics as something hard-coded in our DNA, right? Mutations, deletions, etc., etc. And then there is a whole phenomenon of epigenetics where the methylation and other things can contribute to gene expression silencing, and we know that this is another major mainstream mechanism. So people are like, some people subscribe to genetics and some people subscribe to epigenetics, like I was telling about the elephant and blind man, right? I mean, I have seen people saying that, okay, I am a geneticist and I believe in epigenetics. So the key question is how they interplay, right? One of you, I mean, whoever have a strong base in studying the DNA might subscribe to the DNA model, the basic central dogma, and say that genetics is the key thing that's driving, but we really don't know the answer. That's the truth. So in the absence of consensus, right, in God we trust, all others must bring data. So what does that data tell us, right? Whether it subscribes to genetics or whether it says that it's genetics and epigenetics, we know that it's both, but how much of it, right? I mean, the problem is that the data, now we have a deluge in biology, right? Explosion of data sets. Now we have the NGS, you're all bioinformaticians, you know about all these things, I don't have to tell. But then the primary aspects of the data are mutations, copy numbers when it comes to DNA. Of course, gene expression could be controlled both by the DNA as well as the RNA. And there are so many other things like structural variants and so many other things that we could study that we are unable to get a consensus of what actually is driving, right, the tumors. So yeah, I mean, it's like studying the wings, right? And saying that, oh, the aeroplane just runs because of wings. And some people study the engine saying that, oh, it runs because of engine, right? Of course, we know that it's both, but how do we study both, right, without ripping the plane apart, right? How do we really study the entire structure, right? The aerodynamics is not just an outcome of just the wings or an engine alone, right? The aerodynamics is determined by the flow of the air, and it's a process, right? The dynamical process. It's a spatio-temporal process, and not a static thing that we could study by ripping things apart, right? So these are some of the things that I have been thinking for a long time. And that's when I hit upon the systems biology. And I've been thinking about systems biology. I've tried to watch some videos, and everywhere when people talk about systems biology, there is not a definition of it, right? People are saying, okay, yeah, I mean, systems biology, it's tough to define, right? So the problem is because, let's forget the systems biology for a moment, but the whole point of just studying these mechanisms, the biological mechanisms, is that the modern biology is more or less reductionistic, right? We break it apart, we study, hoping that when we study these parts and try to put together, we will get a understanding of the entire mechanism, right? I mean, it is like saying that you have, let's say that we have a bunch of best cars in the world, right? I mean, one is good for, let's say, a BMW as a best engine, and a Volvo probably has the best transmission, and say some other car has a best battery or something like that. So when we are trying to rip everything separately and trying to organize something that we can put together, we will not even get an automobile. That's the unfortunate thing. We will be able to rip it apart, we will be able to study, but putting it together is not, it's possibly impossible. So the divide and conquer approach will not work when studying complex systems, especially biological systems. Especially cancer, right? Cancer is a very complex and adaptive. It's an evolutionary mechanism. The dividing cells constantly adapt to their environment. And so it becomes it's an adaptive system, and so we will not be able to put together and study how the mechanism works in real time, right? So when we think about how reductionism works, it is like a relational database, right? I mean, we break things into tables and we study each tables, right? And then we hope to do some joins and understanding, okay, so I'm going to join this table X with table Y, using some primary key or a foreign key, and hoping to understand table X and table Y together, right? I mean, these joins are made out of inference. It is like a reductionism. So you can actually think about reductionism as a relational database, but this is not going to work, because putting it together, it's just an interpretation at some point, right? It's not going to reveal an answer by itself. So we need an integrative paradigm, which is known as a systems approach. A systems approach is putting things together and studying it in a very comprehensive fashion. Whether are we ready there yet, whether are we, do we know how the mechanism works by integration? That's something that's the biology of the future. But the right approach would be to actually put things together to see that what makes sense and what doesn't make sense. The systems approach is characterized by a simple thing: the whole is not the sum of its parts. So that's the key sentence that defines systems biology. So the entire picture that you see is not going to be the sum of the parts, because the interaction between the parts matters as much as the parts themselves. Without the interaction between the parts, the part doesn't make any sense whatsoever. So that is the entire idea behind systems biology. So how do we then define it, right? So I was looking into the definitions of systems biology, and I came into this Institute for Systems Biology, and they have this model where they say that, okay, you have the technology, and then new technologies give rise to new data, and then new data give rise to new software. And then out of that, doing some bioinformatics, we form new hypotheses, and then the hypothesis will gain rise to new insights and then new biological questions. This is a life cycle of technology, computation, and biology that defines systems biology. But this is not true, because it's an integrative technology that defines systems biology. The integration, like I said, the whole is not the sum of the parts. Systems biology and complex adaptive systems are defined by the integration. And when you look at it, it's just the technology, just doing some next generation sequencing or any of these is not going to be, or imaging is not going to define systems biology. All we will get is a bunch of data. And of course, we can do bioinformatics with a bunch of data and then try to answer some questions, but that is not what systems biology is. So with this, we started to trying to think about the goal of the project. So I started thinking about, okay, what it makes sense, but I'm going to, before I go into the details of how we can really identify systems biology and what it has to do with bioinformatics, right? I was thinking of doing a project actually to define, to really showcase what it is, right? So the goal of the project is creating a unifying computational framework. So it's a computational framework, and we want to integrate all available patient data. Okay, so we have the tumor recurrence data, the primary data, and the recurrence data, and so we want to integrate all of them, and then in an unbiased manner for actionable target validation and predicting disease outcomes. So that's the idea behind the project that we initiated at MD Anderson Cancer Center. When I joined, I wanted to start the systems biology initiative for brain tumors. And the key thing is about integration, right? It's an integrative technology that we have to use here. So why brain tumors? Of course, it has been my expertise for a long time. For about 10 years, I've been involved in brain tumor research, both in neurosurgery and neuro-oncology. In neurosurgery, I hold a joint appointment there. So in general, I have been studying brain tumors a lot. And so that was natural for me to do this cancer systems biology in glioma. So gliomas are in general the nervous system neoplasms that is derived from epithelial cells. In general, the differentiation or classification is by glial cell types, right? I mean, usually the brain cells, if you take, there are neurons, there are astrocytes, oligodendrocytes, and epidermocytes. So in general, the gliomas arise from glial cells, and it constitutes about 30% of all central nervous system tumors. And in fact, in particular, if you look at the brain tumors, we have about 70% of them are brain tumors or gliomas. So they are divided into two major categories, the low-grade glioma and the high-grade glioma. Usually the low-grade gliomas are grade 2 and 3, which largely consists of astrocytomas, oligodendroglioma. And then grade 4, which is a high-grade tumor, it's usually known as a glioblastoma. It is one of the most lethal cancers. People die in about 14 invariably they die in 14 months. It's a very aggressive tumors, and no cure has been, or no reasonable progress has been made on gliomas in glioblastoma for the last 40-50 years, despite there is little progress in terms of chemotherapy and stuff like that. There's not been much progress on that, right? So we know, and then the TCGA, the Cancer Genome Atlas, started with understanding of glioblastoma as one of the key projects in there. And so we know quite a lot about molecular biology of gliomas from a DNA, RNA, and protein and epigenetic levels, right? We know a lot through the sequencing, doing bioinformatics. We have tons and tons of data now in TCGA on glioblastoma, right? But then the problem is that people invariably recur, right? Even though they are given chemotherapy, at some point they get the tumors again, and so we really don't know about what happens, how the relapse happens, and so we thought there should little is known about primary tumor and recurrence, about progression, resistance, metastasis, and so many other things. So that's why there is something called glioma analysis consortium, called the GLASS consortium, was formed to understand glioma from a recurrence and a progression perspective. And so the GLASS aims to perform a comprehensive molecular profiling of matched primary and recurrent tumors from 1,500 patients. And so this consortium includes investigators from 14 academic hospitals and universities. MD Anderson is one of the core sites for this GLASS consortium. And this GLASS consortium, when I joined, I'm a part of the GLASS consortium. It's led by investigators in here. So I thought the best case for defining a systems framework should be to do a GLASS, use the GLASS data that has already profiled tons of tumors, both primary and recurrent tumors, and study how the tumor progression is happening in terms of both bioinformatics and systems biology perspective. So what we know about recurrent gliomas is that the first published study, they analyzed about 222 patients with glioma. So they analyzed mutations, copy numbers across a variety of molecular subtypes of glioma. And there are three major molecular subtypes of glioma. One is IDH caudal, IDH non-caudal, and IDH wild type. So what we know about is that the study also said that, okay, driver genes are initially selected and then they were retained at recurrence, but there is little evidence of recurrence-specific alteration. So most of them happen at the primary stages. And these are some of the inference that they have derived. The alkylating agents introduce what is known as a hypermutator phenotype. And then aneuploidy, abnormal number of chromosomes, is frequently detected in recurrent gliomas. And then the clonal architecture of each tumor remains similar over time. And no difference in the level of, from the immune standpoint between initial and recurrent tumors, there's no difference. And these are the inferences that was derived. But the thing is that we have to understand these are done just using the basic bioinformatics. They have not done any data integration across several types of data to understand glioma recurrence in a very integrated fashion, right? So that's the key thing that we wanted to study again. Is what is missing is that the key thing is that they are what they have understood from doing some rudimentary bioinformatics is that it's largely stochastic, right? But then we have to integrate gene expression data, we have to integrate methylation data, clinical data. Think about the initial, the elephant and the blind man slide, right? I mean, we did some things separately. We did gene expression analysis separately. We did DNA mutation separately, methylation separately. But then we have to put it together to understand it. And how do we do that, right? That's the goal of the project. We wanted to create a unifying, again, to reiterate what we wanted to accomplish is that creating a unifying computational framework to integrate all available patient-centric tumor recurrence data to generate hypotheses in an unbiased manner for target validation and predicting disease outcomes. So this is the systems approach, not the regular traditional individual bioinformatics approach, right? So, so how do we do such an integration? So here comes the meat of the idea of how are we going to integrate, say, very disparate data, let's say the gene expression data with mutation data or methylation data with copy number data. How are we going to even integrate all of these things, right? So that's where what is known as graph databases come in, okay? So you might all have a known graph. By graph, you might have meant a pie chart or some kind of a pictorial representation, but that is not what a graph is. From a mathematical standpoint, it's a name for a network, right? It consists of nodes and edges, right? So in traditional databases, you have these separate tables, and we are trying to infer things together, right? So you have mutations, let's say you have a copy number table and a methylation table, and you join them, try to do some understanding based on the mutations, copy number, and methylations would be done based on some bioinformatics, right? I mean, there are mutation callers, some pipeline, GATK pipeline, and so many other things, right? But then there is, there is no explicit relationships that are embedded into these tables, and so you have to infer these things separately, right? But graph databases, the relationships are defined, right? Think about Facebook, think about Amazon, think about all these things, right? I mean, how do they even work? For instance, Amazon, you buy a product and immediately says that, okay, this product also, what do you mean? When you buy this product, people who bought this product also had bought another product. So how do they infer that? In a real time, right? There is no person sitting there and trying to analyze your product and then try to match up with other people's product who also purchase the product, right? I mean, these relationships are already embedded in here. And so it's just a matter of query, right? There is a system already in place that has these relationships defined. And so it immediately points out to the product people who bought this also bought this. It's an association, right? Those associations are already mapped. Same with things happens with Facebook, right? A friend of a friend. Friend is a relationship between two people, right? So when you have is a friend of can be embedded so that it can be naturally rendered in real time. And similarly happens with Twitter. And also many new technologies are just based on these kinds of graph databases. So the graph architecture, the integration is natural, and it's an unbiased analysis becomes very, very easy, right? So all you need to do is define the relationships, and so we can write appropriate queries to answer that, right? So is this idea new? Not really. If you think about this idea, it's already been one of the papers that was actually a very good paper. In fact, that was key for me to develop this. Is that they have already done this in what is known as a colorectal cancer. They had these mutations and that they have these relationships defined. And then they have tried to integrate all these together using the graph databases, right? And so the problem with their model is that they had to create additional relationship for new data. Let's say that you're introducing image data. So they have to introduce new relationship type. And also, it results in restricted survival analysis. We will go over that a little bit later. So so then there are problems with the existing approach. The key attributes of the system that it should be integrative. You have to put in all data that you can find, and it has to be unbiased, as well as it has to be absolute in time. So these are the three characteristics of the system that we have to develop. And so, I was thinking about it, and the idea is very simple. So you have two molecular events, and you connect them, saying that if there are patients who undergo those events, then that forms a relationship. So the patients are the relationships in our model, right? So in our database, there is this basic representation. There are these molecular events, and you connect them if there are more than one patient who undergoes those two molecular events, right? So with that, we built this graph database where we were able to integrate copy numbers, where we were able to integrate methylation, mutation, expression. And if you are having imaging, we can integrate whatever data that you can get, we can actually put them in this database. So this is in this database, we call it as Graph Engine for Systems Medicine, or otherwise Genome. In this database, they have in this database, it's an integrative database. And so we would be able to query anything that we want, right? So there are 370 patients in this database so far, about 11K nodes and 16 million relationships. And this is a Venn diagram of how the distribution of the data looks like. So why bioinformatics critical? Now that comes to the critical question for this, right? So like I said, garbage in, garbage out, right? I mean, it depends on how you define the system. And it's very important when you are constructing such a systems biology approach, it makes sense that we have to make sure that we really put in all the things that are important, right? So we make sure there is a good bioinformatics that is built in developing this system. So for instance, identifying mutations, right? We identify key driver mutations. So there is a pipeline already. We have alignment and GATK variant calling, and then we do the driver passenger analysis and do the mutation, driver passenger mutations, and then we upload it into the database. So it's important. Otherwise, it's all garbage. There's going to be millions and millions of data points. So this is where the bioinformatics is critical. Before it's the next, the whole systems approach is the downstream of bioinformatics. That's critical to understand. And there is, secondly, there is this patients. There is another filter in there. It's determined by the number of patients who undergo the molecular events. And also we have a robust statistics and connected events as another filter. And then we do a random walk and survival correlation. So these graph databases have a survival correlation. So like I said, right, two events are connected by patients. So a patient trajectory A could be different from patient trajectory B, could be different from patient trajectory C. So they will have different types of survival. So in general, we could enumerate all type of paths that's going to give different survival, but that's going to be an extremely hard problem. It's an NP-complete problem in computer science. So we will not be able to do that. So what we do is that we simulate a random walk. Random walk is like how many, it's just a tossing the coin. So if it's like, say there are 6 paths in here, if you throw a die and if it says 5, you take path number 5, etc., etc. So in general, we would be able to simulate a random walk and then do the survival correlation. So for instance, in this, one of the walks in here, we have the path number 51 and path number 54, right? They show the survival difference between the patients. And what you see is a bunch of different biomarkers or genes. That's a combination, right? It's not just a combination of it's just mutations or copy numbers. It's a combination of these that gives the difference in survival. So in this way, we would be able to determine the survival of patients. And this is another random walk simulation too. So we have these 2 paths which show completely different survival times. This is random walk 3 for another specific type of glioma, where we see the survival difference between the patients for path number 16, patients who take that path and patients who don't take that path. So the power of this approach, we have identified several functionally important genes for conducting biological experiments. So for instance, there is a recent study that says that glutamaterate synaptic signaling, it's important, reelin signaling. So we have identified all of these things. In general, right, this provides a strong framework for systems biology paradigm in medicine. It's patient-driven than patient properties-driven. It's truly unbiased and integrative. It's not subject to time. As long as there are patients, we are going to have this framework, and it's a correlating with survival, combining multiple markers has not been attempted. So with that, I would like to acknowledge the support I have got, the patients and their families. My student, PhD students, he has been actively developing the database. Dr. Hughes, Puduvalli, and then Dr. Lang, neurosurgery chair and neuro-oncology chair, they provided an unwavering support. Of course, my senior vice president and CTO of MD Anderson, he was like totally in sync with these ideas and helped us in providing all the support in here. Moonshot gave us the funding. Brain Tumor Center gave the resources. Of course, the database support at MD Anderson and the IT staff, we were available anytime that we wanted to go and had some problems. And of course, this was done on Neo4j and Neo4j team. Rob Martin, Fanny, and Greg Shirley, they were critical in providing the Neo4j for us in developing this system. So with that, I would like to end my talk. and open the floor for questions.
Very good. Thank you. Thank you, Doctor. We do have several questions. If you want to go ahead and pull your screen off, we can just see you instead.
Oh, sure.
So
yeah.
one of the questions is, yeah, we'll even make you get me off the bottom here. We don't, well, yeah, there we go. So is the database you're describing being used by clinicians right now?
Yes, they have been utilized by clinicians. We are testing out certain biomarkers that were derived out of this database. Yes.
Okay, fascinating. So how important is it for a clinician to be aware of the data analysis methods to use it properly?
We wouldn't expect too much insight. I mean, for clinicians to understand the database and the queries, which we will handle. All I think now what we would expect for the clinicians to know is that we can provide some actionable targets, and for them, if they understand a little bit of biology, to see that what makes sense and what doesn't make sense, for them to conduct some biological experiments. I think that would be fantastic.
Okay, because it's probably really important to know where and how the data was accumulated so they can use it properly. Is that right?
Yes, of course. Yeah. I mean, yeah. So we have to develop systems like this for each and disease types. Yeah.
Thank you. Do you see any challenges that need to be addressed to integrate data when it does not come from the exact same patient, but rather different individuals?
Oh, yeah. I mean, this database is about different individuals, right? It's about population. So it's not just one patient. We have about 370 patients in our database, data that we are integrating. So yeah, any type of data, any type of patients, we should be able to integrate this database using this approach.
Well, thank you. Thank you for your remarkable work that you're doing with cancer. I loved all the metaphors throughout, the elephant and all that. That's pretty good stuff. We're going to ask you to stay on and join your fellow presenters from this session for a panel review. So if we can get all of our panelists to be coming back on, I first want to remind the audience to register at learnomicslogic.com/learn or /projects for your free learning portal account. And if you forgot earlier, I'd like to encourage you to do that now. I'll put the link in the chat box here so everyone can see it. If I get the setting right, here we go. So you can make sure you get there and click on the register free button. So if you can all do that, that would be great. And we need to find Sarah to bring her back in. So Sarah, if you're out there, if you can come back in, we'd appreciate that. And I think we're ready to roll. So gentlemen and ladies and gentlemen, there we go. There she is. We've got Dr. Kasturi Kannan, Associate Professor at Translational Molecular Pathology that we just heard from. Dr. Sarah Zapico, an Assistant Professor in the Department of Chemistry and Environmental Sciences at NJIT. Mr. Ilya Brody, CEO of PineTech Bio in New Orleans. And Dr. Oskalov, who we heard of from earlier, Senior Bioinformatician at the Department of Biology, Lund University, Sweden, and the Science for Life Laboratory. Ladies and gentlemen, the first thing I'm fascinated to know is how and when you decided to get into this broad field, and of course, your current niche. And I'm going to call your name. Ilya, let's start with you.
Thank you. Yeah, so I guess, I came to this field just because I was interested in how data analysis could be applied, more in a more applied way, in more biomedical research kind of application. So I think that's the major role. And we talked about how this is being used, to treat cancer, to study different important things that I think are really much more concerning than, where a lot of things in data science are being, used like marketing or something like that. So it seems like a much more compelling reason to apply these skills to these kinds of data sets.
Cool. Sarah?
I realized, yeah, I was mute. Well, yes, in terms of forensic science, as I said before, I always wanted to be a forensic scientist. So I switched after my PhD. So particularly the application of these technologies is because I see the potential for the field. in terms of, for example, in challenging samples, identification of human remains. I'm sure that although they are not as using as everyday needs, but I hope in the future we can use it in a regular basis. So, for example, the phenotyping, it would be really helpful to identify human remains, which I mainly what I do, and yeah, other things. And for example, in terms of post-mortem interval as well, we have really a need for technologies, and I think based on our previous research, that is what we need to apply these technologies.
Just an observation before we go on, Grant. I just noticed that, we had people here that dealt from childhood cancer, right? So all the way to kind of post-mortem studies. So it's kind of interesting, but there is a distinct difference, right? So there is applied and then there's research. So maybe as you describe your, how you got to this, maybe you could mention, so I understand, Sarah, you're mostly doing research with this, right? It's not actually being used?
Yes, I am doing research right now. For the moment, it's not applied yet to the field. So with my some research, what I did in age estimation, we were able to apply to 1 or 2 cases. But again, it's difficult because of the legal problems. Basically, and also it depends, for example, when I talk about phenotyping, for example, in New York, it's really difficult to apply in cases to determine, for example, who was the physical characteristics of the perpetrator. So for the victim, it's different, but still you need to ask for permissions, etc. So here also the problem is legal issues in terms of with the genetic genealogy, although I am sure you heard about a lot. You have still to ask for permission to use genetic genealogy, and at the end, again, you need to do the SPRs, the full DNA profile. So yeah, the problem right now is the actual application cases, but slowly we are trying to do it. So yes, is our main limitations.
Fascinating. Absolutely fascinating. Thank you, Sarah. Nikolay? You're muted.
Oh, is he a co-host or maybe? Yeah.
There we go.
Yes, sorry.
I think there you go.
Sorry, I was trying to unmute myself. Yeah, so I'm originally from physics, and yeah, this field. I guess you mean how I moved to this field, broadly speaking, to life sciences. I met medical doctors, clinicians who showed me their data, and I got fascinated by the amount of open questions. Basically, compared to physics, I would say. You feel in life sciences, in biology, in biomedicine, you feel like a child in a candy shop. There are very many questions. In physics, you kind of have a stable knowledge accumulated over many ages, basically. In life sciences, it feels differently. There is very little well-established knowledge, which is good for researchers because it's we are curious. We want to know something new, but maybe it's not good from the point of view of patients, because we would like to have more knowledge, of course, and help them.
Okay, thank you. Thank you much. Dr. Kanan, we'll come back to you.
Well, I mean, like I said, I was in a medical research institute where I was doing some core microscopy, and then, I've been a computer scientist, so in a biology institute. So I think it was a logical destination for me, to do some bioinformatics. And most of the bioinformatics that I did was I learned ad hoc. I did not have any formal training per se. It's like I'm very curious to know and to understand. So So most of it is like trying it out and seeing what it is and stuff like that. And initially I have no idea of what the file formats are, even when I was introduced to SAM and BAM files and things like that. So, but most of the stuff is like, for me, is about being curious and trying to figure out, right? I mean, like using computers, I mean, the terminal was at hand, the tools are there. I mean, nothing really stops me from identifying or, discovering stuff on my own. So, I learned it on the fly, I would say, and it's, yeah.
Very cool, very cool. We want to touch on careers in bioinformatics and data science. So open this up to the group to discuss as you will, how bioinformatics has revolutionized research. What skill sets are required for students to excel in these fields, bioinformatics and data science?
Yeah, so maybe I can briefly start. I think what is interesting to see is that there is this diverse range of data types and approaches, also depending on where they are used, right? So as we saw today, maybe in a clinical setting, it is very useful to have that population level view, so databases and finding the connections between clinical and omics data is very important. In other cases, I think that, what was explained by Nikolai is very applicable to the type of data that just is starting to be generated, right? This is not some widely adopted translational research that is being described. These are cutting edge algorithms and methods that still need to be developed and validated, I would say, right? So I think that depending which direction you go, bioinformatics is becoming one of those domains like data science that becomes, it diverges like a tree into many different domains. So knowing where you're going with this, are you going to go to clinical research? Are you going to industry? Completely different story. Or are you going into research, and then which area of research, I think really will determine and help you understand what kinds of skills. But universally speaking, I think we can all agree that, coding, databases, these are going to be your tools of the trade. And so you should excel and know how to do that. And depending on whether you're a developer versus user, that's also going to determine how much of math and statistics and kind of the underlying algorithms you would need to understand and, potentially develop on your own.
Who else would like to jump in on that?
Okay, let me jump in on that if that's okay. So one of the things is that I feel that math and not math, forget mathematics, but statistics and computing, right? Those are the essential skills that I think every high school student needs to learn, because the applications are enormous. So you can get into any field that you want. So fundamentally, if you're learning statistics and programming, whatever type of programming, I think those are critical. That we can build upon biology or bioinformatics for that matter, right? I mean, if we choose to focus our efforts of programming and statistics in biology, it becomes bioinformatics naturally. So those are the critical skills I think any aspiring bioinformatician or should have, or anyone for that matter should have. And the second point that I would like to focus on is actually network science, learning some network science. I mean, we know that the data are intricately connected and the world is getting connected more and more. So learning some network science will go a long way in applying the learned bioinformatics into clinical practice. So I think these are the two main things that probably that people should be focusing on, statistics and programming, and the second is network science.
Interesting. Nikolai or Sarah? Y'all can just stay unmuted if it's easier for you. You don't have to keep muting. It's up to you. Sarah, go ahead if you got your mute button ready.
Okay. Yeah, I understand. So actually, if you ask me my point of view, I was a student for biotech, as you know. So I learned a lot. I mean, again, you can be a developer or a user. So I think depending on what you want to learn or what you want to apply, you can be simply a user. I mean, is what I am learning from these courses. So yeah, I think the skills again, one of the important things, at least for me, when I choose students, is their interest and willing to learn. At the end, is the most important thing, and you can learn everything if you are interested in the subject. So yeah, not so much helpful, but I think yeah, this is the point of a researcher that I am mostly a user in this bioinformatics stuff.
Nikolai?
Yes. So what skills are important? Yeah, I would say 3 types of skills: biology, understanding of biology, programming skills, and mathematical background is also good to have. Yeah, I basically see maybe I would say 2 types of bioinformaticians. It can be biologists having a wet lab background who learned computing, who learned programming and math to some extent. And there are also computer scientists who come from a different angle. So they can do linear algebra maybe, but they not really understanding cell biology. And both, I think, can be learned. So basically people with mathematical background can learn biology to some extent, and people from biological background, they can learn math to some extent. So I think, but talking about Lund University, I would say we still have a lack of bioinformatics training. So maybe because bioinformatics is still, it's a new discipline, so maybe there will be more bioinformaticians, but we are kind of still there is no formal training in bioinformatics. It's, as I said, it's either biologists who learn math and programming or computer scientists who learn biology, but there is no like a separate education in bioinformatics, unfortunately.
Maybe to just pick up on that, I think what we heard both from Kasturi and from maybe Sarah as well is, you know, not always is it about those technical skills, right? Sometimes it's that curiosity, that need in your project that really brings you to the point of learning and mastering a skill that you need at the moment. And maybe another perspective on this, bioinformatics is a very diverse field. So it has changed significantly. Today, we heard about how it's continuing to change. And we know that the pace of change is just going to increase. So I think that's another component, right? You have to be driven by your ability to be curious, find answers, but also overcome challenges. And that, I think, is the reason why you need the algorithmic perspective as well, is you need to be not only solving challenge like a tutorial that somebody gave you, but really trying to kind of solve the challenge maybe in a new way, thinking out of the box. You're muted, Grant.
Sorry about that. What would you say to a chemist trying to join the trade? That's from chat. Nikolai, since you were already up, let's let you take that one. You start in on that one. What would you say to a chemist coming into the trade?
Yeah, I've seen people with chemical background, with a background in chemistry. I think they have a relatively smooth path to biology wet labs, because they know all the routines and they have similar, I would say, similar standards. I mean, it's not uncommon. I think chemists who start doing protein research, I've seen many people who migrated from chemistry to biology and computational. I think in chemistry there is, I mean, these guys, they have a good education in, I don't know, quantum chemistry. So it means they understand some computer. They do have good skills in. They should, in my understanding, they should have good skills in computational work. So I see no problems for chemists migrating to bioinformatics.
Cool. Well, let's switch over to what job opportunities and internships for students and researchers there might be. And Sarah, I think, did you have a job available or something like that? If you did, we'll start with you and then we'll hit the rest of the group.
Yes, actually, I have a postdoctoral position. In principle, for a bioinformatician, but I am open to biologists, biochemists that are familiar with this data, precisely because we are going, with the different projects I have right now, we are going to work a lot with omics. For the moment, with transcriptomics and metagenomics. So yes, someone who knows how to analyze this data. So it doesn't need to be a bioinformatician, but at least biologist, biochemist who knows how to deal with this data. So yeah, the post, yeah, I think, yeah, it's already posted.
Great, I think Ilya just posted the link, so thank you for that.
Thank you.
More generally, what do you think, guys? Job opportunities, internships for students and researchers?
Well, okay, maybe I can jump in here and just say that I think it's very critical for people, the students, to have opportunities to intern and maybe even for free participate in a project, right? Because many times when you are taught bioinformatics in a very standard way, you don't have the opportunity to see the project as a whole and really know how useful your work is. So I think internships and especially mentorship are critical. So I know that Kasturi, you do a lot of mentorship. I think we also spoke with Nikolai, you do some mentorship as well. I mean, maybe you can add on to this. How does this actually work in your organization? Is there a process? Do you see where it's very critical? At what stage of that learning process are these, is it a critical component? Kasturi, do you want to?
Yeah, so I mean, one of the things that we are trying to do here is
who can talk analysis, and then it's important for them.
Nikolai, I think you were on mute for a while, and Kasturi started speaking at that time. So,
I'm sorry. Go ahead.
No worries.
Oh, okay. No, I mean, I think, one of the key things that I find it challenging, I mean, in terms of, as you saw that we are developing this systems framework, which is heavily dependent on bioinformatics. And of course, I'm mentoring high school students as well as the key thing is that sustained effort is critical, right? I mean, on this, learning some bioinformatics. I mean, there are opportunities, very good opportunities, especially the research that we are doing is actually translational. It's directly impacting patients' lives in here. And so, there are these challenges, which I say that, it's about like getting to, like you exactly pointed out, right? I mean, it's not just about trying to learn, but also navigating the difficulties that are, on the way, right? I mean, having some troubleshooting the errors and persistently doing that. So, I mean, I think these are critical skills that are needed for anybody. But then, yeah, we are training people in there, and so yeah, our program is expanding. So, what was a critical key question in this? Whether I think I'm missing the point here. What were you asking? Trying to ask?
What the job opportunities and internships for students and researchers might be, either specifically with your organization or generally.
So, yeah, so as this program expands, obviously there is going to be, as I see the future is going to be more systems oriented, and already there is something called a Breakthrough Cancer Foundation that has announced about $500 million for integrating data and data integration, and these are going to be the key future prospects. So I think if students can get trained on this aspect of bioinformatics, where they could not just learn how to do just the pipeline alone, but how to integrate together to have a meaningful patient-driven systems, then combining with the databases like the graph databases, the Cypher query language. I mean, Cypher, the Neo4j has been like I'm seeing there are lots and lots of opportunities that are coming, not only in the bioinformatics space but outside, that are requiring this graph databases knowledge. So I think those combinations will go a long way in securing a good, internship and job opportunities for students.
Go ahead, Nikolai.
Yeah, so within our organization, the National Bioinformatics Infrastructure Sweden, we have a bioinformatics advisory program. So all PhD students in Sweden and Swedish academic institutions, they can have a bioinformatician, a senior bioinformatician as a mentor. And it's very important. So I found it very important for the PhD students, because very often those guys, they come from they don't have a good environment, don't have a critical concentration of people who can talk analysis, and they need to discuss analytical tricks with analysts. And sometimes I see that people are sitting in the clinics, they're embedded into this clinical environment, but they want to do analysis, and then for them talking to other analysts is absolutely critical, and this is what we are trying to help them with this bioinformatics advisory program. So it is very critical to interact with other people who are doing analysis.
Fascinating. That's really cool stuff. That's a nice program. I want to ask how the interdisciplinary fields are integrated with multi-omics, if you guys want to pick that one up.
Well, I was actually surprised to hear about the way that Sarah is using multi-omics. I think so. Here we have 3 different ways that multi-omics was described, right? Sarah was talking about just using different omics to estimate the same parameter. And then Kasturi was talking about building a database that has those connections between clinical and maybe multi-omics. And then Nikolai was actually talking about integrating them to see the same experiment, but just from different perspectives. And so I think these are very unique, different types of challenges. But I mean, maybe it's a question whether multi-omics is a must. Is it just something that happened to appear, or is it something that is going to be like a replacement of a single omics? I mean, what does everyone think about this?
If I may go answering that, I mean, I think it's a natural thing, right? I mean, whether we have to subscribe to the single, like I said, right? I mean, it's about reductionism versus systems. I mean, reductionism is not going to go. Studying single omics is going to be there. There are lots of things to uncover. For instance, doing single cell sequencing. I mean, this is about on a single cell level, right? I mean, so it's not yet integrated with a bulk population level. So that's there. But then if we want to understand in a very population-centric way how we can put together to have understanding, in a holistic fashion, then obviously multi-omics is the way to go. I mean, it's ultimately the difference in the philosophy, whether we are going to go reductionistic or whether we are going to subscribe to systems. I mean, from our 40 years or 60 years of decades of experience, still we have not yet gone to a place where things could be translated to an actionable, where it can benefit a large class of population. So if you ask me, I would place my bets on systems biology. So multi-omics, is a way to go, in terms of understanding and in fact translating the biology to practice.
Anyone else? All right, let's look at some of the strategies. This is the next question that's come up. The strategies to evolve and involve students in bioinformatics and data science. What kind of strategies do we need to approach?
And maybe before we jump into that, sorry, but I see Ryan here has a good question in the chat. What are your thoughts on the interactome and study integrative cell biology? So I think it's a little bit close to what Nikolai was describing in his talk. I don't know if you want to answer that.
Yeah, so by interactome, I think, yes, talking about maybe by interactome, Ryan means sort of network analysis. And the graphs that I was showing, those were graphs where each node was one cell or one sample, one patient. By interactome, people typically mean a different thing. They mean maybe it's proteins. Then the nodes of the graphs, the features, so I think it's not exactly what I was showing, because my graphs they looked a little bit differently, exactly to drive network medicine. Yes, this is what Ryan is asking. I think it's a slightly different focus. How features interact and how they're linked, how they cluster. Basically, as I said in the talk, you have two dimensions in your data matrix, and you can do cluster analysis and look at the links between your statistical observations, patients, samples, or you can do cluster analysis and look at the linkage between your features. I mean, it's two sort of sides. So I'm doing maybe more one thing than the other. So I'm not very much into the interactome, but it's basically, yes, it is another way of looking at the data, and a very interesting way. So
I think that maybe, since we are coming to a close, I just want to maybe spend a few more minutes. I think Nikolai, mentioned this aspect of, you can look at either, cells, or you can look at
a lot of robustness.
How features interact. I think, Nikolai, sorry, I interrupted you because you were on mute again. But you mentioned how, the cell biology, right, is important here, because you are studying cell biology ultimately by looking at these different aspects. I mean, how far away is that disconnected from then understanding how those different features are linked? Let's say the cell develops into a certain phenotype, there's some sort of a, separation of these different cell types. Do you look at all at what defines that cell type afterwards? I mean, you must be looking at what genes, right, or what methylation patterns, so that there is kind of like a way to go further into understanding how those clusters are formed and what defines them, right?
Yes, so first of all, you can hear me, because I think I get muted and unmuted randomly, and it's not me.
I think it's the network. I think your network.
You're good right now. You're good right now.
Okay, I'm sorry. Yeah, that might be on my side. Yes. Yeah, so you were asking about looking at the mechanism. So if you see cells clustering, if you see cells clusters, you would like to understand the what biological molecules drive this clustering of the cells. And if you have the molecular information from the cells, yes, you definitely want to interpret this clustering. So it's a little bit like looking inside your machine learning model. So your machine learning model can maybe find these clusters, but then you want to understand why. It's a next natural question, because you want to get a deep understanding, not just see the clusters, but to understand what they mean. Yes, if you have the molecular type of information from the cells, you definitely can look inside the model and understand why. I hope this is what you're asking about.
And I think that, the next part of what he is asking, the network medicine, that's exactly what Kasturi was talking about, actually, sort of, right? So I guess, as we are wrapping this up, how do these things where do they connect? On one side we have, single cell techniques that generate multi-omics data, and we need to go deeper into that data to really understand what does this mean biologically, and all the way to population health level, to try to identify patients that are very different individuals that maybe also will benefit from identifying these unique clusters of cells that might drive metastasis or something like this, right? So, I mean, where do these things converge, I guess?
Okay. Can I answer this question, or do you want me to? Okay. I mean, I think this is. This is where I think like you exactly pointed out, the cell level. You have these two levels. On one hand, you have the population, and on one hand you have this single cell level, right? Obviously, they have to intersect, and the model that will integrate is again going to be a graph model where we can actually integrate the tables, for instance, that has these specifics of cell, like patients who have those specific alterations. I mean, these could be modeled based on the graph databases. I mean, as I see it, it's not going to be such a huge problem. Right now, the problem is that we are like trying to think these networks are totally different and not looking at a database level, but less looking at just how these connections are from a basic network level, right? I mean, even in network, there's a difference between network biology and systems biology, right? That was my initial question, whether like, okay, why don't we call it as network biology? It's still a biology, but why do we have to call it as systems biology unless we have the power to integrate any type of data that we want, that the graph databases provides us with such a technology to do that, right? I mean, it's a very recent technology. It has a basis on wonderful computer science, right? I mean, pointers and stuff like that. So, it can be integrated, but I feel that the integration will go in the direction of graph databases, and it cannot be just thinking in things in isolation, if you ask me.
Anyone else want to pick that up?
Okay.
Well, not seeing. Oh, go ahead.
Okay. Sorry. No, because of the integration of technologies, I know this is in a completely different field. One of the things we are looking into is the integration and combination of omics for postmortem interval estimation. So it's kind of what I think you are meaning in terms of health, but in the application of forensic science. So I think also it could be the integration of the different omics could help in that.
Nicolai, any closing comments?
Yeah, I understood Leah's question a little bit differently. He was asking population versus single cell, and I know I heard this opinion that typically when we work with single cell data, it's just one individual maybe, and then how does it generalize on the population? And I think it's still early days. We will eventually, hopefully, reach this state that we can sort of utilize millions of cells from many individuals. Sort of the question of generalization of what you discover on one particular sample. It is valid. So maybe all the conclusions might not be generalizable for the population, but still, those are early days. So we just want to see the heterogeneity between cells, and usually it's consistent between individuals. At least the types of cells. I'm talking about maybe blood cells. They are more or less consistent. You see the same cell types from one individual to another. But indeed, there can be diseases where a certain cell type looks strange, and this is what we want to catch. But the more we sequence, the more data we have, the easier it will be to answer a question.
Very good. Well, I'm going to go ahead and close us up. We're at our ending time, and I'm going to put my closing in the chat box also. We want to make sure that we thank our honored speakers today, Dr. Oskolov, Dr. Julia Panov, who was on earlier, Sarah Zapico, Dr. Lisa Taraneko, who was on earlier, Louis Sakamoto, and Ilya Brodsky. So we want to thank you all for being here today and for contributing to this fantastic session. Be sure to join us tomorrow for our second day of learning, thinking, and being challenged by our amazing speakers. And one last reminder, and it's in the chat box to sign up now at the Omicslogic sign up page. You click the register for free button, get your free account, and start learning. Thank you all again. We'll see you tomorrow. And yes, the session was recorded and will be available to those who registered. Thanks again. See you tomorrow.
And I want to mention to Connor, I know that we didn't get to your question, but it's a whole another topic. So maybe we'll pick that up tomorrow.
Very good.
All right. Thank you, everyone.
Thank you all.